Okay. Hi, good morning, everyone. We were just joking that we're in Switzerland, so we have to be starting on time. And the seconds just hit the 9:00. So warm welcome to our 6th Precision Oncology Symposium here at the Comprehensive Cancer Center in Zurich. My name is Matthias Guckenberger. I'm chairman of the Department of Radiation Oncology, but here today as Director of Outreach and Education. This is the 6th education of our symposium, which brings two messages. One is that the first 5 ones have been a success. So we very much enjoyed them. There was an excellent exchange clinically, but in particular scientifically. So we want to continue that tradition and now moving to the 6th edition. And the second most important reason maybe is that precision oncology is the focus topic of our Comprehensive Cancer Center. And so this is one of the flagship symposia we are doing throughout the year. Let me give you a very quick introduction about our Comprehensive Cancer Center. Over there. So obviously, as a comprehensive cancer center, we try to have a comprehensive clinical program. You know that there is some kind of distribution of responsibilities in Zurich. For example, the orthopedics department is a separate hospital. The children's hospital is a separate one, but anything else is located at our Comprehensive Cancer Center and underneath the umbrella of the University Hospital of Zurich. We try to bridge between science and between clinical patient care. That is the clinical science in terms of clinical trials and also the translational and basic research. And obviously also to train, to educate the next generation of clinical scientists. Why are we being here today? Because we also have an ambition to outreach, to educate, to bring researchers and clinicians together. So this is one of the symposia, and we are very grateful that Thorsten has been organizing that. Thorsten, yes, that is you. No one is being behind you, Professor Zenz, who has dedicated a huge effort in preparing this. Just giving you some key numbers. I know it's early in the morning, and so I don't want to kind of stress it too much, but in total, we have 20 tumor boards. We have almost 200 clinical trials, where on average about 12% of the patients are being enrolled, and we have about 2,000 primary patient cases according to the accreditation of the Deutsche Krebsgesellschaft. With that, I would like to hand over to Thorsten. Many thanks again for the preparation. I'm wishing you an exciting meeting, and in particular kind of an exciting exchange in the coffee breaks to build networks and to bring forward our research and our clinical program. Thanks a lot.
Yeah, thank you very much, Matthias. A warm welcome also from my side. I will focus just on some housekeeping before I hand it over to the first session chairs. So we're very grateful for the support from the companies. They're listed here. This is the 6th symposium, and we usually do it a little differently every year. So you're welcome to bring back feedback to us. This is the program. I will not go into too many details here. The meeting is in person. You're here, but there's also people in Zoom. So if you have questions, you can open the silver thing on your desk and push the button, and that will allow people to hear your question here, but also at home or wherever people join. So we also, for the first time, have a poster session. I think this is great. We need to be on time to have enough time with the people presenting posters. So we have about 20 posters from different areas, and this at least for me will be the highlight of the meeting. So with that, the two more points I wanted to make. We've invited a lot of experts from the field, but the challenges we face in terms of improving outcome for cancer patients cannot be solved by any of these experts individually. So we meet to discuss things, but it's one way of collaborating, and these types of collaborations will be needed to solve some of the relevant questions that we will be discussing today. So I'm very grateful that you come. I'm very grateful to the speakers who come. And I'm excited about the day of science and discussion, and that takes me to my second housekeeping. I asked the chairs to be very stringent regarding the speakers, so they need to stop at some point. But we want to have time for questions. So there's the 30-minute slots. So after 20-25 minutes, start making eye contact and then come up, and feel free to cut us off, especially the local presenters. So with that, I will hand it over to Jana and Davide, who chair the first session. Thank you.
Can you hear me? Okay, I'm Deputy Head of Hematology and also Group Leader at the Research Institute. I'm grateful to Thorsten for having invited me to chair this first session, which is dedicated mostly on blood cancers. And now I would like to hand over to Jana, who is the co-chair or chair with me this session.
Yeah, good morning. Good morning, everyone. It's our absolute pleasure to open this very first, I think this is not working. Yeah, to open this very first session of this symposium, and we will have three outstanding speakers, and all the first series of talks will be focused on the definition of clinical needs in precision oncology. I'm an assistant professor here at the University of Zurich and also an attending physician in the Department of Medical Oncology. And it is my absolute joy to introduce Dr. Nastassja Scheidegger from the University Hospital, from the Children's Hospital here in Zurich, to open up our scientific symposium. She will give you a talk about pediatric blood cancers. And she is a project leader on many exciting projects and also an attending physician. And the floor is yours, and we are very much looking forward to your presentation today.
Now, okay, I got it. Thank you very much for this very kind introduction. I'm very excited to have the opportunity to speak to you today, and excited to represent the children's oncology aspect in precision medicine. Let me get laser pointer first. Okay. So cancers in children are different than the cancers that we see in the adult world. And they are not only different cancers, but they also come with a fundamentally different biology. Looking at the survival of the last 50 years, there is still room for improvement. We are mostly fighting with relapse and refractory patients and the late toxicities of treatment that in children, of course, have a heavy value. The current state in pediatric precision medicine was investigated by several groups. A first generation of large-scale pediatric precision medicine studies have been completed. And I want to show you as an example a very big precision medicine study, the Australian PRISM trial, that included over 300 high-risk pediatric patients that were that received deep molecular profiling and discussion at a molecular tumor board. Of note, there was no functional diagnostics included in this diagnostics, and immunotherapy were excluded. From those patients, 67% received a precision-guided treatment recommendation, and of those 67%, over half had clinical evidence tier 1 and 2. And of those, 43% were translated, of those recommendations were translated into the clinics, which is actually quite a high number comparing to the other studies. And the nice thing about this study is that they showed benefit in outcome after application of these precision-guided treatment recommendations. So what can we conclude from this now, from those different studies that are now out? What are the needs in pediatric precision medicine? First, we need a broad and standardized diagnostic testing culture and also define our high-risk population in detail. In Switzerland, this is actually a challenge. The Swiss pediatric oncology field is quite fragmented, and not all children receive the same diagnostics and access to personalized treatment at the moment. We also need robust preclinical testing for relevant drug identification with different models. Actionability, so we need to find out which targets are relevant for efficient treatments, and therefore we need comprehensive data collection of outcome data, which we still lack. And we do also have at the moment limited access to drugs, especially in the pediatric world, due to many different reasons. Therefore, the international network is very important, even in children where cancer is a rare disease, to get access and to reach statistical powers of all the options we're giving. So in Switzerland, to address some of those concerns, recently a Swiss initiative has been launched, the Kids Can. Application has been fulfilled as an NCCR, a platform to address the needs in pediatric precision medicine. This is led by Jean-Pierre Bourquin. And the Kids Can platform, this would be the first NCCR dedicated to children with cancer in Switzerland. This Kids Can platform is led by Lausanne and Zurich, and aims to stream the clinical and cancer biology questions to provide new solutions and optimize options for children with cancer. An important aspect of this Kids Can platform is the drug response profiling. Under the roof of this DRP, we are now harmonizing the different entities of childhood cancer, leukemia, brain tumors, and solid tumors. And the fundament of this DRP is the assay, the screening platform with single-cell deep imaging, but also data science unit that is scaled by the BioVision Center that you will hear more about later. And also the clinical trial unit that is crucial for integration into international clinical trials. The current state of our drug response profiling platform is depicted on this, for hematologic diseases, is depicted on this graph. First, our team on the right, with Fabio Steffen as the lead of our DRP team. In short, the workflow of the platform comprises the collection of leukemic cells from bone marrow or blood, and cultivation of an stromal cell culture of immortalized mesenchymal stromal cells to mimic the bone marrow environment. Compounds can be added and incubated, and the readout is a viability stain or a leukemia-specific stain with automated microscopy and the AI-assisted image analysis. We receive IC50 values or area under the curve values, which then helps us and supports us in prioritizing the drugs for the patients. And this all happens in a turnaround time of around 10 days. We have already screened over 400 patient samples, and mostly the lymphoblastic leukemias depicted here in blue, but we are picking up on myeloid hematologic malignancies like AML and MDS. And we have implemented the platform in Kolkata in India, where it is working and is implemented on a daily basis. And we are at the moment building up another platform with our colleagues in Frankfurt. The patient samples, they are from Swiss or international patients, adults and kids. And we have now also integrated DRP in the international first-line pediatric AML trial, CHIP AML, in the Nordic consortium that we are also part of. And we are also running a pilot study with, within the European Consortium for Myelodysplastic Syndrome for Children, and I will show you some data later. So screening a large amount of patients gives us the opportunity to build a landscape of drug activity across patient samples and across different drugs. As you can see here in this huge heat map, where we can recognize from different disease stages the drug response of different patients. An important aspect of this DRP platform is the accumulation of a big cohort. So not only the sensitivity, the ex vivo sensitivity to a certain drug is important, but also putting it into context to the response within the same disease group. So, for example, you can see in this patient a high venetoclax sensitivity, but not just activity of the drug, but also in the context of the whole cohort here depicted in the different dots. This patient stands out for its venetoclax sensitivity. And correlating this to clinical response, Fabio Steffen and others have collected a nice data set of 28 patients that all received DRP, including venetoclax, and after that were treated with a venetoclax-based treatment. And we see a cohort with that achieved complete remission after this treatment, and they also, this correlated with a high drug sensitivity to venetoclax, a low AUC. So this really gives us the opportunity to determine specific drug sensitivity borders for correlation with clinical response. We also have included myeloid diseases, AML and MDS. And here we face new challenges such as low blast counts. So we are introducing leukemia-specific antibody staining that we receive from the diagnostic flow cytometry analysis. Such as exemplary in this patient that had 20% leukemic blasts expressing CD34, stained with an APC staining here in red. And the drug sensitivity curves then really showed us different drug sensitivities, either looking at the whole cell population in green or only the CD34 positive population. This is even better seen if you look at targeted agents in this patient with KMT2A rearrangement and RAS mutation. We see a great sensitivity of the leukemic blasts to the menin inhibitor revumenib and the MEK inhibitor selumetinib. We also see other specific responses to targeted inhibitors, such in this example of FLT3 mutations. This patient with a FLT3 ITD showed great sensitivity to FLT3 inhibitors, whereas we do not see that in FLT3 wild type cases. And similarly, again, in KMT2A rearranged AMLs that are supposed to be sensitive to menin inhibitors, we can confirm that in our assay with 2 different menin inhibitors. We do see a nice sensitivity to menin inhibitors compared to the other groups. Translating this into the clinic, I have one example that we treated at the Children's Hospital in Zurich. This is a 2-month-old baby with AML and KMT2A rearrangement. He relapsed quickly after the first stem cell transplant. And looking at his DRP profile, he had quite a resistant profile to conventional chemotherapies that you would give in this setting, but showed great sensitivity to all 3 tested menin inhibitors, also shown in this sensitive viability curve. So we had the courage to send the baby abroad for inclusion in a clinical trial with menin inhibitors, and he achieved complete remission and is now 2 years after second transplant in remission. We're not only translating our findings of the DRP into the clinics, but we are also exploring with the data of the screened patients, biomarkers or synergistic drug combinations. As an example here, I'm looking at the AMLs with a high HOX gene expression that usually come with poor prognosis. We did, as expected, to see correlation with menin inhibitors, but we also found a correlation of those HOX-driven subpopulation with XPO inhibitors such as eltanexor. And then checking this in cell lines, we could confirm a synergy with menin inhibitors and XPO1 inhibitors. And to find more out more about the mechanistics and further experiments, you're welcome to visit poster number 4 of Anna Dehlen. As a last example, I would like to show the data in our pilot study with our myelodysplastic syndrome with excess blast patients, looking at a cohort of patients with UTBTF tandem duplication, which makes about 1/3 of all the patients. The pediatric patients with MDS, which harbor a poor outcome, are not responsive to conventional chemotherapy. So we looked at their DRP profile and found a striking, really outstanding sensitivity of the UBTF tandem duplicated cohort to venetoclax compared to the other samples we tested. And this is already investigated in the clinics. Now we are implementing the DRP in the clinical routine, as you see, and decision-making. We have implemented an international federal board within the consortium of the European Consortium of Relapsed Acute Lymphoblastic Leukemia. And this 2-weekly held tumor board, international tumor board, includes patients that have genomic and functional profiling. They are discussed in the international board with international experts, and personal treatment attempts are recommended. And there's a structured follow-up. From the pilot study, from the first year, from 2023 to 2024, we included 80 patients, most of them with follow-up, and we can retract a lot of data, not just DRP data, but also compliance with therapy recommendation, and of course, outcome data, and many more variables. The next steps for DRP include the scaling of the drug response profiling platform, scaling of the standardization, and scaling of the collaborations. We already have collaborations with the Tata Center in India, which gives us not only more knowledge and also more patients. And for this, we need to scale the data analysis platform. We have to decentralize it and make it interoperable. And therefore, the BioVision Center is pioneering and providing this data analysis platform. And I'm very excited to hear the talk of Virginie Uhlmann this afternoon. So for my last slide, I would like to conclude that we are in the process of harmonizing and providing a state-of-the-art precision medicine platform for children through the KidsCan pipeline. The drug response profiling provides added value for patient care. We are in the development of further structuring this precision oncology setting with implementation of tumor boards, with inclusion of DRP in observational studies to validate the DRP. We have validation ongoing in a first-line AML study, and we are preparing a study in TALL in a joint effort with our adult colleagues. And we are taking baby steps towards certification. And of course, we're constantly further developing the assay and the bioinformatic of our DRP with a next generation DRP+. So with this, I would like to thank you for your attention, and I'm very happy to start a discussion.
Well, thank you very much, Dr. Scheidegger, for this comprehensive discussion about this platform for precision oncology in children blood cancers. Is there any question from the audience? One.
Hello, beautiful talk. It looks very promising, especially for hematological diseases. But you were mentioning also that you're taking it to solid tumors, such as sarcomas, I think, and brain cancers. Will you use the same platform, imaging-based single-cell resolution?
It is, in the end, the same platform with still optimization efforts to optimize of course, the culture conditions, and just the whole biological setup is much more challenging there. But the aim is really to standardize at least possible things, to standardize, obviously not the exact same culture conditions, but the bioinformatician, the imaging, etc. Yes. Yes.
Are there more questions, Thorsten?
Yes, thanks a lot, Nastassja. So the UBTF translocated MDS, that looked pretty clear. So have you looked at BCL2 expression? So is the explanation that somehow the translocation leads to unusually high BCL2 expression?
We have not looked at it. There are a few papers, because it's also prevalent in AML. That's why I'm thinking, I'm not aware of the data, but maybe someone else is. Yes, I'm not 100% sure. Mm-hmm.
Additional questions? Yeah.
So I have another one. So obviously your team has huge experience. You have 110 drugs, and then you can you pick some of them. But I assume for some of those drugs, this particular assay is not, like, somehow the drug doesn't work. So my question is, so what's your kind of least favorite drug that you would say you guys always disregard? Because we always zoom in on the examples where it works, right? But for the community, it would be really very important to also look at this information. So what's the spectrum of drugs where this is not very reliable to predict what would happen with the patient?
I mean, there are a few important drugs that where we do not see a signal. Typically, is methotrexate, busulfan, and we're not giving up completely. We try different concentrations. For the menin inhibitors, we had to increase the incubation time to 6 or 7 days. You might even think of changing the readout, for example, in AML to differentiation markers and not apoptosis. You still have options, but there are definitely there are some drugs that are not active. They're not in an active state because they have to be metabolized through the liver. So there are some challenging drugs that we have to exclude. That's completely correct. Some are really grateful, such as venetoclax with a short incubation time, working really in the apoptotic pathways. But there are some that we do actually even exclude. Yeah.
Sorry, another question. So in the molecular tumor boards, how do people receive this information in terms of drug response testing versus genomics? Like, are we in a transition period where genomics is not needed in 20 years, or
in our international tumor board?
Yeah.
So the clinicians actually send the data they have. So if they did NGS, they do it themselves. We don't provide that information. What was the? Sorry, I didn't.
No, I'm just curious. Like, is there a transition period where we don't need molecular data in a couple of decades?
Yeah. Okay. I mean, there is a lot of data we don't understand. I would say if we maybe need it in a few years, I don't know. Yeah.
I also had a question because you mentioned that you're using this platform for several tumor types. Is the drug selection always the same, or do you adjust it to the disease entity?
This was just a discussion that was done by the DRP team. So every of those tumor entities have their experts, of course, and we just recently looked at a whole bunch of drugs, and we set for a bunch of drugs that will be tested for all the different disease entities, and then each group has their favorite drugs they would add, so we could combine this in the plates with quite a straightforward way. Yeah.
Thank you.
I have a few questions. One is technical. You said that your readout is apoptosis, right? But some drug may have anti-proliferative and not apoptotic effect. I come from the field of CLL, where if you test glutathione kinase inhibitor in this way, you will not see great signal, but instead they are working very well in patients because they are anti-proliferative. Do you plan?
So our readout is a live stain, and so therefore what we did try is for AML look at differentiation markers. But this is still in a research state. But we do see signs of upregulation of 11B or 14, and but for really just proliferation, you would. I mean, we do check the sample 18 hours after seeding, how it proliferates. So we have a measure there about just the biology of the sample itself, but not of the drug then.
Because, for example, methotrexate may have anti-proliferative and not apoptotic.
Yes.
Then I have 3 more clinical questions. The first is whether you see any unexpected response compared to what you would have predicted based on genetics of the disease, or the phenotype, AML, biphenotypic ALL, MDS. For example, I don't know.
Unexpected more in a way of more resistance, not necessarily. For example, for targeted drugs, we rarely see a response if there is no genetic reason. Or if we see, we really have to.
So the genetics drives also.
Yes, it's rather that they are just resistant to whatever
But also if they are sensitive, for example, for many inhibitors, if you have the translocation of MML2, you see always response, I guess.
You see, for this example, we see some cases that are not sensitive, but this is also true in the clinical trials. And I think there it depends. With the revumenib, we did not see unexpected sensitivity, but with the other menin inhibitor, actually, we did see 1 or 2 unexpected sensitivities that maybe we will hear more about in the future about those
something related to second driver events. For example, you mentioned RAS, and MEK inhibition as a potential target in one of your patients. Did you ever tested this in patients? So going to a drug that is not expected to work in ALL, like a MEK inhibitor.
Yes, we did not translate that into clinic.
Okay.
Yeah.
And I also see that calicamycin is always on top. No, I guess this is your positive control.
No, it's actually not. It's actually really we included this because we wanted to maybe get a sense for inotuzumab and Mylotarg.
Yeah, because did you use them? That's the question, my clinical question. Because if all cases are responding to calicamycin, it's obviously
it's not all cases, but it's a really strong drug, and we actually are slowly retracting it because we now can test inotuzumab and Mylotarg itself with a specific staining, and we still have to check how the correlation to calicamycin then
but in patients, did you ever translated this? So since
calicamycin, no, but
I mean inotuzumab and genotuzumab.
There we have a small set of patients that received inotuzumab, Zurich patients, and the response seems to correlate, but it's 5 patients, so it's not a lot.
And final, very brief question: when we use venetoclax, did you use it as a mono or as a combination with chemo? Or
I mean, mostly in combination. Yeah.
Okay.
Yeah.
Any further question from the audience? Well, if not, thank you very much.
Thank you. Thanks again.
Now we can go ahead. The next speaker is Thorsten, Professor Zenz, the host of the symposium. And he will talk about INTeRCePT, brackets lymphoma, close bracket. Thank you very much, Thorsten.
Yes, thanks, Davide. Yes, so this is a very good kind of start with drug screening, and what I will talk about is I decided to change the topic a little bit. So I really want to show you many of the programs or projects and thoughts that we are following in lymphoma, and I guess I hope it'll be some food for thought for how maybe some of you can apply some of the ideas to your favorite target or disease. And I wanted to introduce the kind of my view on how does this work clinically. How do we go from a good drug to a better drug? And this takes a huge effort. Right, and it takes a long time, and I guess that's good unless you're impatient like myself. It takes a long time, and also for us, the community takes some time. It needs some time to be convinced, right? And we have the guidelines; they're notoriously slow. It's like until you have all the CLL experts agree on something. So. That's an issue. We usually go by risk factors and standard of care, and this clinical reality that we all have that has a lot of advantages now kind of meets this amazing development in science, knowledge, technology, and so that's really the big theme for what we're trying to do. So I want to illustrate this with a patient that we've treated for some years now. It's an aggressive B-cell lymphoma, and if you treat a patient with this disease, initially you would say, okay, we give you some chemotherapy, some antibodies, and there's a 75% chance that we will cure you with that treatment, very good treatment. But it's an individual patient, and 75% cure means that for 25, this good treatment doesn't work. So then we needed to do something else, some other chemo, some antibodies, and at that point in time, second line, we would have had to say, okay, there's this new treatment CAR T cell, so we have 40–50% of curing you with that treatment. But for this individual patient, even though that's an amazing innovation, the disease didn't respond. And then at the last try, this is DLBCL, which usually doesn't respond to checkpoint inhibitors. But obviously there was a reason for this choice, and against the odds, if you just play it by the general rules, this would not be expected to work, but for a number of reasons that I won't go into, this worked amazingly. So after 2 infusions, you could see that the tumor disappeared, and the patient is now off checkpoint inhibitor for over 3 years, and I think she's cured. So we could now just kind of discuss where are the opportunities from this kind of reality that we face clinically. But I just want to deposit some of the ingredients that I think we have technically to improve this process of what do I give a patient and who should get which treatment in what order. So Nastassja has mentioned it. So we have all these wonderful drugs. So if you want to do omics profiling on the tumor, it may be good to first put the drug and then see how the tumor reacts. If it dies, that's very simple to read out. But also you can see feedback loops. You can see things coming up. So that's a major focus of what we try to do. The immune system is very important. We always, when we consider these treatments, we ask, okay, are there T cells? But I mean, I'm not an immunologist, but there's much more than that. So, and we have the technology. So, monitor does the immune system work, and then how does that evolve with treatment? And this could help us choose treatments. Many of those technologies obviously depend on high-level data analysis and visualizing that data. And many of you here are experts in that field. Like for us, that's a blessing. But there's also a major task for the community to educate people regarding interpretation of these things. And then the last point, I showed you those PET scans. We cannot do this every few weeks. It's expensive, it's radiation. But there's all this amazing technology now where you can draw blood and see how does the tumor disappear? Is there a reduction in the mutations that are tumor specific? So these are some of the ingredients that I think could be applied to any patients, obviously at different levels. So we have for a very long time done these types of experiments, and just a summary. A lot of this was done with Wolfgang, taking a lot of patients, and this is very different from the challenge that Nastassja and Jean-Pierre have, and it's really a complementary approach that reduces the impact of noise. And we really try to retrieve signals from analysis or omics, multi-omics profiling on lots of patients, including perturbations. And this puts us into a nice position to, at some point, we'll have done the right experiment, and this can lead to a discovery. It also puts us into a great position to cooperate, both on a level of sending samples to someone who has a good assay, or sending data to other people. So what do we use the drug perturbation for? Nastassja has talked about the clinical stratification. You test the drugs, and then you make a pick, and then you can see how that improves. You can also use the data for stratification, but really the main focus of the research we try to do is trying to make sense of it, find association with genotypes, but also when you think of many of the small molecule inhibitors that we have, these drugs are very specific. If they're not specific for one kinase, you know what other kinase they target. And then if you do these types of experiments with sufficient resolution and with sufficient patients, you can use drugs as a way to measure pathway dependence, and that's been a big focus of what we're doing. So if you do this type of experiment on a lot of patients with 63 drugs that were not specifically selected for any given disease, we do this at very low concentration. The response to these agents will separate the diseases, and then sometimes it will split up in blue. It splits up these two groups of AML, and so this works very well, similar to what you expect with gene expression. But with gene expression, you're looking at thousands of genes. So it looks as if you take a lot of tumor samples, you take your inhibitors, your favorite inhibitors, and just look at ATP, like how well are the cells, you get an amazing resolution. But now obviously you can take advantage of the technological developments. You can start to look at gene expression in each of the sample as a function of different inhibitors, and that's what Lan did over a long period. And the experiment was done on 116 samples, a lot of it CLL, chronic lymphocytic leukemia, and she measured viability with flow, and then RNA sequencing, shallow RNA sequence to really capture what happens in these cells. These inhibitors sometimes kill cells. So obviously you would expect a dying cell to have a very different gene expression profile, and that's what you can see on the left. The first principal component that captures this dimension of a dying cell. But what you also see is the majority, so each dot is a patient times a treatment. Majority of samples were actually fine. So then you can take this out, and then if you look at the second and the third dimension, on these just over 100 patients, what you see after freezing, thawing, culturing 48 hours and putting all these nasty drugs, you still see the main disease dimensions. For CLL, IGHV, unmutated, mutated, those are the two biggest groups, and that's what's shown in red and green. And then you also see trisomy 12 gene dosage, lots of genes. So this patient effect is still very strong. So now that we've established this, yes, a dying cell can be an issue. We can remove that, and to now look at what do the drugs actually do. We need to somehow correct for this inter-individual differences. So we took some different inhibitors. It's not critical. We can for this particular disease, B-cell receptor signaling is very, very important. So we blocked that. We also targeted some other AKT, mTOR, MEK, a lot of the textbook notes of important signaling. And then we had courtesy to Moshe, we had a p53 activating drug. That's a great tool compound. And we can do lots of things with these types of data. And I will just show one example because Lan gave a nice presentation last year. And we're hoping to wrap up this paper. So the first question we can ask is, how do those inhibitors, is activity of the pathway needed to, do they contribute to this disease dimension? So I said IGHV is very important. So you can now ask, okay, if I put any of those inhibitors, let's say I block the B-cell receptor signal, does that reduce the number of, does that have an impact on the signature? And for some genes on the left, these are marker genes. They don't seem to depend on B-cell receptor signaling. But on the other hand, we found a lot of genes, including a lot of B-cell identity transcription factors that seem to depend on B-cell receptor signaling. So that's an example how you can now answer, how does a given pathway contribute to a major disease dimension? So that's a specific example, but obviously you can also just count the number of genes. So what we're showing here is basically the number of differentially expressed gene with DMSO control in red, dark red. And then obviously if you're less stringent, you will find more genes depending on your FDR. And then the orange or the blue line is how does that number of differentially expressed genes change? And I would say, okay, with inhibition of the B-cell receptor, like the example I just showed you, this somehow collapses. So there's a subset of genes that make up this difference that are dependent on B-cell receptor signaling. And with this inhibitor A, this doesn't seem to impact. So fortunately, we also had the combination of the two, right? So what's going to happen with the combination could just be the addition, right? Could just be, the blue curve coming down a little bit. But what we see actually, because of the nice selection of drugs, we see a cooperation, we see a synergy. So what does that mean? So to me, it means that there must be some crosstalk, like inhibition of pathway 1 doesn't do very much, and inhibition of the B cell receptor does something. But then if you kill both pathways, you start to massively collapse what makes IGHV unmutated different from mutated. So we have many different applications for this data. I think it's an amazing data set that we need to bring out. But I now want to take this bulk data as an introduction to bulk RNA sequencing data as an introduction to some of the other things we're trying to do. So doing this type of experiments with gene expression is very tedious, right? You need to extract the RNA. The data is very crisp, but now with Christoph and Lopa in the back there, we want to do more of this type of experiment, and then the logistics is becoming very important. So here, we would like to move to the proteome, and there's lots of other advantages, right? You can actually measure the client, see the data will be hopefully more interpretable, also because we'll be able to go through a lot more perturbations. So in this Polar project, we are going to run a lot of lymphoma samples on the proteome, but also we're adding something to the original proposal, which is using massively scaling up this drug perturbation. And one of the motivations for this is that there's now a big development in the degrader field. So drugs that don't just inhibit a kinase target, but actually degrade it. And the beauty of the mass spec is that you can actually, it's one-stop shopping. You can actually see if your drug works, and you will see what other targets are deleted. So you haven't interrupted me for questions, but usually the questions come up in these talks are, okay, but what about the stem cells, and then what about the immune system? So obviously we now need to move this whole concept to single cell technology. We're very fortunate that we have one of the big loop projects which does just that, and that's an amazing consortium of pediatrics, us in hematology oncology, data scientists like Nico and Wolfgang, immunologist Burkhard, who's here this afternoon, and Andreas back there, where we really follow very similar concepts, but now the reader is becoming much more complicated on single cell level. And what we try to capitalize on is that we don't just take one sample, but we take samples over time as patients relapse with treatment. We do this drug perturbation. We continue to pinch the cells with the drugs, but then really we're putting all of our money, and it is costly on single cell technology, which is great, but also a major bioinformatic challenge. So these are different work packages. You will see many of these things on a poster. I will go quickly here, and this is basically the setup. So we resample patients, and really the main area of investigation is we take the cells, we put the drugs that we would want to use clinically, or lead compounds that we're interested, like induction of p53, and then we do the same type of experiments, but can now look at what happens in single cells, looking at clonal heterogeneity in the tumor, but also at T cells, monocytes. How do the treatments affect the normal immune compartment, which we need to successfully use many of our new drugs. So this would, if you think back to this initial patient presentation, this also leads to a nice situation where we invest more money in patients where things are not working well, right? If we have someone we treat and the disease disappears, this will be a very finite experiment. But for patients where we have a long list of drugs, we can resample and try to make more of the individual patient. So we've gone through a lot of big projects. We need to run a lot of samples at the same time for a number of reasons, and we've touched different diseases, and you will see some of these on the posters. And then just one summary on one technology from Burkhard and Stephanie. So they use high dimensional flow. So they have different panels, and so you get up to 150 markers that can look at the tumor, but also the immune system. And obviously you're all aware this these funny things going on, interactions of the host and the tumor and the treatment, and I think this will be extremely useful and applicable to all sorts of other tumors. So we've started with mantle cell lymphoma. We always see a similar pattern. From patient to patient, the normal cell compartment looks very comparable. So all the T cells are, they're similar, but the individual tumors are very different, and that poses a challenge to some of the analysis that we need to do. So we have this map of the immune system, and we're also generating similar data after drug exposure, and we're starting to find markers of like what does a responding patient look like, and this takes us back to that very first patient. If we start a treatment, maybe we should be looking at these things early on, develop stronger markers of this is going to work, this is not going to work, and use this information to more quickly get certainty on if drugs work or if they don't. So finally, we are the Loop translational projects also have a big discovery part, but they also fortunately have a part to run a trial, and the trial follows a lot of the logic that I've tried to introduce in the beginning, but it's really focused on this early response prediction. So it's a feasibility trial that we can come up with a better prediction of outcome after CAR T-cell treatment. So CAR T-cell treatment is an amazing treatment, but also very, very expensive, costly, and about 50% of the patients are cured with the treatment. So there's much room for improvement. These CAR T-cell treatments also are funny in a way that it's a living drug, right? And these T cells expand as they fight the tumor, but then at some point in time they're gone, right? So I think one of the key clinical take-homes from this trial could be, okay, but if I can tell after 30 days who has a very high risk, maybe I should, do something while the CAR T cells are still there, and I think this would be very useful. So it's a feasibility study that we can do liquid biopsy, immune profiling, PET analysis, clinical risk management within 38 days, and we have a control arm where we do the same type of analysis, but the lab is not as stressed. So this takes me back to my summary. So I hope I convinced you that yes, there's an amazing potential in pinching the cells with drugs. We're extremely excited about the ability to measure the immune system. This will need a lot of data points, a lot of data, lots of patients, and this last point we really want to use a trial to integrate this ctDNA measurement into into earlier triggering of new treatments or a prediction on future outcome. So to come back to what INTeRCePT and the funding from the Loop and also the Horton Anniversary Grant and other projects so we've really built an extremely kind of motivated group of researchers, but I think we've also put us in the position where we are for lymphoma certainly the global leaders in single cell measurements on these types of tumors. There's this very rapid therapeutic development, so we really need to speed up and make sure that we translate some of these findings, and we also many of you are here. We need to make this data available, but also when funding runs out after 26, we need to get the scientific community here, but also elsewhere to take advantage of the data, because we no matter how we look at it, the data is very, very crisp and clear, but too much for the 5 PhD students in the consortium. So with that, I will close. I thank the INTeRCePT Polar team, also my group. The Loop Zurich is instrumental for the things we do, but the Loop is doing much more than just cancer. And there's also a lot of additional funding. Horton Foundation with Davide, we have a joint grant that's very important, because I have to admit, we are touching on diseases where we're not the experts, like marginal zone lymphoma, but that's an opportunity for collaboration. So thank you very much.
Yeah, thank you, Thorsten, for this impressive talk and this overview of all those inspiring activities that you are pursuing. Are there questions from the audience?
I have some questions. The first is about checkpoint inhibitors, because we don't, clinically speaking, we have very unprecise, I would say, biomarkers. No, we rely generally in cancer, not in hematology or lymphoma, but we rely on tumor mutation burden, PD-1 expression, sometime on the type of microenvironment. But I'm always a little bit tumor-centric, and I think that the microenvironment just responds to what the tumor drives. So the neoantigen, to me, are the drivers of the immune reaction. And it's not just a number, but quality. For example, BCL1 is one neoantigen, but it creates a big immune response. Do we have a new way to analyze this quickly?
Well, we have in the single cell sequencing, there's also the T-cell receptor that can be sequenced by Andreas and his team. Yeah, so I mean, there's plenty of ways. I think I mean, still the initial, when we see these patients clinically, the initial question is PD-L1 expressed, right? If a lymphoma that normally doesn't have PD-L1 has PD-L1, that could be that's an outlier that could provide an opportunity. The other one is sometimes the pathologist is not right, and he's not here, I don't think. But that's like for lymphoma, that's very normal, right? It's a challenge. So when we get a very long report where there's a lot of trigger points that this could also resemble something else, and then like the simple interpretation from my side, well, maybe the drugs that we use in this other disease work. And then there's well, maybe if this works, we will find out very quickly, right? And what is lost? So the patient that I presented, yeah, the standard of care is CAR-T, but with the next patients, I've always wondered, maybe I should try the checkpoint inhibitor first. If it doesn't work, it takes some time to get the CAR-T. So I think there's this. So I'm not answering your question directly. So there's a lot of complicated stuff, but there's also a lot of simple stuff that we should do more, and in parallel, kind of take advantage of the material that we have from patients that don't respond, take advantage of the technology, but really work this up, right? A lot of times, it's like, oh, what's happening with the light? Yeah, but a lot of times it's like you are looking for the home run and you forget all the base hits.
Thanks.
If not, I have a question. So I think it's, yeah, no. So I thought the data on these additive drag effects were very interesting. And did your data, there's those two hypotheses where you can think about two drugs adding on top of each other because they inhibit the same pathway in a stronger way. But it could also be that one drug leads to some compensatory upregulation of a different pathway, and then you add drug B, and then you get a stronger effect. Did your data allow for any conclusions? And if there one or the other mechanism works better in the diseases you've been looking at?
Yeah, I mean, we see these effects, right? We see things going down, and then sometimes for some of the drugs or the effect that multiple drugs have, we see upregulation, and that's something that we need to do in the future to kill that pathway. We can look in the data which drug would do that. But the example I showed, right, this is a function of the genotype, right? If you just take what does drug A do and drug B, and then look at the combination, you would not see this. But where you see this synergy is really when you consider the genotype, when you ask. So I think there's an unlimited number of questions that can be asked, and the kind of next time around we will have more drugs, like more pathways to inhibit, and hopefully more patients. And then it's just a function of, okay, did we include the right drug? If we, but I think the power is really this kind of gene or something that is a tumor driver in this disease, the B-cell receptor, and then the mix of drugs. And we've not, I mean, we just put one combination for a particular reason, but I think there is a lot of potential to just agnostically test for this, but there's also, and I guess that's what you're alluding to now, now that we can really take apart what do the drugs, what does the PI3K inhibitor really do, right? There's the textbook knowledge of how the pathway should be arranged, but now there's this data from 100 different patients where you can actually put a weight to this, how much of this PI3K activity is linked to mTOR and AKT, how much of it is linked to the B-cell receptor, how much of it is linked to some other stuff that may be noise. And I think that's where the opportunity is. But I feel very strongly that our value chain is kind of do these experiments on a big scale, try to understand, like find signals, try to understand them to then take them back clinically.
Thank you. Any more questions?
Yeah, we have a question.
Very nice talk and lots of interesting data to look at. So with your single cell approach now, you can start to look at tumor intra-tumor heterogeneity and perhaps target that also. Are you thinking to use your drug combinations to target that based on your molecular profiles, or are you more thinking about using like the drug combination and look for synergy for drug combinations?
Yeah, so that is the ambition, and they also put it in the proposal that I guess we have 100, or we have a number of patients, but within a given patient there are multiple clones, and if we can kind of pinpoint the clone and follow it with treatment, right? So now you have 2 tubes, and it's not trivial to link this back. The copy number is very easy. So, but I think we just haven't had the time to get all the data into a format also across batches to more systematically look at this. But we can ask Andreas later in this talk. It's yeah. So but that was one of the reasons for doing this, to kind of multiplex an individual patient by looking at multiple clones.
Yeah.
Additional question? Yeah.
I have one more question about indolent lymphomas, let's say the prototype follicular lymphoma. And the issue is the incurability, and it might be linked to the fact that there are common precursors of the CPC around in the patient. And this comes to one of your comments, the question on how tracking the stem cells, do you have any way to, and also links to the concept of tumor heterogeneity, because in the samples that you will collect, likely there will be some CPC, Do you have any way to figure out whether all these, also these precursors are sensitive to the drug you are using to perturb?
Yeah, I don't know. We would first have to look whether we capture these cells. I mean, usually this is from the blood, sometimes from the lymph node, rarely from the marrow. So and on average it's about, like for the high dimensional flow, it's about 1 million cells. For the site-seq, it's only 1,000 cells. So it's very unlikely that there will be stem cells in most of the samples. But I think those things can be done, and we would have to include more marrow samples. But I think the challenge that I see is how do you tell the good stem cell from the one that's already on a bad trajectory, and it's not the point picking up point mutations from the site RNA-seq is very, you have to be very lucky, right? So that's a challenge. So we're focusing on the copy number, determine clonal heterogeneity, and the modules, like for each of these tumors. Andreas and Marcel now have the modules, and we keep discussing how many are there. But anyway, I think and we see this, right? So I think the value is we see much more than just copy number determined subclones. We see the proliferating compartment. I think this may be more tangible, right? To see Is there something that kills the small proliferative clone, and how does it relate to the other? But also this natural heterogeneity that can now be captured with gene expression.
Thanks.
Thank you. If there are no further questions, we move on to our third speaker of the session.
Claudia, Professor Haferlach, will from Munich, the leukemia laboratory. She will discuss on precise diagnosis and prerequisite for precision oncology. So thank you, Claudia.
So thank you very much. Good morning also from my side. I'm very happy to be here today. And I'm also very happy about this title because it gives me the opportunity to talk about nearly everything what we do in diagnostics. But as it is still early in the morning, I'd like to start with some basics and ask ourselves, what are our aims? We want to cure all patients. And what would be even better, to prevent disease. And maybe we have the time to discuss about it. To do that, we have to understand the biology. We do this by basic research. And now diagnostic comes in because diagnostics is a kind of bridging. So we use what has been learned in basic research and we develop diagnostic assays to transfer it, to classify our patients and to help to find the right treatment. And also questions from the clinics go back to diagnostics, go back to basic research. So where did we start in leukemia diagnostics? We looked at the blood and saw that there were too many white blood cells. Then based on clinics, we decided, or we could distinguish whether these were acute or chronic leukemias. And then the first kind of diagnostic test came in, cytomorphology, and we could differentiate between the lineages. Since then, a lot of more techniques have been developed. And today, all these that you see here on the left side are currently standard in diagnostics. So we use phenotypic characterization by cytomorphology and immunophenotyping, but of course genetic plays an important role and goes from cytogenetics to molecular genetics. But we already heard this morning that new technologies have come up, like gene expression profiling, whole genome sequencing, single cell sequencing, and looking at epigenetics. And all these tests are already available in diagnostics, not for the whole patient population, but we can use them. And the question is only, when do clinicians need these tests and when should we apply them? So and what I'd like to do right now is to go through our different diseases as an example and show you what we've done in diagnostics so far. So the topic in AML will be classification going from phenotype to genotype. And what is, from my perspective, also very important, to go from prognosis to therapy selection. So you saw when we look at cytomorphology, we had 8 classes of AML. Now, if we look at genetics, we can nearly for every patient find a primary driving force. But what's also very important is that we see that there are a lot of additional abnormalities that we find in addition to the primary one. And this tells us kind of that every patient has his personal leukemia. And if we go for precision or personalized medicine, we have to take this into account. This is a challenge, but I show you that we made a lot of progress. So what did we do after we found out the different genetic subclasses? Of course, we looked at what does this mean with respect to prognosis. And you can see here, just for cytogenetics, that there are huge differences. If you only look at cytogenetics in the outcome between those with the cytogenetic abnormality like inversion 3 compared to 15/17 translocation. This has been also shown for molecular subtypes and is now very, very out-differentiated, and you can see for every class how the outcome is. And you can also differentiate between subclasses with co-mutations. So there's a lot of data out where we can look at different subgroups and tell how the outcome is. But what I really like to stress, and we don't have to forget it, prognosis is always determined by biology. That's what I showed you, so the genetic class, but also by therapy. And I wanted to remind you, for those cases where we have a poor outcome, we just have not found the right therapy. So after all these genetic parameters have been identified, of course, there has been a lot of effort taken to find for the right genetic abnormality, the right treatment, and that was a real success in APL with the PML-RAR alpha rearrangement, where we now can cure these patients definitely without chemotherapy. So I'm very addicted to genetics, but of course there are also surface markers that can be used as a therapeutic approach. So, but how can we use it now? So, you know, all the large clinical trials where you put different patients to different therapies, but this does not make sense anymore. And if we want to take biology into account, but it's rather difficult to get away from this classical trials. So in fact, where we want to go is to have the perfect therapy for each patient, but now as kind of in between, we go by the genetic subgroups. And here's just an example in AML, which was started already in 2016. And this was an open trial, so they expand the treatment arms based on the genetics. Always if a new drug was identified, they added an additional arm for this subtype of patients. This is pushing a lot of on diagnostics to be quick enough that we really can start early in the beginning. And you can see here that those patients that were treated in this experimental arms, they do better than those that receive standard therapy. It's always difficult to compare this, but I think it shows that we are on the right way. So in the next example, acute lymphoblastic leukemia, I want to talk about new technologies, which impact they have on classification, and a very important aspect, the role of measurable residual disease, and to evaluate our therapy effect. So especially in B-lineage ALL, gene expression profiling has added a lot to our knowledge on biology. So using this, for example, this new subtypes of DAX4 rearranged cases have been identified on gene expression profiling, which are difficult to pick up by other technologies. And you can see here that this has also impact on outcome. This subtype is doing very well, not only in children, but also in adults. And also an important subtype was the identification of Philadelphia-like ALL, where we can also have certain treatment options. This is just the WHO and ICC classification showing you those subtypes that can only be identified by gene expression profiling. So as I said in the beginning, in ALL, gene expression profiling has moved into the routine diagnostics because it's mandatory for WHO and ICC classification. I showed you in AML that we have prognostic systems, and they are very valuable. Usually they were used to intensify treatment in those patients with poor prognosis. But why I show this also in AML, this kind of prognostic system, you see here variable, standard, high risk, and very high risk, is this new category. And this is something that we should work on, and this should be in future what we use, because it says eligible for tyrosine kinase inhibitor therapy. So our new systems should use which patient subgroup is eligible for which treatment. And also many years ago, there have been proposals which new inhibitors to use. They have not reached the clinic in many cases, and this has been expanded also more recently, where you always find the target that has been picked up by diagnostics and then the respective treatment. What I also want to highlight in this table on the right side is, if we luckily, we have a very, very good treatment, we also should think about reducing intensity of the treatment, because we have always to take the adverse events into account. It's nice to cure a patient, but if we have a high cure rate, we must think about it, whether we treat too intensive and whether we can reduce it. So the next topic is, how can we do it? How can we know even better than just by doing genetics at the beginning of the disease? How can we do better if the patient, this single patient, really benefits from the treatment that we are applying. And this is, of course, measurable residual disease. That has been shown for a long time in ALL, that patients that show a molecular remission do better than those that don't. And you can even do it more sophisticated. So those patients showing only no residual disease with the sensitivity that we apply at several time points do much better than if you have a higher load of measurable residual disease. This very interesting paper also shows, of course, what we have to keep in mind, that in patients with high-risk cytogenetics, there are some patients that do better and show undetectable disease than others. So in every genetic subtype, if it's favorable or unfavorable, it makes sense to look for residual disease. And you can also see here what's also very important, that you have to look at the thresholds that really mean a certain relapse rate within the different subtypes. As you can see here, for example, in the favorable subtype ETV6 ranks 1, an MRD of 0.1 means a much lower relapse risk than in the high risk or intermediate risk genetics. So the MRD level plays a role in conjunction with the genetic subtype. We have to take both into account. So now I'm coming to the prime example and the template for all the diseases that I would hope that we can use. It's CML, because it really nicely shows us the ways we have to go. So we started with very simple cytomorphology. Then the cytogenetic abnormality was identified. It was shown that it is a translocation between chromosomes 9 and 22. Then next, the molecular genetics was found out, the BCR-ABL rearrangement. Then on the protein level and the pathways that are driven by BCR-ABL. So by understanding biology, we can make much progress in diagnostics, but also, of course, in therapy. And you can see here on this ground, tyrosine kinase inhibitors were kind of invented. And what we had to find out, and that diagnostic comes again in play, that under the pressure of tyrosine kinase inhibitors, certain mutations can be selected, and therefore resistance can occur. And therefore, if a patient becomes resistant, we have to sequence the BCR-ABL and look what we can do. And this prompted the development of new tyrosine kinase inhibitors. And especially recently, a new type of inhibitor which binds to another part of the BCR-ABL, so that we have the opportunity, if a patient becomes resistant to a certain type of tyrosine kinase inhibitor, that we can pick another one. And also new combinations are thought about, whether if we target early with different inhibitors, that we can get better outcome. And this is really a success story. Also, of course, minimal residual disease monitoring has been implemented in CML for many years. And the good thing here is that we really have developed an international scale so that we can compare MRD measurements between different labs. And the time until patients develop a major molecular response is also used to compare the efficiency of tyrosine kinase inhibitors, as you can see here in both figures. And you can also show what can be done with optimal therapy. As so many patients reached major molecular remissions in CML, there were the first thoughts, can we stop therapy in these patients? And then it they started doing it under measurement of residual disease. And you can see here that we can, it depends on the way you look at it. In 50% of patients, there was no recurrence of residual disease, while there are 50% that showed BCR-ABL again in monitoring. But the good thing is, if you start therapy, nearly all of patients respond again to tyrosine kinase inhibitors. So it's going on a very individual basis. So every patient is monitored, and depending on the result, you change the therapy or you stop it. So, but then again, Beck, clinics is driving basic research. The question was, why do some patients come back with BCR-ABL positive cells and why others don't? And therefore, this group sorted the different cells in residual disease, and then they found out that if the granulocytes were still BCR-ABL positive, all of the patient relapsed. If only the T cells were positive, only about 2/3 relapse, and if both sections were negative, then only 25% relapse. So, and now the next step would be to introduce this in diagnostics to really look which patients can be safely stopped on tyrosine kinase treatment. And there are different parameters that have been found out that influence if it is successful to really stop treatment. This is the treatment duration, the type of treatment, which is nicely shown here, and the MRD kinetics. So patients that go into remission very quickly do better if you stop therapy than those where it takes longer. And therefore, and I also showed you the lineage. So therefore, based on all these parameters, in every single patient, you can decide, should I go on with treatment or can we safely stop treatment? The next aspect I want to touch with respect to personalized medicine, of course, is also to look at the adverse effects and the comorbidities. And in CML, it's already out that you can look for the adverse effect and, depending on the comorbidities, select which tyrosine kinase inhibitors would work better in one or the other patient. Another thing that we always do in clinical trials, of course, there is a certain dosage of the drug that you are giving, but is this really the perfect one for each individual patient? And there are some guidelines discussed where you can go lower the dose or where you have to have a higher than standard dose, and also monitoring of plasma levels, which would again be diagnostics, are discussed whether this makes sense to do it on an individual basis. So kind of summary of the blueprint here shown as an example in CML. Of course, we have to start with a diagnostic workup. We have to look for risk factors. I did not go into detail there, so additional cytogenetic abnormalities or molecular abnormalities are risk factors. We look for the comorbidities. Based on these data, we make the best treatment selection for each individual patient. Then it's really, really important to go for response monitoring. If we have treatment resistance, we have to go back again to diagnostics and adapt our treatment, go for response monitoring. If that's excellent, we can discuss about stopping therapy. If it's not, we have to restart therapy or again use another treatment. So now I want to show you which progress we have been made based on all of these different aspects that we are doing, better diagnostics and better treatment. And you can see here that we go up the top, and if we would have the newest curve, it would be even higher. And we can say in the majority of patients with CML, we have now reached normal life expectancy. We are not as good in the other leukemia settings, but there we are also making progress. It's a little bit depending also on age, but I think this really shows us that if we really understand biology, we can have better diagnostics, better classification, and better treatment. So now I showed you in the beginning that we have a lot of different diagnostic tools in our toolbox, and they are listed here again. And the question is, what is the optimal choice of diagnostics? Because it's currently, it's not possible to use every diagnostic test in every patient, and it also makes not sense from a financial aspect. And therefore, I always think we have to think backwards. So we have to think of, first of all, what are the treatment options that we have in a certain patient, and then we can decide which diagnostics we really need in this certain patient. Is it just sufficient at the beginning of CML just to look for the BCR-ABL, or is it a more complicated disease where we need, for example, whole genome sequencing because it has a lot of different abnormalities? And then, of course, we have to determine the prognosis, but better even find out what is the right treatment for each individual patient. So what we are currently doing is classification, prognosis, and therapy. But what we should do in the future more is that we separate it into different fields. So we have on one hand the disease factors, so looking at the biology of the disease. So from my perspective, of course, the genetic profile, but there will be added other aspects as well. And it's also important, and I will focus on that in a few seconds, the risk of progression, because not all diseases have the same risk of severe clinical symptoms. We have to look at patient factors, so performance status, comorbidities, and clinical symptoms. And of course, what is the greatest shortcoming from my perspective so far are the therapeutic factors, so the availability of drugs and the side effects of drugs. But because it has a great effect whether the drug has many side effects and severe side effects or not. Because in the future, in the individual patient, we should always think about when is the right time to start therapy. So we not only have acute leukemia. So this depends on the severity of clinical symptoms, the side effects of the treatment. So if we have no side effects, we can also treat with very limited symptoms because you do not do any harm with your therapy. But it's also important to look at the risk of progression, because I will show you in the next slide that we have a continuum of the diseases. And I showed you an example where we have a disease which we can nicely treat. But this is a disease where you just mainly have one driving genetic abnormality. Unfortunately, in many other leukemias and solid tumors, we have a lot of different genetic alterations. And therefore, maybe we should discuss starting therapy earlier. And this will be the challenges in the future, how we decide this in each single patient. And this kind of figure tries to illustrate it, that we have pre-leukemic or pre-cancer times. And if we have there an actionable target, just in best case, a single target, and we have a drug with hardly any side effects, we can treat really early. If we don't have these drugs, only drugs with many side effects, we might wait longer until clinical symptoms occur, because we have to keep in mind, not all patients with pre-leukemic disorders really will develop leukemia. And this is also very important. And I said, I just show it here for AML, that it's a continuum. We start with very it's not really a disorder with CHIP. Then we go to clonal cytopenia, MDS, and AML. And here the question is also, where should we start treatment? And therefore, for each situation, groups are working on that to find out what is really the progression risk. And that is the major task, so that we only think about treating patients that really have a high risk to progress to a more severe disease with clinical symptoms. So this is kind of a summary. So I talked about a lot of genetic alterations that are found, comprehensive phenotypic and genomic assessment, risk stratification, MRD monitoring, and comorbidities. What I did not talk about is about the germline constitution, but I think this will play also an important role in the future. And in combination with this, also the drug metabolism that is dependent on germline constitution. And this will also be factors that we have to take into account if we really want to do very, very good personalized medicine. So I do not want to skip all the other things. So I mainly touched on genomic analysis, but of course there are other things like metabolic profiles, surface and signaling pathways. So it's really a challenge to bring all these together. But I think, as I showed you, we have done a lot of progress. So I think especially with artificial intelligence, we maybe can handle all this data and make the best for our patients. And I stop with a little change in my title slide. So it's not as a prerequisite, but it is a prerequisite for precision oncology diagnostics. So thank you very much for your attention.
very comprehensive overview and great presentation. I'm sure there are questions.
Maybe I can start with first a remark. I appreciate that you mentioned the patient profile, because for some diseases it's becoming more important than the disease profile. For example, for CLL now we decide on drug more on the comorbidities, concomitant medication of the patient rather than the genetics of the tumor. What we didn't do in the past when we had only chemoimmunotherapy. So that's something that we have always to keep in mind, clinically speaking. Then I have a couple of questions in terms of translation of what you are saying in the clinical practice. Is a transcriptome widely accessible for identifying Philadelphia like ALL? Because it's not something that many labs can do. So do you have this in your lab, and how widely it is accessible, I don't know, in Germany?
So I think currently not many really use it, but we have it available, RNA sequencing, whole transcriptome sequencing, and also whole genome sequencing. We do not use it on a large scale, but in certain instances where we think that it really helps the patient. So in ALL, we would offer it for everybody who knows it, but not doing as kind of a screening tool, but do the routine diagnostics. And if you do not find, so if you have a BCR-ABL fusion, then you don't have to do the RNA sequencing. We have it as an option for those who do not fall in one of the categories, because we need it for classification, and also it has therapeutic impact. So from a diagnostic point of view, it's always quite disappointing that we are very much ahead in diagnostic, that we can classify the patients, but it always takes a certain time, and from my perspective, much too long until it comes to the clinics. So like MRD monitoring, it's so obvious to do it that it makes sense. And we've done it for 20 years in the labs, and in CML it has been done for a long time in APL, but it took so long until it is now being adapted. So in ALL that is also for quite a while, but for AML it's just starting. And I think we should more discuss what you do with these results, because everybody's sticking to the trials, and the British, they have done MRD trials and they have shown that it makes sense to use it to adapt your therapy, but it takes a long time to convince it. And the classical trials, they are not made for personalized medicine. So if you break out from the current protocol, it's always a problem. And therefore, I think the clinical trials have to change so that we can really get into personalized medicine.
Yeah, that's important, because this is a typical task of academic trials that are largely unfunded, compared to a pharma-sponsored trial, which have not yet.
Yeah, but I think, so I don't want to go away from that, but I think this is a real problem. In the old days, trials were driven by academia, and now it's driven by pharma companies, and I think that's not really optimal.
Question.
Thorsten?
I have another question on MRD. Sorry. Go ahead. On MRD, because now we have 10 to the minus 3, for example, in ALL, more or less 10 to the minus 3, 4 is, or a 3 log reduction is acceptable. Then 10 to the minus 4, CML, more or less, MRD 4.5, and 10 to the minus 6 in ALL. And then we have a further challenge with if we move to ctDNA, no? So what's the best?
So I think with respect to the thresholds in the different, I think we have to look at the different diseases. And it's a little bit like trial and error. We have to find out, as I showed, this is why I showed the ALL slide. It's dependent in ALL on the genotype with how much MRD the body can cope. And therefore, I think we have to really find it out for every certain disease.
Thanks.
And maybe also in every single patient, if it's stable or if it's increasing.
So you're in a unique position. You get a lot of samples from all over Germany, but also Switzerland, and then obviously they send you samples to get a short diagnostic report, like 5 markers or 10. But the technology you use to get to those 10 markers can be whole genome or can be RNA-seq. So my question is, are you also taking advantage of all the data that's not needed to fulfill the the initial question? So how are you dealing with extra important data that you can use to contribute to many additional things, byproducts of the whole genome or RNA sequencing data that will not go into the report directly, but you have that data?
So for the large cohort that we did for research, the whole genome sequencing cohort of 5,000 cases, we did not report them because it was just on a research basis. If we now, for single patients, are asked to do whole genome sequencing, we of course report everything, to the best of our knowledge today. So we cannot interpret everything, but to the best of our knowledge today. And in the early days, when we already did panel sequencing, but just a few genes were asked for, we found a way also to distribute the information without. So we could also write in the report, it's more about reimbursement. So we gave the information in another way, so for the benefit of the patient.
Alex?
So, talking about MRD, in CML, MRD is very successful and guides our treatment. One of the reasons why MRD is maybe not as successful in AML is because it's not standardized. So do you have approaches to standardize this in Germany or in larger consortium in the future, like for FLT3, NPM1, other genetic markers for MRD?
Yeah, I think this really should be our goal, but it's very, very labor intensive, and you have to find one who will take the lead and just do it. And there we were very happy in CML that, for example, Nick Cross was doing that, and I think it needs someone who is willing to do it. But I think this is necessary, and I think there's another problem in AML. In some AML subtypes, it's not really sure which markers you should use for MRD monitoring. And therefore, I did not touch on that. In the more complex diseases, we have also to look more for clonal hierarchy. So which is really the first event, and where do we have to push back the leukemia? Let's say we have DNMT3A and NPM1 as the second hit. It will be sufficient to cure AML to get rid of the NPM1 positive cells, and DNMT3A might be allowed to stay. Would be a higher risk for secondary AML, but I think we have to define it really in every specific subtype and in the end, in every specific patient, because the hierarchy can be different. We have cases where DNMT3A comes first and NPM1 second, and we have cases where it's the other way around. And we haven't taken that into account in MRD monitoring, but also in the clinical trials.
Maybe a very short last question, just because you mentioned this on your slide. Can you comment on the role of methylation profiling in the space of heme malignancies from your perspective?
Yeah, so I think this is really just on a research basis, but it would not be problematic if there is a need to know it, then also to introduce it into diagnostics.
Well, thank you very much for this interesting session and great discussions. And I think we managed to stay very good in time and have deserved a coffee break now until 11:05, I guess, right? Thank you. Thanks again to all speakers.
Thanks. Okay, we need to get back to work. And we will start with a welcome from the medical faculty president. I'm not sure how is your what is the name of the position? It's the the
well, in English, it's vice president.
Vice President of Medicine. So Beatrice is an anesthetist, or was an anesthetist before she took over this job, but she's also the key person pushing for precision oncology, helping the cancer center, helping the research in that area. So I'm very grateful for your words of welcome, but we are also, the community here is also very grateful to your push for this particular area of interest. So thanks for coming, Beatrice.
Yeah, thank you, Thorsten, for your kind words. Dear colleagues, dear guests, I'm sorry for my strange voice. I just had a hacker attack from some bugs, which tried to overcome my immune system. But you know, I love precision medicine, so my therapeutical approach is ginger shots, and I think it works. Let's go back to the topic. On behalf of the University Medicine Zurich, it's a great pleasure to welcome and meet you here at the 6th Zurich Precision Oncology Symposium of the Comprehensive Cancer Center Zurich. Thank you very much for the invitation and excellent organization, dear Thorsten, and members of the program committee. Today's event is one of the key symposia of the Comprehensive Cancer Center Zurich's education and outreach program. I'm excited to see the exceptional lineup of speakers we have today, leaders in the field from Zurich, from Switzerland, and from abroad. We are privileged to have such a distinguished group of experts, and I appreciate also the diversity of speakers. I strongly believe that this interdisciplinary diversity is crucial for scientific and clinical innovation. Interdisciplinarity and excellence are, by the way, also the cornerstones of University Medicine Zurich, of UMCH. UMCH integrates the 2 Zurich universities and 4 university hospitals. I think you are well aware of that, providing scientists and physician scientists with a unique environment to advance medical sciences. The Comprehensive Cancer Center is one of our 4 UMCH centers next to the ORX, the Tumor Profiler Center, and of course, the Loop Zurich. In recent years, UMCH has made significant investments. We heard that from Thorsten in precision medicine, particularly aiming to develop a strong leading hub for precision oncology in Zurich. We are proud to host numerous pioneering initiatives here. Just to mention 3 of them. First, an impressive example is the Tumor Profiler Center, which employs cutting-edge multi-omics tumor profiling techniques to advance the understanding and treatment of cancer. Second, another important asset is the Loop Zurich, which, amongst others, fosters translational cancer research projects in lymphoma and blood cancer, artificial intelligence tools for better decision-making in oncology, and a biomedical informatics platform which promotes an interdisciplinary, data-driven research culture in Zurich and hopefully beyond. Third, at the Comprehensive Cancer Center, 4 lighthouse projects develop disease-focused programs for blood cancer, melanoma, glioblastoma, and oligometastatic disease towards targeted therapies, personalized immunotherapy, and precision radiation therapy. These are just a few highlights of forward-thinking projects that have been developed here in Zurich. It's evident that we are creating a vibrant hub for innovation, collaboration, and most importantly, of course, patient-centered care towards precision oncology. I would like to take this opportunity, and of course also on behalf of the executive board of the university to say a big thank you to all the colleagues here in Zurich who are committed to research, the promotion of young talents, and patient care at the highest level. I would also like to thank the board of directors of the Comprehensive Cancer Center Zurich for their outstanding leadership, and in particular, Professor Markus Manz, where are you? There. For his outstanding performance with his vision for precision oncology as chair of the board of directors. I think a big applause to all of you. Coming back to today's symposium, the fact that the Comprehensive Cancer Center Zurich educational program is organizing the Zurich Precision Oncology Symposium for the 6th time today underscores the center's commitment to advancing knowledge and fostering collaboration in the field of precision oncology. Thank you once again for being here, and enjoy the rest of today's symposium. Thank you.
Okay, so I think everybody had a coffee, which was desperately needed, I have to say. I feel now much better than in the first session. My name is Markus Rudin. I'm chairing the second session. I'm the director of the Loop, which has been mentioned before. And we have been also involved in some of the projects which have been reported before. The co-chair is
Björn Menze. I'm here also from the Unispital, and we do image analysis that relates to quite a few of these topics here. Yeah, we have a session with several esteemed speakers. And the first session is very much to, what we research-wise work on. So it's a pleasure to have Virginie Uhlmann here as the first speaker. She was group leader at the EMBL in Heidelberg, working in the field of bioimage analysis, and joined UCH last year, I think, to head the BioVision Center, and would talk today about quantitative bioimage analysis and how to scale it to large quantities, which is at the heart of a lot of computational techniques. Thanks, Virginie.
Okay, so that's working. Let me just set up the computer. All right, perfect. Great. So thank you very much for the kind introduction. Also, thanks to all of you for being here. That's, of course, not what we want. This is one of my grants and an update of a software. So hopefully we stay with that instead. Oops. So yeah, it's really a great pleasure to be here today. I'm learning a lot already from the first session of talks. And I hope that with this, I can convey a little bit about the excitement of what we are doing at the BioVision Center towards developing methods for quantitative bioimage analysis on large scale data. So everything I'm going to show you is really the cutting edge of research, so to say, right? So it has some application to precision oncology, as I will show you at the end, but everything is sort of moving targets and in development, right? Which is why it's exciting. As Björn mentioned, I also actually still have a joint appointment with EMBL EBI, which is part of EMBL, and it's based in Cambridge. And there I have a lab that develops novel quantification methods for microscopy image data based on AI methods and other sort of approaches that are more classical. So a lot of the things that I do there are not in this talk, but sort of feed into it, obviously. Yeah, so perhaps as a start, I guess if you're here, you're probably interested in image data at some point. But as many of you may have experienced firsthand, one of the things that we are struggling with biomedical imaging today is data size, right? We have technologies that allow us to acquire large amounts of data, which is really exciting because it means we get to observe a lot of things in a lot of details, and we get to gather a lot of information on the experiments that we do. But obviously, everything that is in the megabyte range perhaps can be handled by a human. When we move to the terabyte range, we get to a realm where manual annotation and manual exploration is just not feasible, right? We all have to sleep at some point. We all have to do other things than looking at data. Of the different things that you see on the slide here, perhaps the one on the right is the one that is the most relevant to you today. This is the sort of things we get with high content screening and high content imaging or high throughput screens. As some of you may have experienced, if you set up your experiment with your plates, you have all of your different cells and different conditions in your different wells, you put it in the microscope, you hit run after having set up everything properly, and then you come back a little bit of time later to look at your data, and what happened? Oh, now you have 160,000 images, something like a couple of terabytes of data. Each of them contains lots of cells, tons of information, tons of really valuable insights, but obviously this is not something that we can handle by hand, right? This is not something that we can go through as a human being. And that means that in order to harvest the information that is in there, we do need to think about how to handle this data. And we do need to think about the analysis per se. So basically computer vision, how do we use a computer to pull out visual insights that this data would contain? But actually even before that, sorry, we need to think about much more mundane but equally important things, like how do I even store all of this in a machine in a way that would allow me to visualize it at once? Because this is far beyond what the memory of any regular computer and even a powerful computer could handle. And this brings us to a discussion on file format. So that is usually not the thing people find the most exciting, but that is obviously really, really important, right? I've heard in different contexts, people use this saying that I like a lot, if you want shiny fountains, you need very good plumbing. This is the plumbing. And the bioimaging analysis, the bioimaging, data community has really been thinking for a long time about how we handle these massive amounts of data, because we do know that the machines we are depending on to generate data generate gigantic amounts of images. And the answer of the community to this problem is something called OMISAR that I don't know if some of you have heard about. I wouldn't be surprised if not, because this is a new thing, but hopefully you will hear about it in the future. This is basically a proposal for a standardized open file format that can handle all the different kind of imaging modalities that biomedical research generates, and that scales very well from megabytes to multiple terabytes in size. And again, it may not sound that exciting per se, but the reason why we, people like me and many others, are excited about it is because if we have a good way to decide how we store these digital objects on our machines, then, in a scalable way, obviously, then we also can start thinking about how to build ways to store, visualize, and analyze this large-scale data in a way that is modality agnostic, right? In a way that can handle equally well high-content imaging, electron microscopy, digital pathology images, and so on and so forth. And that means we can then develop methods for analyzing this data that can be reapplied in lots of different contexts. Now, just to make this a little bit more concrete and hopefully convey the excitement, here I'm going to show you a video. This is the same data set you saw before. So this 160,000 images, couple of terabytes. It's a screen looking at cardiomyocyte differentiation. You could look at each individual field of view individually, as I showed you before. But if you use this sort of scalable file format, you can actually look at them all at once. And I hope that the video starts. If I click there, So here we see actually the full plate. And as you can see, we can zoom in individual wells. We can also look at analysis results, and we'll come back to that later on, where each individual objects are segmented. You can see that as we zoom in, the resolution kind of gets loaded as we go. And this is really the same sort of thing you get if you do Google Maps, right? It's basically allowing you to explore a data set that is much larger than your computer memory in a very smart way, because under the hood, we have a good infrastructure to deal with it. And this is really just interactive exploration visually, but of course, then we can go and do much more quantitative analysis, and that's what I want to tell you about in the second half of the talk. The reason why we are excited about this standardized file format is because on the analysis side, standardizing the way we store the data also means we can start developing interoperable analysis modules. And again, this is maybe something that may not be immediately obviously exciting, but that for people like me who are method developer is really, really key. Because if I come up with a new machine learning technique to analyze cells in one modality, I don't want to have to re-implement it for another modality, or to have to do a lot of software engineering to scale it to large data set sizes, or to move it from 2D to 3D, right? Having a standardized file format allows us to do that. It allows us to implement something once and then essentially scale it up to large amounts of data. So who's behind this initiative? It's a really big international community effort. The original paper was from 2023. I invite you to have a look if you want more details about what is the idea behind this file format and what are sort of the technical details about it. Now, the reason why you probably have not heard about it yet is because at this stage, we are still essentially deciding how to implement this idea, right? And how to turn this into tools that people like you can use. And we actually had a week-long hackathon here at the University of Zurich last year, where we pushed forward really a lot of the aspects that need to be implemented for this to become something that everyone can use. And we are continuing to do that. So if you're interested in that space, again, yeah, please reach out. We have lots of different community events and things that we would be really, really happy to include you in. Now, these standardization and community efforts are also what motivated the creation of the BioVision Center that I am leading at the University of Zurich, because one of the key missions that we have is specifically to develop fair standards for bioimaging data, so the sort of thing I just talked about. The other thing we want to do that really builds upon this is to make state-of-the-art bioimage analysis at scale accessible to all. So to build tools that allow building an interface and creating a bridge between those like you who want to analyze the data they generate and people like me and many others who develop advanced methods for image analysis. So the flagship project that we are developing and that I want to tell you about now is called Fractal. This is a no-code platform for bioimage analysis with OMISAR. So in this standardized file format I mentioned earlier, the development of this project is led by Joël Luthier and Lorenzo Cerrone, who are both at the BioVision Center with me. And the idea behind this is that it is a computational platform that allows you to process terabytes of images as in this OMISAR file format. Of course, if it's less than terabytes, it can also do it, but I mean, that's where it really becomes interesting. This is fully extensible and customizable platform, as you shall see in a minute. So it is something that we really envision people to build upon. We don't want to be the ones owning it and developing everything for it. And it basically allows you to create large-scale bioimage analysis workflow, manage their execution and run them without ever facing code, because everything happens through a web-based interface, as I will show you later, and without essentially needing to be an expert at scientific computing. So we really want this to essentially provide a platform where the method developers and the potential users can meet in a way that scales really well on large amounts of data. Now, if I go a little bit more into the details of how that works, so what Fractal is really space in which you have building blocks, and this is why our logo is with Lego blocks, as you shall see, right? That are nothing else than processing modules. We call them tasks, and we represent them as little bricks like that, that do individual processing operations on this OMISAR file format. So it can be anything from, okay, I want to convert a proprietary microscopy file format into this OMISAR standard. I want to register my images. I want to segment objects. I want to extract measurements. I want to do other things. As you shall see, you could just develop your own. But the sort of common point in all of this operation is that, except for conversion, we will start from an OMISAR file and end in an OMISAR file. And again, for us, this is really what is key behind having this standard, is that it allows us to write processing module that will work equally well on a couple of megabytes, on terabytes of data, on 2D, on 3D, on whole images, on field of views, and so on and so forth. So again, coming back to the plumbing analogy, there is a lot of plumbing behind this, but then it allows doing really, really beautiful things. Now, as I briefly mentioned earlier, the way we develop this is really not by envisioning that we own everything and are the only one building the processing modules, because if we were doing that, then obviously, you know, we would be a little bit limited in terms of throughput, right? We only have so many hours in the day. So from the onset of this project, the idea behind is to make this a community effort and to build infrastructure that can be expanded upon, that can be further developed by people who develop new methods, by people who want to implement specific processing modules for their need. If you know how to code and you're interested in building for this platform, we provide template repositories, videos, documentation. We have regular meetings to help onboard people on how to develop for that. So you can basically include your own methods into this if you are interested. And we actually, as a matter of fact, have quite a lot of people who are doing that already. So of course, some of the processing, the sort of core processing elements that are present in the platform are developed by us, but there are many other packages of these processing modules that are now growing and being developed by other people who use the platform for their own needs. And I will come back to this one later on that is being developed at the INTeRCePT. Another thing that just really illustrates this ambition that we have to create a bridge between method developers and users is that we don't only want people to implement new processing modules for this platform, but we want that when they do so, they are visible, right? And their methods can be reused by others to really amplify the impact of the work that they do. And so we also essentially list all of the different methods that have been developed by others for this platform. This is, I mean, you can check the link if you want. You can just access it in your browser now. It's online. But basically, we are really assembling this collection, this growing collection of capabilities for the platform, and it keeps on expanding. And that's really how we want it to be. Now, just maybe perhaps one technical slide, because it has implications later, as you shall see. I'm always talking about a computational platform. You know, that may sound a little bit abstract. What is it very concretely? Concretely, under the hood, what this is a server that runs somewhere where you have compute, so where you have computational resources, either in the form of CPU or GPU, and that has access to data you want to analyze, right? And then there are tons of different elements that I'm not going to go into. But the key part is that this fractal platform runs in a federated fashion. So the idea is absolutely not that it runs in one place and the entire world connects to it to do their analysis. The idea is that it runs wherever data and compute is, and people access it when they have access to this data and this compute. And so what may be perhaps most relevant to you in the room is that if you have credentials with the University of Zurich, you can actually access the fractal server that we are running on the science cluster at the University of Zurich. You can either go to this link or flash this QR code. We can basically give you access to it, and you can just go there and do your analysis once you have appropriately located your data. Now, what does it look like if you were to actually go there? As I mentioned, this is something that is meant to make it easy to do large-scale analysis. So basically that you don't have to be an expert at coding. And that's basically, yeah, so this is a video. That's what it looks like. So if you log into the server through a web browser, there is a place where you see your data, and then you can go ahead and build workflows. And as I said, workflows are nothing else than collection of building blocks that can be converting data. I mean, usually that's the entry point, converting data from a proprietary file format into OMISAR, and then doing all sorts of different things that can involve deep learning-based segmentation, that can involve measuring things, and so on and so forth. When you hit run to start a workflow, what's happening under the hood, and you can kind of get a glimpse of here, is that your processing and your data are essentially dispatched on the compute cluster. For UZH, that would be the science cluster. And Fractal orchestrates how much resources are allocated to what, and so on and so forth, to make sure that all of this runs even if you have massive amounts of data, yeah? And then, okay, you have error logs and progress bars and everything that you would expect from a normal interface. But perhaps one thing I can convey also that I'm very enthusiastic about with this is that because we rely on this OMISAR file format, that as I said is very appropriate when we deal with large data structures, we can actually also visualize results and explore results in the web browser without doing any sort of data transfer back and forth. And that's what you see now on this video. So you see I'm still on my browser. I have the server here open, and now I have a viewer open on the other one that basically now allows me to check the results of my segmentation on this couple of terabytes data set without downloading anything on my machine. And as you can see again, when we zoom in, it takes a little bit of time to update the highest resolution because this is streamed over the internet connection, right? So that's the limiting factor. But yeah, I hope you get a sense of the fact that, again, personally, knowing that we moved from having 160,000 images to being able to do that on the browser, I think, yeah, there is something there, right? So this is actually happening through a viewer called Visa that is developed by Trevor Mans at Harvard Medical School and that has been working with us to set this up. It's still very much work in progress, right? As in at the moment, it just allows you to look at things. It's not particularly interactive, but we hope to develop this further. And if you want to be interactive, you can actually do so in other types of viewers. Here, this is an example with something called Napari that is developed by the Guckenberger initiative. Sorry, where we do something quite similar in the sense that we are streaming this data set from wherever it is, and then looking at it together with results of the analysis. So this data set has gone through a fractal pipeline, meaning that each and every object has been segmented, things have been measured on this data, yeah? And because all of this is now stored in the final OMISAR, we can use this one file that contains all the information to do interactive data exploration. And this is the work of a lot of members of our community, including Fabio Steffen, who's in the room just here, and a couple of other people, Max Hess from UZH and Adrian Chan from Acute Therapeutic. In the video now, you will see again this thing of basically interactively browsing a couple of terabyte data set that is much larger than memory. But now that we have the analysis results, we can do things like saying, actually, just bring me to well, whatever. And in this well, give me the segmentation results just for that field of view that I'm interested in. So again, it will take a little bit of time to load because this is streamed through your network. So if your internet is not so good, then it takes time, but this is the limiting factor. Once it's loaded, you basically see the segmentation results for that one field of view that you asked to load. And you can then do interactive visualization in saying, okay, since each of these objects have measurements attached to it, I could ask to color them, for instance, based on a morphological property that they have. And that's what you are going to see here. Now we take the area and we color everyone by their area. Or we can take another one of the many measurements that were extracted, solidity, we color everyone by solidity, and so on and so forth. So you really get this very intuitive and visual way to explore the different types of morphological features that your data have. And you can go even beyond, because again, this huge collection of measurements is all stored at the same place together with the raw data and the segmentation. You can use them to do machine learning classification. So here now what you will see is saying, okay, let's pick a couple of examples. Here that's going to be mitotic cells versus non-dividing, so interface cells, and use the underlying measurements to infer what's the identity or the state of the rest of the cells we did not annotate. So now you see, we go ahead and pick a couple of cells. This is mitotic, this is mitotic, this as well, and so on and so forth. Then, okay, we'll move on to the interface cells that will be tagged in blue. So a couple of examples of interface cells. And now under the hood, based on these examples and their attached measurements, machine learning, not deep learning, shallow learning, random forest classifier is trained to predict on everything else, yeah? So again, in a very intuitive and interactive manner, you've built something here that allows you to infer, okay, on that field of view, but that you could very straightforwardly propagate to the rest of the data set. That is, again, a couple of terabytes big. So everything that I showed up to now is obviously general for microscopy data, right? You can apply this to precision oncology and to many other things. Now, I just briefly, in the last couple of minutes, want to show you how this is actually being used in the context of precision oncology by our collaborators from KISHP, Jean-Pierre Bourquin, Natasha, who gave the talk just before, and Fabio Steffen, in the context of their image-based DRP platform to inform personalized therapy options for leukemia patients. And so basically, I mean, Natasha did a fantastic job at explaining the medical part of this, and I'm extremely not appropriate to talk about that. So what I want to tell you about is more the actual analysis that is happening under the hood, right? And what is being done here is that Fabio developed a fractal workflow by building his own modules to essentially go through the whole process of converting the images that they acquire here from the Operetta proprietary file format into OMISAR, to segmenting each and every one of the cells in the data with a deep learning method called StarDist that some of you may know, and that is state of the art for the segmentation of roundish objects, to then extracting quantitative measurements on each of these cells, using this to classify cell types and states in the same sort of fashion that I showed you before, and finally save everything into the OMISAR along with metadata about the experimental condition of the different wells that are in the data set. And so all of this was developed by Fabio and is now a task package that, again, is listed on our page that can be used in the Fractal platform, that can be reused entirely or in parts, right? If you had data from Operetta that you wanted to use and do another piece of analysis on it, you could use just this module along with others from other packages. This is really what I mean when it's meant to be modular. And what that enables is really to go through the whole workflow that I showed you in my previous slide, but here in the context of this DRP platform. So starting with huge amounts of images, multi-well plates in which we have cells in different conditions with different drugs, concentration, medium samples, and so on and so forth, to processing these large amounts of data with the pipeline I just showed you, saving everything into an OMISAR that has all the relevant information and analysis results, and then using that to do interactive data exploration in the fashion that I showed you with the video just previously. So this is really something that, yeah, can and is being used in the context of precision medicine and oncology. And I think maybe one of the things I should highlight also is that for us, really the key aspect of this collaboration is that, as I said, you know, everything we do here is at the cutting edge and is in development. And we learn a lot also on how to further adapt the platform by the feedback and the sort of, yeah, the fact that Fabio, Jean-Pierre, and Natasha are pushing what we have to the limits, yeah? So we would really want to have more of these use cases that bring what we have currently to the edge of what it can do, to know how to develop it further and make it even more relevant. And that's, yeah, a good transition to my almost last slide. Perhaps the most important message behind everything I showed you here is that everything that I talked about is under active development, right? We are developing this actively. Everything is changing as we speak, so to say, for the best. And we made this conscious choice to develop in the open, so to basically not wait until we have a, so to say, finished product to put it out there. Because we want to hear from people who may want to develop or use this platform. We want to be able to incorporate feedback, and we want to be able to make it relevant. So if you're interested, we have absolutely all of our code, documentation, and so on and so forth on GitHub. You can also just reach out to us at the Biovision Center, and we can tell you more about that. And that's, I guess, yeah, what I want to end up on. Fractal is one example of the things that we do at the Biovision Center, but really what we are is a group of people who are experts in machine learning and image analysis, who want to develop useful tools that meet professional software development standards. And we are really keen to collaborate and hear from people like you about their needs, about their challenges, about how we can use our expertise to advance research in biology and in medicine. So if you're interested in Fractal, or if you're interested in any sort of image analysis related thing, please don't be shy to reach out and to connect. And with this, I'll thank the team at the BioVision Center, our funders, the people who help us with the backend development, and of course you for your attention. And I'll be happy to take questions if we have time. Thank you.
Yeah, thanks a lot. Of course, we have a bit of time left.
You decide.
Do we have questions? Yeah, let's start here at the front and then move
Yes,
on.
hi.
It's fascinating and amazing. How do you see the relation between this sort of regular bioimage analysis with the spatial omics that's coming up now?
Yeah, this is a fantastic question. Thank you, Wolfgang. So, I mean, we have on our roadmap, and we want to be able to develop this platform to also be able to include spatial omics at some point. I think the sort of big open question that we have to solve right now collectively between these two communities is that the omics community is sort of historically evolving in R, right? We evolve in Python. We, I think, need to find a little bit of bridges there. But really, this is to me that there is something extremely exciting in the fact that I think we are really at a convergence point where the omics world and the imaging world is coming together, and we can do good things together, yeah, with this sort of discussions on file formats and large-scale analysis.
There was another question up there.
Yes.
So you mentioned and of course there's a bit of a conflict because thank you. There's a bit of a conflict because each microscope maker, and also for all of the new spatial transcriptomics platforms,
absolutely,
they have their own software to sell.
Yeah.
And how open do you find they are in terms of sharing their file formats? Do you see that being a problem in future with maybe stability if they decide they want to change something that breaks?
Yeah, no,
breaks the code?
That's a really fantastic question. Thanks a lot. So, I mean, I think maybe I should have been clear with this, but like the intention of this community work towards OMISAR is at no point to say, okay, this is the one standard to replace everything else, and we are going to convince Zeiss to give up on their file format. That's not going to happen, right? I think we see this more as an interoperable unit that in an ideal world, we would want all the manufacturers to agree to map to, right? So to basically be able to convert to. And this is something that has actually happened in the past. I don't know if some of you are aware of this OMI-TIFF format. Yeah, that is kind of the ancestor of OMISAR, and that really has a history of broad adoption by the instrument manufacturers to not essentially be the immediate format they output into, but to be a format they all allow conversion to, because then this allows interoperability between machines. And so, yeah, this is really just the natural evolution of this. So again, because we are in early days, it's not that we have a buy-in from absolutely everyone already, but we do see actually very, very positive signal in these meetings that I was referring to earlier, where we are discussing how to develop this file format. We actually have microscope vendors represented, and they are interested in working towards this. So I think there is, I think there is really a path forward into this becoming a real open standard that is the convergence points from the different proprietary file format. And I think the reason why this does not necessarily antagonize, the fact that, they sell their own software, is that the sort of platform that I was presenting here, like Fractal and other methods that are developed on the research side, are really meant to be at the bleeding edge of, what happens in the realm of method development on the research side, right? And classically, a lot of the software that are sold by manufacturer incorporate these methods, but with a couple of years, lag, so to say. So I think it is really a mutually beneficial thing, in the sense that having a common file format allows everyone to map into a common space in which new methods are accessible to those who want to, but for those who develop software that they sell, they also get a little bit of a playground to assess, of these different methods, which one are promising, which one are meant to last, and which one should we then incorporate in our commercial software. So we have seen this work in the past, so I'm confident that this will work out. It's just that, again, this is early days of the sort of new iteration of this common file format, and so it will take a bit of time to get global buy-in. Yeah. Sorry, that was a long answer.
Very good. It's all yours. Do we have another question? Valentina, we'll have a lot of you. Let's hand over to the
hello
auditorium.
Hey, fantastic talk.
Sorry. Yes. Sorry.
Yes, I really appreciate the efforts of bringing this more advanced analysis tools to people that are not expert in programming. But I guess there is also a community of experts, if you want to, that could benefit a lot from what you are bringing here, this unified platform. How do you see their contribution in this development?
Yeah, absolutely. So yeah, this is a really good question as well. I mean, I can tell you, being a developer, method developer myself, and kind of having a group of people who do method development, this is also something that's like, I would dream of a world in which, when we develop a method and we publish a paper on the method, we can basically just dump the piece of code we used into an infrastructure that would then make it scale and work on different types of data, right? Because doing this work of making sure that my prototype that I wrote for my paper would work equally well on like 10 terabytes over the 10 megabytes they tested it on is software engineering. This is not research anymore, right? So as a researcher, as a method developer, I would also benefit from that. And that's really what we envision here. We really want to have a place that essentially, coming back to my analogy with plumbing, that provides the good plumbing that, allow those who bring really, really shiny things from method development and those who want to use them to meet without either of them having to do too much extra plumbing for this to work. Yeah.
All right. Okay, thanks. Thanks for the questions. Thanks for the talk.
Yes, thank you. So that was really a fascinating talk about modern phenogenomics of phenotypical analysis, I would say. Now we move more to the molecular side, and since I'm a chemist by training, I speak of molecules rather than omics. So because DNA is a molecule, RNA is a molecule, proteins are molecules. So molecular analysis. And we have seen in the first session, actually, that a lot of information is collected about molecules which are involved in the different processes. And we need, say, kind of a Rosetta Stone, which translates this into actionable information, which is certainly one of the big challenges which we have. Another aspect which we have to face whenever we collect data is noise. And noise, of course, are interfering sometimes in analysis. Sometimes noise contains a lot of information as well. But in this case, it seems that we would like to get rid of noise effects as much as possible. So I have the pleasure to introduce Valentina Boeva. She's a computational biologist at ETH Zurich. She works in the omics field, particularly actually genes involved in cancer developments, whatever. And one of the topics she's interested in is actually the title of this talk, Noise Resistance of Multi-Omics Survival Models in Cancer. So the floor is yours.
Thank you so much for this kind introduction. Thank you for inviting me to talk about very maybe niche subject in our research group, which is survival analysis, but I'm very proud of these results because I think they are very useful for the clinical community. So I guess most of you in this audience, you work with this high dimensional molecular data from cancers. And one thing you can do with this data, you can build a survival model, which will tell which drugs may cure or maybe help patients to survive longer under certain treatment, given that we train a model on the data and we have the information about the survival or event-free survival of cancer patients. And more technically speaking, what we try to do here, we try to build a method. It can be statistical method or machine learning method, which will estimate the survival function for each patient. So given characteristics of the patient, which can be clinical data, molecular data, we try to have this estimate what is the probability for this patient to survive longer than a certain time after diagnosis. And when we have this model trained, then we can use it for different tasks. For example, we can use it to decide about good treatment decisions. We can also analyze the model and understand what are the risk factors for a given cancer type. And when we train these models, we can use clinical data. This is what we usually do. On top of this, we can use imaging data. And also we can use all this molecular data, mutations, copy number aberrations, DNA methylation, transcriptomic data, proteomic data, and so on. So all these different modalities, I will refer to all this as modalities. And the intuition that you may have is that the more modalities we include, the better the model will work, the more accurate predictions we will do, right? Because each modality should add something complementary to the previous modality, right? However, as you can see from my title, maybe this hypothesis is not actually true. And why this may not be true, this is because these data are very complex, this high dimensional molecular data. Even if you look at one modality, say gene expression, we see that many features correlate. For any gene, you can find another gene which correlates to the first gene with Pearson's correlation of 0.99. So there are these correlations within the modality, but also across the modalities. DNA methylation correlates with gene expression correlates with protein expression. So all this masks causalities, and a model can be lost in having so many correlated variables. And the more modalities we include, the longer the vector that we use to train our model. But we have limited number of patients for whom we profile these modalities. So we may have 100 patients, right? And we have 10 million features. So will these models work? And to answer to these questions, we developed a benchmark study where we profile different models, including statistical models and more advanced deep learning models, to make predictions of survival on different data sets. So in this study published in 2023, we used just TCGA, which is the Cancer Genome Atlas. In a more recent benchmark, I will give you a reference later, we use 4 different cancer data sets. And then we train our models, we assess their performance based on different metrics, and then we can rank the models and say this model works better on this set of modalities. And when we have this benchmark, then we can answer our research questions. The first one is very simple. What modalities are most informative to make accurate predictions in cancers? When I say accurate predictions in cancers means here we take all different cancers, mostly solid cancers, and we look on average which model works the best on which type of modality. Then we can also see which methods can tolerate noise the best way. So if now we have an informative modality and we add some non-informative modalities, which are also high dimensional, which models will keep the same accuracy of prediction, extracting still valuable information from the first most informative modality. And then we also just want to know which methods perform the best, so that if you need to build a survival model, then we can use this model to make the predictions. And so to address these 3 questions with about this benchmark, in this paper is on TCGA only, 17 cancer types. And for the 17 cancer types, we have access to 7 modalities. The clinical modality, this is age, stage, and gender, right? So not so many modalities, plus sometimes we have additional modalities, maybe like smoking status. And then we have gene expression, mutation information, DNA methylation, copy number aberrations, protein expression, and microRNA expression. And then we test different models. So this is the state-of-the-art linear model, which can take into account the group structure. So it will use the first most informative modality to make the predictions. Then it will take residuals, which are not explained by the first modality, make the predictions using the second modality, and so on. So the user provides the order of modalities, and then the model makes the predictions. Then we had also a nonlinear model, which is a block forest model. It's like random forest, but it uses the information about different modalities that, again, can be more informative, less informative when building these trees. And also we had a set of neural networks which combine these different modalities using different fusion mechanisms. So we had a number of neural networks to build predictions of survival of cancer patients based on different modality inputs. And then we assessed the performance. In the paper, we used 2 metrics, anti-leading concordance index, which counts the number of concordant patient pairs. So it just orders the predictions for each for patients, and then it looks whether the order of predicted events corresponds to the ground truth order of events that happen in this cohort, and events can be relapse or can be death. And then we also have an integrated Brier score, which measures the calibration of the model. So this is how close the estimated survival function, how close it is to the ground truth. Today, I will only show you the results for the antilinear C because the other metric provides very similar conclusions qualitatively. So what are the results? I'm going super fast. So first, we asked ourselves what modalities are the most informative to make model predictions across cancer types, which are mostly solid cancers. And here we look at the antilinear C, which looks at the ordering of the patients according to their risk. We see there's 7 different modalities, and here I'm showing just 3 most representative models. Here we have clinical data, which show the best accuracy of prediction if we just use this clinical data alone. The next one is gene expression. And so in clinical data, in this benchmark, we had 5 to 6 variables. In gene expression, we have 20,000 plus variables. And still, we predict much better the survival, it's so much better, significantly better, with clinical variables than with gene expression variables, right? And the less informative modality is mutations. With mutations, the concordance index is about 0.52, and the random one is 0.5, right? So at least, maybe for one cancer type, it will work. But here we look at these 17 cancer types. On average, mutations do not provide information alone to make accurate predictions, while clinical information is very predictive and gene expression as well. So we now know this. There are these 2 most informative modalities. Will combination of them further improve the performance? Yes, it will. So all models, neural network models on the right, statistical model, block forest and priority lasso, they all benefit from including this additional modality of gene expression. However, you can see that the gain in performance is quite small, unfortunately, but it's still significant. If you look at the Kaplan-Meier curves, when you stratify patients into good prognosis, bad prognosis group, it's significant. So it does give you an opportunity to improve the predictions if you include gene expression on the top of the clinical variables. But then we have 5 other modalities. We have mutations, copy number aberrations, DNA methylation. Let's try include it and see what the performance increases. So if it doesn't increase when we include all modalities versus just these 2, gene expression and then clinical, you will see a dark blue square. So here you see that it's mostly light blue, meaning that in most cases, here you have cancer types on X and different models, statistical models or neural networks on Y. In most cases, when you have just 2 modalities, clinicals plus gene expression, you can build a better model than if you use these 2 modalities plus 5 others, which is quite surprising. You say, I give more data to the model, right? I don't remove anything. I just add more methylation. Come on, like in glioblastoma, it's very important. No, on average, the performance goes down because the model needs to cope with more and more data to do the selection of features based on more and more inputs, given the same number of patients. And so the models just struggle, even the best ones, even the ones which were developed specifically to work with different modalities like block forest or priority lasso. And this is just an example how it will look if you then stratify the patients based on these models that you train on the left with clinical and gene expression data, and on the right using all modalities. And this is just 2 representative examples, one for esophageal cancer and one for liver cancer. So here you can stratify the patients on the left, kind of. On the right, you include all modalities and you lose the performance. And this is one of the best methods, right? So now we were interested to see what other actually methods that can tolerate noise. So if you just throw random vectors, which models will still continue to perform well. So we took the most informative modality, clinical modality, and then we just start throwing noise. 1 additional modality, 3 additional modality, 5 additional modalities with just noise. And we see that only the 2 statistical methods, block forest and priority lasso, kind of keep showing the same accuracy, although it still declines a little bit. So, and neural networks, no, they don't. And so, fine. So we saw that not, like most of the models or all models, they are sensitive to adding more modalities which are less informative. But then the question is, on average now, which methods we should use. If we have different options, we can use these two most informative modalities. We can use all modalities, and then maybe we'll add some noise, which we don't know what is the noise in advance, but we just add it. And then we rank different models and these different scenarios, and we see that again, these two statistical models, not deep learning models, outperform the rest. Okay, and one is a linear model, and one is based on decision trees. So now, if you need to train a survival model, you know at least that these two may give you maybe the better result versus maybe more sophisticated neural networks where we have fusion across different modalities. So the takeaway is no method exhibit noise resistance if we throw more inputs which do not contain truly additional information to what we already have in the first most informative modalities. Methods do not outperform on average models which are trained on clinical pulse gene expression only. And also, most methods barely outperform models which are trained on clinical only baselines, right? So they do, but the gain is minor, as you could see. So more models are needed. And this is the gap that I would like to highlight, specifically to the computer science community, how can we reduce this huge dimensionality, maybe using some prior knowledge, biological knowledge, or using foundation models, so that now we have fewer inputs, but more informative inputs. So these models, they don't overfit and don't lose their performance when we train them on so few patients for whom we have data available. And to facilitate this development by the community, we created a leaderboard, which is called ServBird. So this paper now is under review, but the bioarchive is accessible, and also the website is fully functional, where you can download the data to train your model, and then provide predictions, and then your model will be shown here in the leaderboard, and you could see whether you outperform the state of the art, which is currently models which are trained on clinical pulse gene expression, which are statistical models. And also we see that if you train on all cancer types together in the pan-cancer setting, you further improve the performance of the models. So somehow it's beneficial for the model to see 10,000s of cancer patient samples, despite the fact it's not the same cancer type. And the clinical takeaway, the best model is still clinical plus gene expression. The second one will be clinical only. And I would really like to have, as a computational scientist, more data for patients where I have this gene expression data in addition to clinical, to see whether we can actually build better models and further increase the stratification of the patients. And what I see is that rarely generating bulk gene expression data is included in the clinical practice. For some people, it is included, so it's amazing, but I would really be happy if it would be a standard that for every patient that comes to clinic, you have this gene expression data, and then we see what we can do with this data by improving the predictiveness of the models. And on this, I would like to thank you for listening to me. I would like to thank my team. They work on many different things: spatial transcriptomics, foundation models, single cell analysis, heterogeneity in cancer. But today I'm presenting just the topic, which is mostly driven by David and Ayush and Nikita in my team. Thank you very much.
Thank you, Valentina, for a very fascinating talk. I thought a little bit depressing because we are collecting so many data, and we're hoping that the computational biologists will solve the problems for us.
This is on average, right? For one particular cancer.
Okay, there's hope.
There is hope, yes.
Wolfgang, please.
I think it's not that depressing. Coming from what I know about TCGA, the data was not collected with the objective to predict overall survival, but for instance, to understand the disease biologically, to find new targets, new biological mechanisms. So maybe this goal of just predicting overall survival is not perhaps that interesting, and maybe looking at the data for other more biological questions could also maybe be of value.
Of course, yes, absolutely, yeah.
Yes, please, Burkhard.
Yes, so thank you very much. It was super interesting talk, and it's very interesting to see how useless mutations were for you, which, considering all the effort that's been put in there, is rather surprising indeed. But there's the thing that I don't quite understand, is you're referring to GeneX and clinical data, the most predictive value. And then you said adding modalities makes it worse. And I was wondering whether you checked the individual other modalities, let's say clinical and modality 3, modality 4, not combined, but just together with the clinical, whether they were useful or not. Because I mean, if the other modalities per se were useless, then it's not surprising that the addition of noise didn't deliver anything. But I would be surprised if you could really state that mutations are useless, but let's say all the other things are also useless.
So what I say is that alone without clinical mutations are predictive, but very tiny bit, right? Others are more predictive, like DNA methylation is almost as predictive as RNA-seq, right? Then the question is, clinical already has a lot of information in it. So what modality should you take to top up the clinical, right? And what we see is that the best way to top up would be to add gene expression. If you add methylation, then it doesn't really change much. I need to check whether already these combinations are present in the leaderboard that we have. I need to check, and if you're interested, we can also add this prediction so people know for each, because we have per cancer, right? So for example, I'm pretty sure for glioblastoma, DNA methylation will be very informative because we see the ADH1 mutation effect on DNA methylation. So this is a good question. I would say on average, combination of clinical with gene expression would be the best one. But if you look at specific cancers, it can be some other modality.
Yeah, and I'm also wondering, for instance, in hematological malignancies, whether the single cell data sets that come out of the blood would be predictive or not, because that's some indication in the literature and our own experiences that there would be. And I would be really interested if you were to actually run that as your, instead of gene expression, and look at what would it look like if you did that together with the clinical information. Because then maybe you start being able to combine useful modalities per cancer type, right?
Right, yeah. But also what we work is extracting some manual features. Mm-hmm. This can also counteract this curse of dimensionality. For example, we can extract features just doing PCA. We can extract features as like average pathway activation. We can also, so in foundation models, we can get gene embedding. So we can group genes by similar gene embeddings and then use an average expression of this group of genes. So there are different ways how we can continue improving these models.
There was another question here by Davide.
Yeah, so thank you very much for the presentation. What I didn't understand from the clinical standpoint is whether you adjusted this analysis for the treatment, because that's critical, no? The phenotype that you can profile by gene expression may have different interaction with a given therapy. I don't remember the cancer model you were studying, but for hematological disease is different if you treat in one way or in another way. And this interaction between the phenotype and the outcome is tightly related to the type of therapy. Right. And whether you use overall survival also as endpoint, because OS in some cancer may be affected by other reason than tumor progression.
Right. So I think here we use overall survival as an endpoint. Of course, we can use other things, progression-free survival. We didn't use the treatment information, because it's not available for many patients, TCGA or ICGC or other databases we used. Also, because we didn't want to leak the information for the specific question, because we know that when a clinician chooses treatment, he or she already assesses. We see that if the patient does receive radiotherapy, I don't remember which cancer it was, very like after the diagnosis, it means it's a good, very good sign. So the model picks it up, like, right. It's not that radiotherapy works, it's just maybe the clinician knows that it will work because this is a like curable case, right? When we are addressing this question of predictiveness of modalities, we excluded the treatment on purpose. But of course, if you want to use a model for your specific field, then we need to think what information to include to have the best model.
There is a question at the back of the room.
Yeah, thank you for the nice presentation. I wonder if you haven't done the opposite and simplified even more. So have you compared the outputs with just using either the age only as a predictor or the tumor stage? And I wonder how much of the progression-free survival or overall survival is in your work is just driven by the tumor stage?
Right, so in this particular graph, it doesn't come from the first study. I just asked my student to generate it. This one, as that's from our recent benchmark. Here I know we use just 3 variables for clinical: age, stage, and diagnosis. And already you capture most of the information this, if it answers to your question.
Okay, and now the final question.
Thank you for a great talk. So I'm curious, well, you're saying if you have different modalities, the more orthogonal they are, the better it would be for the model potentially. So if we would, well, there are lots of people gathering new data. If you would design a new data set, which modalities would you like to include that are potentially most orthogonal?
I mean, if in addition to gene expression and clinical, so I guess you have many cancers which are driven by translocations, right? So it would be nice to have this information. Also, I'm not sure that RNA data, so RNA, of course, reflects mutations and copy numbers, everything. You can actually even build supervised models to predict whether you have a translocation or not. What you don't have is maybe the clonality of the event when you look at RNA. When you have the genetic data, you can see, oh, actually, I have 80% of cells that do have it and 20 that don't. While in RNA, it's very difficult to quantify this. So I would say genetic data are still very important data, specifically for the cancers where you know that, for example, translocations are main drivers. So I would guess still genomic data.
Okay, but how about like the single cell where you can look at subclones or spatial where you can look at the microenvironment? Would that provide additional?
I mean, of course. So if you, I mean, what we try to do now, we try to build supervised models for survival based on spatial omics. So what we try to do, this is a work which is called GrassMill. Can talk to me about it. Lovro and my team is developing it. So we try to find patterns which are linked to survival. So it will be combinations of features, and this combinations of features means cells in certain arrangements. And we use some cool graph neural network plus attention thing to find them. And it works super, super nice, like all different cancer types. So I think in the future, if this is not too expensive to do, we should go for it. And we see that we can work with the spatial proteomics, even like Akoya. We can work with Xenium when we have much more markers. You can get different resolution, you can different accuracy of your predictions. But clearly, we beat the accuracy of just proportions alone. So the interactions matter. So we have a baseline. What model will predict if we just use proportions of different cell types versus the patterns? Patterns are better. And also, by analyzing this model, we can see which cells talk to one another. And maybe we can have get a hypothesis. What should we break in the interaction between tumor microenvironment and malignant cells so that we actually cure these patients? And we see a lot of interactions with everybody between calves and cancer cells. We see huge classes of neutrophils, which are bad prognosis for medications. So, yeah, thanks.
Thanks.
Yeah, thanks a lot, Valentina.
Thank you.
Thanks for the questions. We had a lot of them. Our third speaker is Wolfgang Huber from EMBL in Heidelberg. He is heading a group for computational method development, has contributed to tools for RNA sequence analysis in particular, and most prominently is also one of the co-founder of Bioconductor, that brings all these statistical tools to the genetic and omics analysis. And as such, we'll also talk about related topics such as data availability and responsible research, reproducible research. Thank you.
Thank you very much for the kind introduction. Does the microphone work? Are we here? Very good. Okay, so this is a bit of a side hobby of mine. Oh, it's loud. It's a bit of a side hobby of mine, but I guess, oops, the reason why this is, okay. Here is probably being tested here. Papers and statements like this. So this is the reproducibility crisis in science. It has probably been with us for the last 20 years or so. This is a paper from 2012 that raised a lot of sort of reactions from Amgen, where so this big pharmaceutical company took 53 different preclinical cancer research studies published in high-profile journals, and they sort of tried to reproduce these results in their own labs because they wanted to use them for product development, and basically failed in a large fraction of these, and then they went public with these statements, and sort of that sort of fueled the discussion on reproducibility crisis. It's also present in other areas of science. I think in psychology, for instance, this has also been a big discussion. On the right-hand side, you have a clipping from a very recent article in Nature where they still sort of complain about the reproducibility crisis. So this is something that is with us, and it has important implications, as I will point out. But partly to counteract this and deal with these, there have been now various different efforts, and I'll be talking about some of these over the next half hour or so. So the main keywords are DORA, Declaration of Research Assessment, and assessment of research or reform of research assessments. A little bit giving these talks, I have given this talk before, is a bit like, you know, going to people and telling to eat a vegetable. So it's all sort of very good thoughts, and we all know it's good, but of course it's also a little bit dull maybe. But actually, I think this has now really gained a lot of, how to say that, current importance, urgency over the last months and weeks, with the changes that we're now seeing in the US into its science landscapes, with an anti-vax person at the head of the health ministry and the NIH being dismantled. I mean, the developments are really dramatic. This is plots of the grants, the money being paid out as a function of time in 2025 and 2024, and you basically see a reduction by over half now research funding. So this is really hurting. I mean, this is basically decapitating science in the US, and this is, of course, this is all happening without any reference to research quality. The decisions on which grants are being terminated is all politics now. And also, another sort of thing that is very shocking about this is that the general public, at least for now, doesn't actually seem to care very much about this. There is this disconnect that maybe scientists have not managed yet enough to convince the general public that what they're doing is important. And that is a topic somewhat connected to research assessment. If, for instance, our own criteria for promoting people and giving funds to people are based on criteria that the general public doesn't recognize or even maybe despises, we have a problem. Well, this is sort of a corollary of the current. This is also something that came out recently in Forbes, this overview over Nobel Prizes over the years. I don't think Nobel Prizes themselves are necessarily a good measure of research performance, but they somehow correlate to some extent with good research environments where those people live, or choose to live once they are successful. So you can learn something from this. And so one thing you can learn from this is, of course, that, for instance, Germany was really dominating that scene in the 1920s and 30s. And then the changes that we all know about in this time led to a complete change of the situation, and everybody basically moved to the US or to the United Kingdom. Now we're maybe seeing similar times. Another point, research assessment. There is a research, I mean, what we do is all based on sort of rational thinking, on the Enlightenment, basically. But in fact, the whole idea of enlightenment and sort of ratio-based approach to life is actually not a given. And we may well be in times when this is all sort of being pulled back. And we all have challenges here. So I want to put this in a bigger picture. How do we do research assessment as scientists? I mean, we all here, of course, have bought the idea that science is good, but we need to organize what we do, our promotions and career structures in a way that is good for the world, or is in the context of the world. Another challenge that is sort of now actually making this whole topic very current is sort of the emergence of AI in science. So I come from an institute, EMBL, that is super excited about the opportunities that AI methods are giving us to improve the way we do science. I mean, sort of we were involved in AlphaFold, DBI. We get prizes for AI. The health minister of Germany comes to us to learn about AI and science and so on. This is all very good. At the same time, of course, there's also things like this. So this is from a recent paper that came out in Frontiers in the Cell and Developmental Biology, a completely made up AI-generated paper with gobbledygook, nonsense text, nonsense figures in a peer-reviewed journal. So there's this whole problem of AI slop now in science, which is, I mean, getting worse. And we sort of also need to defend against that. So that's another sort of an upcoming challenge for research assessment in science. But now, after this sort of maybe somewhat sinister introduction, I want to come back to a more beautiful and sort of more sane and tranquil approach, how we can now actually move forward here. So how do we decide basically who gets promotions, resources, and recognition? But this is what research assessments in the end is about. And a well-tested, sort of very well-working approach that has really been as nepotism. This has worked over decades, over millennia for us. You know, people promote their nephews and their sons and whatnot into the next job. And in fact, this tradition also exists in science. I mean, I have often heard throughout my career when we're talking about job candidates, for instance, oh yeah, he comes from a good lab, he has a good pedigree. We have this, I mean, the well-known networks of Ivy League in the States, so the Golden Triangle in the UK, and maybe similar things also on the continent. So this nepotism is, of course, very present. It's not died out. I mean, we're seeing a resurgence of it. If you just look at the political system in the US. So, but it is a system that, you know, it works. Of course, what it leads to is lack of diversity, the closedness to outsiders, this kind of gatekeeping. The people who work on particular positions tend to be very uniform, very selected, and there's good reasons for assuming that they're not always the most qualified people who do this. We're missing out a large fraction of the world population if we sort of use these kinds of mechanisms to promote and give awards. So in the second half of the 20th century, I think the idea of using metrics in science and sort of to replace nepotism became very popular because it went along with the overall quantification of what's done in society in this time. I mean, for instance, the social sciences became a lot more quantitative. Psychology became full of statistics and surveys and trials in the second half of the 20th century. And we thought that we could just measure everything and control things. And maybe also in research assessment, is there maybe one metric that we can just use and solve the question? And the answer that the scientists found in the maybe 1970s, or starting from there, was the impact factor. It's very curious because this is a quantity that is very much, actually, you can really smell the 1970s in it, in the type of compute that's needed to compute it. It's just complicated enough that you need a PC to compute it, but not more complicated. So, for instance, it predates the internet or everything we have now. That could be much more fine-grained calculation. So, I mean, as you all know, the impact factor is the average number, is the total number of citations to all papers in a journal over a year. I think divided by something else, but the total number of publications that they published in this year. So it's something that you could compute basically on these big paper-based catalogs in the 1970s, and it has somehow now survived and is still with us. This is from a recent paper in, I think also in Nature, and somebody else came up with a slightly different version of impact factor, which they called impact quotient, defined using somehow different arrangements of the numbers. I mean, one interesting point here is there is basic agreement for the really high-ranking journals, so nobody really disagrees, probably that New England Journal of Medicine or Lancet just generally have good research, but then everything down here at the bottom below 10, for instance, is almost completely random. So even small changes in how you compute this, whatever this quantity is, lead to very big differences. So if you ever again see an impact factor quoted with 3 decimal points after the commas, that's certainly a reason to smile. But I guess we all know that there's many different flaws with the journal impact factor, and I don't need to bore you with too much details, but the way it's computed is very sort of susceptible to manipulations by the publishers themselves, by, for instance, putting in front stuff, editorial and reviews, by authors for doing self-citations and whatnot. But maybe the main problem with this, it's really what we're doing with relying in our research assessments on things like the impact factor is sort of outsourcing our core business. Yeah. Because as a university, for instance, when we hire the next round of PhD students or the next round of professors, we should really be interested in hiring the best people. This is our core job. I mean, if we don't do that, then everything else we do sort of doesn't really make that much sense. But by just sort of outsourcing this and, for instance, saying, yeah, so the person has to get whatever 10 papers in impact factor 10 journals and then they become a professor, those kinds of very simple rules, they were meant to overcome nepotism, but what we now do instead is outsourcing our research assessment to some editors and reviewers and publishers and sort of giving away complete control. Another thing, Goodhart's law, very sort of widely valuable law. I think Goodhart himself was a central banker, and this was about macroeconomics, but it's as true in research assessment. When a measure becomes a target, it ceases to become a good measure. One of my favorite papers in this context is this one, that means how to improve your teaching evaluations without improving your teaching. That sort of makes the point of that measures may correlate with what you want to measure, but once you start putting the measure as the objective, things happen. Another thing, of course, impact factor is, so it's based on venue. So it's a journal level metric. Would you, in other areas of human endeavor, would you really judge the quality of the performer just by the venue? I mean, on the left-hand side, you see obviously a very big and powerful venue. It's some country music festival, fine. On the right-hand side, you see Yo-Yo Ma playing on the street in Leipzig. Venue is not everything. Now, the DORA movement, the sort of this movement of research assessment has sometimes been misunderstood as saying, okay, now we should now stop publishing in Nature and Cell because it's somehow bad, and no, let's don't do it. And I'm actually not making that point. I think if you're a good musician, you should by all means aim to play in good big venues. So if you're Yo-Yo Ma, it's great that you go to play in the Metropolitan Opera. Just we should not, in general, mistake the venue for the quality of the performance. And another point that sort of is important in this context is the scientific paper often really only is the advertisement. So this is a quote from John Clairvaux from 1994. The scientific publication is not the scholarship itself. It is merely the advertising of the scholarship. The actual scholarship is the complete set of reagents, the data and analysis step that generates all the figures and tables. Sort of, don't mistake the ad for the thing. It's like for the real thing. Like So, based on all of these sort of various different thoughts and concerns, I think in 2012 alongside the American Society of Cell Biologists conference in San Francisco, some editors, scientists, and policymakers met and tried to get down and sort of wrote down this declaration on research assessment, with which they hope they could sort of move forward some of this field a little bit. One of the main points they make here is that we should consider the value and impact of all research outputs. So not just the papers, but also data sets, software, patents, products, services coming out of the research. So take a more holistic view on the impact of science. And I think this is also very important if we want our science to be relevant to society. Society will see the new drugs, the better treatments, the better survival, for instance, in our context. They don't really care about how many Nature papers we have. So to consider a broad range of impact measures, so we need impact measures, but this concentration of one single one, the impact factor with capital I, is wrong and leads to these wrong incentives and to these distortions. So here is a call back to go sort of make this more multivariate, more subtle, more thoughtful. And the third sort of main point this declaration made is that faculties, universities, hiring or granting agencies should be explicit about the criteria that they use to reach their hiring, tenure, promotion, or funding decisions. So this was, in a way, a grassroots effort. But actually, it's interesting. I see this is really a movement that comes from both sides. It comes from the bottom up. Many young researchers want this because they see that the existing system creates these toxic incentives and environments. On the other side, also actually, this comes from the top, all the way from the European Commission, for instance, or from the big funding institutions like Trust and others, DFG, because they, I guess, also see the need to fund research that is meaningful and that is seen as valuable by society and doesn't just feed a sort of self-serving small clique of researchers for themselves. So we have this big momentum that comes from both sides. I think it's a good thing. If it only came from one direction, there would be many reasons for it to stall. Now that it comes from both directions, this is actually, I think, a very powerful fact. So the last 5 minutes or so, what do we do at EMBL about this? And the idea is, of course, that maybe this is something everybody can think about, whether this is useful for their own home place. So EMBL is a European institution. It's an intergovernmental organization with a mission to perform life science research, fundamental research in science, also to perform training to provide databases and services to the community, both at the instrumentation level and at the data level. So for instance, the EBI, the world's largest bioinformatics biological database, is part of EMBL. It's physically located in Cambridge. So why do we need research assessment reform in our institution? So we believe it improves the quality of our science. We want to do good science and we want to be excellent. We want to have the best people and, for instance, recruit worldwide from all genders, all races, and so on. It's also required by funders, so we have no real other option. So it came to us from the top, from Beck and Trust, for instance, and or NIH, UKI, and so on. They want this. And given that we have to do it anyway, we may as well be a leader and sort of try to move with this fast and not only be reactive, but maybe proactive. So EMBL signed the Dover Declaration in 2018. This is actually 6 years after it was written, but anyway. And we convened a working group to actually implement organizational changes. So not just sign something and then forget about it, but actually change organization to reflect those ideas. So then what did we end up doing? We basically found 3 major fields of organizational change. We changed our messaging. We changed our recruitment and promotion procedures, and we changed the way we make decisions. So now we clearly state EMBL's commitment to the principles of DORA on our webpage. So one thing, for instance, that this could counteract is the self-censoring of applicants that, for instance, think they don't even need to apply for a postdoc or PI position with us because they don't have enough cell papers. This is a very real scenario that I've heard and experienced in several cases, but now clearly stating that our view of research excellence is broader and in a way more sane. I hope we reach a larger audience of people that, for instance, would apply for jobs with us. We updated our recruitment material, so people now are told that they need to supply a broader range description of research outputs than just something like a short table that only focuses on journal names, for instance. And also, I mean, with retraining our internal and external decision-making committees, so many of our senior scientists are, of course, lots of granting funding boards or grant review boards and so on, and sort of slowly trying to retrain people to think in this broader way, which is not always easy because they all grew up in the existing system, and many of them became big with the existing system of impact factors. So convincing them to let loose of it is an ongoing discussion. So yeah, this is just maybe more detail about these 3 points: clear statement of our commitment, update of the type of research outputs, and guidance to our decision-making commitments. So, research assessment is hard. There are no easy ways out. Just, for instance, counting impact factors on somebody's publication list is obviously, it's very simple and in a way very objective, but it leads to all of these problems. There is no other easy solution. It is just hard work. But as a research performing organization, it's actually also our core job. So being good at this, so we have to actually invest in doing this well, because it's the core expertise of a research performing organization, and it is differentiating. And you can see difference between organizations that do this well and not. You cannot read all the papers of everybody all the time and understand them. There is some need to pool expertise, to outsource, to team up and use proxies. So I think we need to recognize this and find good and sane ways to use proxies and outsourcing for some of this work. And with this, I'd like to thank the DORA committee working group at EMBL, who developed many of these thoughts over the last years, for their really excellent input and discussions and work, and you for your attention.
Yeah, thank you for the presentation. I think DORA is something that we here in Switzerland know quite well, because the SNF is implementing it in a Swiss style, and we might have questions, comments maybe even. Yes, thanks. Yeah.
I think you can do that. So that was great. And ultimately, again, depressing, because I think the old outsourcing was terrible. But what Björn just talked about, what we're doing in Switzerland right now with the SNF, is perhaps even worse. So, and unfortunately, you haven't come up with any idea as to how to do it better. Now, I don't have one myself either. We think about this all the time, and I think everything we do is pretty crummy at the end, and we end up playing around with this, and I really don't know a good solution. So I had an idea that I wanted just to pitch to you into this DORA thing, and it's like, if you were to compare, let's say, 2 papers on the same subject, whatever the subject is, one is published in the New England Journal or whatever, one of the boutique journals, and the other one in the North Korean Journal of Shellfish Physiology, for instance. And they are both very similar, and you then have the citations. Would it make sense to divide the citations by impact factor to find out which one is the better paper? There are some efforts in that regard that basically show exactly that, so that a paper that's reasonably well cited in a non-boutique journal will actually if that is a much, much better, higher quality paper after all, because the aficionados, the people who really care for the research, actually do the research. How good is the paper, and cite the better paper and not the flashy paper. I don't think this will work overall, but as I thought, a bit of food for thought for you to actually start comparing that. I mean, if this is an activity you do, I think it's super needed. It's fantastic that somebody does it, but I'm still frustrated that there's nothing good out there.
No, so I think we need more metrics, and your metric, this is an interesting normalization. I don't think it's a plug-in replacement for what we have now. I think it's one more. So in the end, we will have a multivariate, so then we're back to omics, a multivariate phenotype for each paper, and this quantity may be one of the useful ones. Of course, the hurdle for the authors to get into New England is higher than maybe for that other. So there's also some indication of maybe the amount of work they put into the one or the, but then there's all of these other confounding factors. So I mean, there are no easy solutions, but taking into account that is very important. I mean, at another level, for instance, when we look at group leader candidates at EMBL, if somebody comes from a lab in Harvard, they where they publish in those journals twice a year anyway, versus somebody that came from a small university in Southeast Europe, I mean, you do normalize their output with such factors, of course.
Yeah. Do we have other comments or questions? Thorsten.
Here we go. Sorry. Yeah, thanks a lot. Great talk. I mean, one of the topics that is a problem I see is the inflation, the sheer number of publications. And I wonder, I mean, I for someone entering a field, it would be very hard to tell, like for Burkhardt, it's easy, right? But for someone new, it's very hard to tell a good paper from a bad paper. So can you comment on how much of this is affected by the sheer increase in numbers of publications?
I don't know. I'm not that worried about inflation, to be honest. I mean, I think it's a bit like YouTube. You have millions of cat videos, but there's still also useful stuff in there. And if you know how to find it, you will find it. And now we have the internet and recommendation networks and Blue Sky and maybe AI that can help you guide and find the stuff that you really care about. So yeah, I think we are navigating a lot of, I think having an abundance of information and then navigating it is probably okay and better than having something that's very small from the beginning.
Do we have other questions or comments? Markus.
Thank you. I mean, I was for a long time in industry and we had exactly the same problem. How do you measure the performance of the people? Then we had these key performance indicators. And a colleague of me cited a philosopher of the 18th century, so it's not a new problem. Since we cannot measure what is important, we give importance to what we can measure. Mm-hmm. And I think, I mean, what we do at the end is a qualitative assessment, but we transform it into something quantitative, which is always something which is very delicate. Mm-hmm. Now, regarding the SNF comment, I was in the SNF Research Council for some, and we had those discussions. And of course, one of the criteria which we have to look, what's the value of this type of research? You may say for society or whatever. And this, of course, is a problem for anything which is basic research, because you cannot add anything to that at this stage. Mm-hmm. And I think we have to be careful with those type of criteria which we formulate.
Yeah, I completely agree. I mean, CRISPR is an example. Lipid particles for vaccines are an example for mRNA. Yeah.
All right. Do we have other comments? Just because you were citing my SNF citation, there's also an element of implementation to it that is very difficult. So it's a very good direction and to move towards, and food for thought at every level of a decision. The question is of what happens on the other side of an administrative process is maybe also something that need to be very carefully designed and implemented, and also requires thought on the administrative side. Okay. Thanks a lot for this talk, for adding this important aspect to the methods we heard of, the comparison of the tools we heard of, the comparison of methods, all the decisions that are in there that are all important for the type of data. If you have digital data, everything scales up. Even the publications that are published in bioinformatics, maybe more than in basic biology research, and questions that arise. So thanks a lot for joining us here in this session, and thanks a lot to our three speakers. It was a pleasure to listen here. Thanks for your insights, and I think some lunch is waiting outside. See you after lunch break.
So we have the lunch and the poster session, and we also want to take a photo of the speakers and the chairs. And if you also want to be on the photo, just come up. So we'll take a photo now in the front.
All right, so good afternoon and welcome back. And my name is Burkhard Becher, and this is Chiara Magnani, and together we will chair this next session, where we talk about yet another of those modalities that we talked about earlier, which at least has become a staple in most of precision medicine activities around the world. And I still have some hope that that it'll perform better in the next analysis that you're doing. So without further ado, I'll pass on to Chiara.
Yes, a very warm welcome also to my side to this topic, single cell technology applied to precision oncology problem. And as a first speaker, it's an honor to introduce Judith Zaugg from the University of Basel. And she's going to talk about gene regulation in blood cancer. And the stage is yours. Thank you.
Okay, yeah, good. And my presentation, is it coming maybe? Good afternoon. Happy to be here. Thanks for the invitation. Kein Signal. Yeah, okay, good. Yeah, so it's my great pleasure here to talk a bit about our work, and I changed the title a little bit. It is about gene regulation, but also about the cellular crosstalk that we have in the bone marrow niche. And just to give a bit of an introduction of what I find very exciting about biomedicine, so I'm a molecular biologist or computational biologist, a mixture of the two. Is to really understand what makes each patient or each individual unique. And of course, when we talk about the uniqueness, we have tens of trillions of cells that somehow make up a phenotype that looks like how we look today. And when we think about phenotypic variation across patients and individuals, of course, we have these kind of sometimes genetically determined differences, such as how you would respond to drugs. But then also we have, of course, differences among individuals, such as age, that kind of determines of how you respond to, for example, a coronavirus infection. So I think there's really kind of different levels of variation that we have across individuals and patients. And what we are trying to do is we're trying to use this kind of systems epigenetics approach to understand or to use this phenotypic variation on the molecular level to understand disease mechanisms. So that's kind of our overall goal in the group. Some of the approaches that we're using kind of rely on this molecular profiling, mostly on RNA, chromatin, and increasingly also surface receptors that allows us to build gene regulatory networks or networks in general. And the reason why I think gene regulatory networks are really interesting is because, or like are a great tool to look at kind of disease modalities, is because they really allow you to integrate different, can you see my pointer now? They allow you to integrate the different levels, right? They allow you to integrate the genetic, common genetics, mutations, epigenetics, as well as kind of intrinsic, extrinsic signaling pathways through the action of transcription factors. We've built some tools. We also use a lot of tools that are available for gene regulatory networks. And then we have actually the one approach that we're using a lot is then trying to build networks based on these molecular heterogeneous cohorts to then look at molecularly stratified cohorts to understand disease mechanisms and kind of predict which direction is kind of the information flowing in a patient versus a healthy person. So this is essentially kind of a framework that we've built to generate molecular hypotheses from multi-omics data. I want to now actually switch gears a bit and go more into the biological topic of this presentation, which is the bone marrow. And of course, you're probably all familiar with the bone marrow. I just want to highlight what I find exciting about the bone marrow, which is essentially this assembly of a lot of these immature cells in the immature blood cells, right? But then it, of course, has these hematopoietic stem cells, very rare cell population that gives rise to the entire blood system. And I always find it super crazy to think about that it produces about 5 billion cells every day. And of course, it's all regulated by transcription factors, right? This whole differentiation process. And then we have these other types of cells that are the mesenchymal stroma cells that are the supporting cells of the hematopoiesis. So these are essentially the cells that allow the hematopoietic stem cells to survive, to stay there and to kind of generate all the blood cells. And we not only look at this, of course, we're then also interested in these kind of malignant transformations of these hematopoietic stem cells in diseases such as acute myeloid leukemia. You're probably more familiar, some of you are more familiar with this disease. I'm not a clinician, but of course I know a little bit about the dismal survival rates of acute myeloid leukemia, which has a very poor survival rate over the 5 years. And also one of the things is it's maintained by these leukemic stem cells that can often then hide in the niche. Another interesting aspect, of course, with of the bone marrow is also the relationship to the immune dysfunction that happens with aging, right? So this is all also happening in this same organ. So we are generally interested in understanding how these cells interact with each other during transformation and beyond. And kind of to just schematically show what we are working on in the group is we kind of treating anything that comes that can disturb the bone marrow microenvironment, such as rare immune disorders, malignancies, pre-malignancies, drugs, therapies, or age, trying to understand the interactions and then kind of maybe understand something about disease and resistance mechanisms. So this is the overall picture of what we are doing in my group, and I want to give now two examples of two things that we have recently been working on. One is the role of T cells in the success of allogeneic stem cell transplantation. And this really shows also the impact of looking at the non-malignant cells in a therapy response. So just brief introduction into allogeneic stem cell transplantation. For those who are maybe not so familiar with this is essentially a treatment that a patient gets a high dose of chemotherapy and then receives a transplant from a healthy donor of the bone marrow containing hematopoietic stem cells or hematopoietic cells in general. These cells then hopefully induce this kind of T cell, this graft versus leukemia effect, where the T cells of the donor would kill the remaining leukemic stem cells that are still sitting in the patient potentially. And then, of course, one question is that sometimes, even though many people go into complete remission after this treatment, there's some people still that actually relapse after the treatment. And our question here in this project, and this was a joint project with Caroline Pabst at the University Clinic in Heidelberg, driven by our joint PhD student, Anna Mathiodaki. And the question was really, how do the T cells interact with these leukemic cells, and what is their role in therapy outcome? So the setup, how we started this study was with a really very small cohort of 3 patients that later relapsed and 3 patients in complete remission. And we took their bone marrow aspirates at day 100 after transplantation, where all the patients were still in remission. So that's very important. There were no blasts discovered in these patients. They only later relapsed. We then sorted the cells. We specifically isolated the CD3 positive cells, so the T cells, and already just looking at the and then we did single cell RNA-seq, and already just looking at the single cell RNA-seq data in a UMAP, you could already see there's a very distinct profile for those patients that went into remission and those that actually later relapsed. When we a bit further quantified the kind of the proportions of the cell that changed between those that stayed in remission and those that later relapsed, we found that it's generally a downregulation of this kind of naive population in the remission samples, or an increase in the relapse. And the remission was specifically characterized by this compartment of CD8 memory cells and CD8 effector cells being more abundant in the relapse. So we looked a little bit more into this population. We stratified them based on their differentiation from naive to mature. So we calculated the pseudo time of CD8 T cell maturation. You can see here the different genes that we kind of see them nicely aligning along the pseudo time. And one very striking observation was here that in the complete remission, most of the CD8 T cells actually managed to go into full maturation, whereas in the relapse patients, we had a lot of cells here still sticking around in this kind of naive form. And when we quantify this, just looking at the pseudo time value of the CD8 patients, we find that there's a lot of these immature cells still in those that's relapsed. So since differentiation is driven by transcription factors, we actually wanted to understand how this may be actually affecting the transcription factors and the gene regulatory models. So we inferred gene regulatory models based on transcription factor gene interactions using a cynic algorithm here in this case. And we then specifically asked for the different transcription factors, are they more enriched in targets that are upregulated in the relapse here in green or in the remission here in blue? And there was only really one transcription factor that was specifically more activated, had a more active regulon, like that its genes were more activated in the remission. And when we looked specifically at this transcription factor, it was also clear why it was maybe more important for the remission. It had all these kind of cytotoxic genes, the granzymes, the KLRs, and also one very interesting gene for us was then this ADR-GR1, also known as GPR56, which in the leukemia field is very well known as a stemness marker in leukemic stem cells. So we were quite surprised actually to see this also on the T cells, and we wanted to follow up a little bit further on this. And it turns out it was really a strong marker between that kind of differentiated the relapse and the remission patients. So it was essentially across the entire pseudotime, it was always upregulated. Whereas, for example, the CD27, this early T cell activation marker, was just activated in the beginning and then really went down in the complete remission. And we validated this also in flow cytometry. So really this protein was highly overabundant in the remission patients. So we wanted to understand a bit more what is the function of these GPR56 positive T cells. And for that, we stratified our population of T cells into the ones that were positive for this marker and negative. And you can already see here, there's a lot of these perforain granzyme Bs, a lot of these cytotoxic genes were specifically upregulated in the GPR56 positive population. We validated this also in an independent cohort of 10 patients where we again stratified sorted the cells into GPR56 positive and negative cells. And again, we see these two markers strongly enriched, strongly expressed only in the GPR56 positive cells. We then went on to even do CAR T cell experiments where we tried to understand what happens to the GPR56 level when the cells engage with their targets. And what we found is that essentially the GPR56 positive fraction gets induced only when the target is there in the cell types for the T cells that actually had the CAR against the target. So essentially, we saw that this is really a marker that gets induced by antigen exposure. We then also, the final question we asked here was then whether these GPR56 cells are better at eliminating the blast cells. And for this, we used an assay that's called the ELISpot, where we essentially take the AML blasts, the original blast of the patient, and then we take the PBMCs, isolate later on, like after the transplantation had happened, sort them into the GPR56 negative and positive T cells, and then measure whether these cells start to have an interferon gamma response, which would be indicative of the blast killing. And what we saw was quite striking, which is that in the GPR56 negative fraction, we find very few of these spots, and in the GPR56 positive, we find a lot of these responses. So essentially showing that really it is this GPR56 positive fraction that is killing the blast cells. So essentially, what we have kind of, to summarize this part of my talk, we have found here a kind of a difference between the complete remission and the relapse. The remission was really characterized by the CD8 effector cells that had a very high level of this specific marker gene, whereas the relapse patient had this kind of inflammation response that didn't really manage to mount a proper cytotoxic cell response. So the outlook is now, of course, whether we can leverage this mechanism to understand or maybe counteract therapy resistance. And with that, I want to park this thought for a bit and go to the next, to my next topic, which is the progressive remodeling of the bone marrow niche now in the pre-leukemic conditions. Right. So this now we take a step back and we go back from the AML to kind of understand, and here we want to understand specifically the interaction between different cell types, but just we take a step back now and go, you know, in a pre-leukemic condition, when we kind of just look at this again in hematopoiesis, we have the healthy hematopoietic niche with all this differentiation. Then there is certain mutations that kind of predispose clonal hematopoiesis, where you suddenly have a kind of an expansion of one specific clone. This is usually pre-malignant, and doesn't have a specific clinical impact. But of course, this can then progress to something that is called myelodysplastic syndrome, where then some of these clones start having more mutations, and then suddenly you become there is a problem with the blood production. And this is now a collaboration with Borjan Geskes and also with Uwe Platzbecker, who provided us with all the clinical samples for this study. So this is a kind of a big cohort of patients that we have used from this BOHEME study, which is a study that followed people longitudinally for different, for like even healthy individuals. So we had in the end a way to kind of have control samples kind of roughly the same age. So this is our age distribution, it's mostly elderly donor. We had then these clonal hematopoiesis patients and also this low risk MDS. And this is kind of the overview of the cohort. What we essentially did is we have bone marrow biopsies, but also bone marrow aspirates from which we did different types of analyses. And I'm going to guide you through this now a little bit. So the first question was, how does the overall bone marrow niche change in healthy clonal hematopoiesis or these MDS patients when we use targeted bulk sequencing? And I'm showing you here kind of an overview of the data. So this is a PCA of the cohort when we just do a targeted panel on these immune exhaustion genes. You can clearly see the MDS is kind of grouping on its own, the cheap and the healthy kind of clustering a little bit further away, but like not strikingly far away. We then actually used this data to kind of disentangle a little bit of what's happening. We used a single-cell RNA-seq data set that was published at the time already, and we kind of did a kind of a deconvolution of our signal in the different cell types. And what you can see here, this is the bone marrow populations that came from the single cell data set, and this is just our genes that are up or down in MDS. You can firstly see it's mostly down-regulated genes in MDS. And then also you can see specific populations that are interesting here. For example, the CD8 populations had a very strong signal, but also some of the stroma and some of the endothelial populations. We also found some effects on the B cells, but that when we looked at the FACS data was mostly related to just having less B cells in this bone marrow. Whereas when we look at the T cells, for example, there was no difference in terms of the abundance of these cell types. So that's why we decided to kind of focus on these three populations to look at these three populations, at the T cells, the hematopoietic stem and progenitor cells, and the stroma cells. And then we also had some monocytes, but that were actually not intentionally coming. We tried to get rid of them. They were just here. So I'm not going to focus on the monocytes. Anyway, so we have these nice three populations. Here the hematopoietic stem cells, here the T cells, and here the stroma cells. And I have to say, the stroma cells, isolating the stroma cells from bone marrow aspirates was quite an effort. And we did a negative gating on all the blood markers, trying to get like up to between 100 and 200 cells per donor. So this was really hard to get those cells. But I think in the end, it was really valuable to look at these, because these were the cells where we found really a very strong difference between the patients. So first of all, let's look a little bit at the stroma cells. I'm showing you here the different cell types. And one striking population that we identified here was this we call it inflammatory MSCs, that had all these kind of inflammatory markers upregulated, and also kind of this ECM remodeling upregulated. And what was quite interesting to see is that we find these IMSC populations. I'm showing you here the proportions for the different indications, the controls, the CHIP and the MDS. And you can see here on the IMSCs, essentially the green dot is missing. So these were really exclusively present in our CHIP donors and in our MDS donors. It was interesting, though, that we already found them in our CHIP donors. I think this was something that we did not necessarily expect. So the clonal hematopoiesis. We also saw a dramatic decrease in these adipocard cells in both the CHIP and the MDS. We also validated, and again, some of these populations using a flow cytometry and also imaging that I'm just showing you here, one marker where we find specifically actually in the splicing mutations of MDS, we find a strong increase. So we wanted to understand a little bit further what these IMSCs are. And for that, we looked first at the main role of these mesenchymal stroma cells, which is to support hematopoietic stem cells. I mean, in addition to being the support, kind of the precursors of bone and adipose tissue in the sense of hematopoiesis, their main role is to support hematopoietic stem cells. And here we look at the signature of hematopoietic support by the stroma cell type. And this has been shown before, that the adipocars are really the cell types that are mostly supporting the hematopoietic stem cells. But what was quite interesting, that even in the IMSCs, actually we found some cells that are quite supportive still of the stem cells. And when we look at this kind of in a bit more kind of resolution here, when we kind of compare the inflammatory signature of the stroma cells with their support signature, we of course see in the healthy there is no inflammation, so there's kind of a whole spectrum of support signature. With the CHIP, we find that the inflamed stroma cells are the ones that also have a lower support. But what I thought was quite interesting is that in the MDS patients, actually the inflamed stroma cells are the ones that are still supporting the hematopoietic stem cells. And that's also kind of in a way shown again by looking at these cell-cell interactions. So we run a cell-cell interaction algorithm, and what we can see here in red is the IMSCs, and what these bars are showing is just a number of interactions that these different cells have with any type of hematopoietic stem and progenitor cells. And we find that this role is mostly overtaken in the adipocars for the healthy donors, but then it slowly shifts towards these IMSCs in the MDS patients. So really indicating that these are the ones that are supporting the hematopoietic stem cells in these conditions. So then the next question we asked was, and also what is quite interesting actually is that inflammation and support that they're not mutually exclusive programs. So then we were wondering what may contribute to this inflammatory reprogramming. And the first thing we wanted to look at was, of course, the hematopoietic stem cells, because this is where the mutation is harbored in the CHIP and in the MDS. Just kind of, again, confirming kind of the finding that we have just a general, in the hematopoietic stem and progenitor cells, we have a general loss of these kind of pre-B cells. We have a bit of an increase in these monocyte-like cells, and otherwise there's not that much change in the overall distribution of these cell types. We then looked at the trajectories, and we wanted to specifically understand whether there's some kind of inflammatory molecules that are being expressed by the different hematopoietic cells across the differentiation. And we looked here at CXCL8, also known as IL-8, one of the pro-inflammatory cytokines. And what you can see here is that in the So I don't have the annotation here, but it's the same as here. So the purple one is the chip, the red is the MDS, and the green is the control. And you can see a strong, high expression of these pro-inflammatory cytokines in the chip. So indicating that in the chip, it's maybe really the clones or the hematopoietic stem progenitor cells that drive the inflammation. But in the MDS, this was less clear. So the only one that really had a bit of a spike in these pro-inflammatory cytokines was the late monocytes. So we then also were wondering whether the MDS blast directly contributed to this inflammation. And for this, actually, one of my students developed an algorithm called SpliceUp that allowed us to identify in the single cell data the cells that have an SF3B1 mutation based on the missplicing profiles of these cells. And you can see here kind of the predictions. So in green are the wild type predictions, in red are the kind of the mutation predictions. And what is quite confirming here is that we find a lot of these erythroid progenitors with the mutation, because that's also the phenotype of this MDS. When we then looked at the differential expression between those cells that had the mutation and those that didn't have the mutation, there was quite some differences, but the only gene really that was a little bit related to inflammation was this interferon response gene. And generally, when we looked at the GO processes, the ones that were upregulated in the mutant were more related to kind of RNA translation, and so maybe more kind of proliferation terms. And the ones that were upregulated in the wild type, so down in the mutant, were differentiation morphogenesis. So nothing related to inflammation at all. So we think that the blasts have a limited direct contribution to this inflammatory milieu. And also this was we then wanted to further investigate this in a co-culture system in vitro, where we had cultured low-risk MDS line with bone marrow-derived MSCs. We then looked at the supernatant and did proteomics on this to try to see whether there's some kind of an inflammatory signature popping up. But essentially what we find when we look then specifically at the genes that are expressed by the stroma cells, because of course the supernatant is a mixture of both cell types, we find mostly genes that are actually expressed in these IMSCs and adipocare, and mostly they were kind of support genes, so AML, like leukemic hematopoietic support gene, rather than inflammation. So it really seems not the direct interaction of the blasts that give rise to the inflammation. So then, of course, next we turn to the T cells. And for the T cells here, again, just showing you the UMAP, and we find here the one population that was really different in the MDS versus and the cheap versus the healthy was this population that we termed interferon responsive T cells. You can also see them here. They were essentially characterized by these two kind of interferon responsive genes. We also kind of validated that in imaging. So you can see here this population was strikingly enriched when we look at the bone marrow biopsies. And when we looked again at the interactions, at the niche interactions, we find that it's mostly this population that was actually interacting with the IMSCs. So what we're thinking here, and I'm showing this here actually in the next slide. So here's the T cell interactions with the IMSCs, and it's really strikingly mostly these interferon responsive genes that are interacting with the IMSCs. So this is now really kind of a hypothesis that we have, that maybe this weird interferon responsive T cells that are driving this kind of inflammation that's happening in the bone marrow in the MDS. And now, what are these interferon responsive T cells? And now I'm going back actually to the first part of my talk, when I talked about the T cell landscape in allogeneic stem cell transplantation. And we indeed actually also find this exactly same population of T cells again in our relapse patients, specifically upregulated. So it's exactly the same markers here that we find. And this kind of indicates that really there is some kind of weird inflammation going on in those patients that did not respond very well to the allogeneic stem cell transplantation. So going back to the summary and the outlook, I showed you already this kind of whether can we leverage maybe this part of the equation to improve T-cell based therapies, and can we kind of try improve this part of the equation to kind of maybe try to see whether there's a way we can treat the bone marrow niche to improve therapies. And we just actually got funding from the Swiss National Foundation to kind of look at this in a little bit more detail. So in the future, what we want to do, we want to quantify bone marrow plasticity over the time course of allogeneic stem cell transplantation. And we want to do this in spatial niches. And I'm just giving you here a sneak peek of some really cool data, you know, where we can now look at the bone biopsies, and we can even within the same biopsy see different niches. Here, for example, one that has a lot of adipocytes, one that has low blast, one that has high blast. With that, I'm ending my presentation, and I'd like to thank everyone. I think I've pointed out the people that have been joining me in these research projects. It was mostly Karin Prommel, Maxim Kolmatov from my group, Eva Sporen-Geskes from DKFZ, and Caroline Paps from the University Hospital. Thank you very much.
Now it's green. Okay, now it works. Sorry about this. Thanks again. Those who haven't heard, I thanked her. So thanks again for this exciting presentation. I'm sure there are some questions. And while people are collecting themselves and what they want to ask, I have a question to the first part where you talked about your the analysis of the after stem cell transplantation. I understand that the data was basically you looked at what happened after, how well did it work, and I was wondering, did you correlate that actually with the flip side of the coin with GVHD and see whether your strong, potent GVL response was also more associated with the development of GVHD? And the other question is, to me, the most exciting aspect would be if you had access to the transplant before it was transplanted, and did you see any predictive features of this is a good one, this is a good transplant versus a bad transplant?
Yeah, this is exactly what we're actually at the moment doing. It's very hard to get access to these materials. So if there's anyone that has access to this material before transplantation, we would be very interested because it's, I don't know, ethically, it's very hard always to get access to this. But the second question, so that the graft versus the host disease, we did exclude those patients that were dying from graft versus host disease because it was a little bit, you know, this was not really what we want. It was a retrospective cohort, right? So we had to be very careful also with selecting the patients. But one thing I want to mention is that we did not see this GPR56 upregulation when there was absolutely no graft versus host disease present, right? When there was absolutely, when the patient just probably had the leukemia cured by the chemotherapy, then we would not see that. But we would always see it when there was a little bit of graft versus host disease. I mean, you know, I'm not a clinician, but my clinical collaborator tells me you want to see a little bit of this graft versus host disease to make sure that this reaction is actually happening. So kind of the mild cases, this is what we have been including. And I think this is what we are, this is when we see the signal.
Yeah, but there's a little disconnect potentially because maybe GVL clearly shows us this T cell phenotype, and that's very compelling. But GVHD may have an additional component where it's actually worthwhile looking at the phagocyte compartment, activated macrophages, etc., the ones that you find in some monocyte.
No, I think it could be interesting. Yeah, we have actively excluded those cases here.
Have another look if you can. All right, Chiara.
Yes, I have also a couple of questions. The first part, first of all, if you correlate the marker expression with the TCR clonality, because I think at the end of the day, you can also basically select the patient for different mismatch and therefore TCR recognition. So maybe the patient that are not responding are the ones that just don't see the antigen, and this is a antigen stimulation response. So if you can maybe talk about a little bit about that.
Yeah, I mean, I cannot talk much about it because we are just currently doing this actually. So we're currently looking, we have not done TCR sequencing on these patients, unfortunately. But we are doing that now with a new cohort, and where we are looking specifically actually at the donor, at this lymphocyte infusion, where we also see some clones expanding. But you know that GPR56 also is actually a marker for T cells that are responsive against CMV, so cytomegalovirus, which is a huge part of the population is affected. So also when you want to look at this in the clinic, you also have to stratify actually the people that are positive and negative for CMV, because that's also, so you will have huge clones, CMV reactive clones of GPR56 positive cells.
Yeah, I can imagine. And another follow-up question, can you use this marker to predict the remission or the relapse? Have you tried to do that? And
that is
when
what
is
we are
the
currently
best
trying,
time
exactly.
point?
Yes, so that is what we're currently trying. And as I kind of explained already in my answer to the first question, that the challenge there is that this marker is only predictive if there's really like a minimal residual disease happening. So, you know, the cohorts have to be really stratified, right? Because we would not see any upregulation if the patient was cured from the beginning and there was no remaining stem cells to be attacked by the T cells. So it's a little bit tricky.
Last opportunity. 1, 2, 3, go. So thank you very much. Thanks a lot. Yeah, give me the other one then. Okay, it's probably just not, it's not full. All right, so distinct pleasure to introduce the next speaker. It's our own Alexandre Theocharides, who is from this clinic here, the Department of Medical Oncology and Hematology, and he has the long and complicated title that he picked, AML, which I think is cool. So please, the floor is yours.
Thank you, Burkhard. Yeah, I think this title was given to me and I didn't specify, so it's, it has some advantages and some disadvantages. So thanks for inviting me, and I'm happy to talk here today. Judith said she's not a clinician, I'm not a biostatistician, so maybe it would be more appropriate for this crowd to be a biostatistician, but let's try to talk about acute myeloid leukemia, and I think some aspects I will cover were already touched, I think, this morning, and will nicely match what I'm going to talk about. I will first start with discussing some data that is not from our lab or from our clinic, but to introduce the AML maturation state as another layer of complexity in acute myeloid leukemia that can be integrated with other omics technologies. Then discuss data that we have generated within the Tumor Profile Consortium, and end with the clinical trial that we have set up in the same category of patients. Now, it's always good to repeat that hematopoiesis is organized as a hierarchy, and the hematopoietic stem cell stands at the apex of this hierarchy, can differentiate into progenitor cells that eventually give rise to mature cells that circulate in everyone's blood. Now, in leukemia, in acute myeloid leukemia, mutations that confer self-renewal capacity arise and give the self-renewal capacity to the leukemic stem cell clone that outgrows normal hematopoiesis. And the morphological hallmark of acute myeloid leukemia, as you know, are the leukemic blasts. We heard about these cells, and these cells typically do not have the potential to differentiate into mature cells, but we can find them in the blood and we can find them in the marrow, and that's how we make the initial diagnosis for these patients. Now, the genes that confer self-renewal potential to hematopoietic stem cells are widely known, and this review from Hartmut Dönel categorizes different genetic different mutations in categories that we know. There could be mutations in tyrosine kinases. Let's see if this works. Oh, this works. Doesn't work, but okay, doesn't matter. In tyrosine kinases on the upper left, or in epigenetic regulators like TET2 or the NMT3A, we heard about it before, TP53, tumor suppressors, or there is very specific mutations like mutations in nucleophosphine 1 that is well known and quite frequent actually in acute myeloid leukemia, and we just heard about mutations in the spliceosome, SF3B1 being one of them, but there is other spliceosome factors that can be mutated in AML. Now, again, I don't need to introduce multi-omics to this audience. Certainly, it's a way to describe life in a concerted way. And what I want to focus on today is really the integration of different omics technologies to characterize acute myeloid leukemia. And it's also quite obvious that multi-omics have became very popular, and the exponential increase in publications, and this is already 4 years ago, just underlines that we are working in a very attractive and timely field. Now, we are used to use flow cytometry to characterize the hematopoietic hierarchy, and I put old on the left, not because it's not used anymore, but it's been here for a while. And flow cytometry has many advantages, one of them being that you can also sort these cells, you can isolate the purified cells and transplant them, for example, into xenografts to do functional analysis of, for example, leukemic stem cells, and I think this has been very successful in the past to characterize indeed the leukemic stem cell function. Now, on the other hand, single-cell RNA technology has become very attractive to characterize acute myeloid leukemia, particularly the stem cell and progenitor cell compartment as well. And as you know, this obviously needs computational analysis and data visualization, as we just saw before. And on the right side, the author from the Van Galen group in Boston integrated single-cell RNA sequencing with genotyping to come up with a description of the maturation state of acute myeloid leukemia. And there you can see that there is not only leukemic stem cells, but that they're in the AML compartment, let's say, there is indeed cells based on their signature that are more stem cell-like, there is some that are more progenitor cell-like, and indeed there is some AML cell-like, the AML cells that may be more like mature cells, but they all belong to the same AML clone. And so the single-cell RNA sequencing defines the AML maturation state. And the big advantage of RNA-seq is that it can be integrated with other omics technologies. And that's what I'm going to focus on a bit in these studies. And so in the BEAT AML trial, single-cell RNA sequencing was, I think, one of the cornerstone of the study, was integrated in particular also with some drug screens. And I will show you some data about this later. On the right side, you see data from the tumor profile, and I will talk about the study later on, just to show you that we've adapted the single-cell RNA sequencing as well to characterize some of our AMLs here. And of course, we can also look at immune cells by the same approach. Now, coming back to the BEAT AML study and the question about how the maturation state and genetic aberrations can be integrated. And I think this comes back to one pertinent question that we heard this morning, which I think alluded to the fact that genetic aberrations may not always be the most relevant, in particular in AML. Now here you can see a correlation of the AML maturation state, again defined by single-cell RNA sequencing, and some clinical parameters that are associated with the risk of AML. So typically we would classify acute myeloid leukemia according to the European Leukemia Net into favorable risk in green, and these are known cytogenetic aberrations that are associated with a favorable outcome in AML. And what you can see here is that these favorable genetic aberrations are associated with a more mature maturation state, and on the other hand, mutations or cytogenetic aberrations that are associated with adverse risk are associated with a more primitive state of the AML. I think, this may be quite obvious, but still, I think had to be shown. And even if you take a single gene that is frequently mutated in acute myeloid leukemia, and actually also very frequently mutated in CHIP, DNMT3A, a gene of an epigenetic regulator, there can be be different mutations in that gene. One of the most frequent one is the R882, and this mutation allows maturation according to the single-cell RNA sequencing in that study. But there may be other mutations in DNMT3A which confer a differentiation block and rather a more primitive AML state. And that also is true if certain DNMT3A mutations are combined with other mutations. So really, the genotype and maturation correlation really becomes crucial in that case. And I would also bet that mutations in DNMT3A, which confer a more immature phenotype, are probably also associated with an inferior outcome. Now, AML frequently relapses, unfortunately, and we still have a lot of work to do in this field as well. We often talk about the initial diagnosis and new treatment for early diagnosis, but there's a high clinical need for relapsed and refractory patients. And typically, when an AML relapses, it goes along with clonal evolution, and it also goes along with a more primitive state of the AML. And I think that's what the figure on the left shows, from more mature to more primitive at relapse. And there is two examples of this study just to show you that indeed sometimes genetic markers are insufficient to characterize AML. In the first example, you will see that indeed, we see clonal evolution at relapse with the expansion of a mutation in the FLT3 tyrosine kinase, which goes along with the increase of this immature state. But in the second example, you see that at relapse, basically, you have no driver mutation that is at least detectable by the technologies that we have. However, we still have a much more primitive state that arises. So in the second case, the change in the AML hierarchy or maturation does not correlate with clonal evolution as we know, which means that the AML hierarchy or maturation state defined by single-cell RNA sequencing indeed represents a novel layer of acute myeloid leukemia complexity that can be independent of genetic alterations. And this may be quite academic, but still, I will show you in this example that it may also have some impact on treatment. Because, as I mentioned in the Beat AML study, but also in this study from the DIG lab in Toronto, since you have single-cell RNA-seq, if you do a drug screen, you may be able to integrate both data sets. And this is exactly this, which means that when you look at the drug screen and you correlate it with the maturation state, AML, you may find intriguing results. In that case, I'm not going to discuss everything here, but the message here is that there is drugs that work much better if the AML state is mature, and on the other way around, there is drugs that work much better if the AML state is immature. And this is shown here with panobinostat, a HDAC inhibitor, which acts more in a mature state of the AML, whereas the BCL2 inhibitor venetoclax, which we frequently use these days in acute myeloid leukemia, much more active in a more immature AML state. And we can take this a little bit further and look at specific mutations in AML, like the mutation in FLT3. And this shows you that an FLT3 inhibitor like sorafenib indeed is much more active in an immature AML maturation state, whereas it's quite inactive in a more mature AML maturation state, although the FLT3 mutation is there. Which means that the sensitivity to sorafenib and to multiple other drugs may not only depend just on the presence of a mutation, but indeed also on the maturation state of the AML populations you're looking at. So I think we are still learning a lot from this, and this is by far not something that is at the moment available for clinical routine, but this is something to keep in mind. If your patient with an FLT3 mutation may not respond to your drug, there may be other factors that may influence this. than the genetic alterations. Now, this is just to summarize this, that you can characterize the AML hierarchy by single-cell RNA sequencing, that genetic mutations are sometimes insufficient to predict response, and that some drugs may indeed act depending on the maturation state which is present in this disease. Now, I will switch gears and talk about the acute myeloid leukemia tumor profiler, which is a multi-omics study we performed here. And this was the work of a whole consortium. And this was also done in a similar fashion for melanoma and ovarian cancers. And both other so-called marker papers are also in the process of being published. And we performed this study in acute myeloid leukemia patients who were resistant or refractory to their treatment. And I would like to highlight that this, again, was really the work of a whole consortium, and that I think really every part of this consortium was critical in developing this project. And the melanoma study and the ovarian cancer study were the first ones to be part of the tumor profiler. The AML study was the last one, which also means that we had a shorter time to recruit the patients. So we ended up with 21 patients, which is, of course, a limited number. In some patients, we had access to longitudinal samples, so we were able to do follow-ups with the different technologies that were available. And in particular, we used two different drug screening approaches. We used single-cell RNA sequencing, CyTOF, also bulk proteomics to characterize these patients in an unbiased fashion. We also looked at the sample composition, which may also reflect what I talked about before about the maturation state. And since these are all patients from our department, we had access to all the clinical data and the genetic data, and this is just a matrix that summarizes this. One of the key technologies, and I think someone talked about it at lunch, I think, key technologies that was really crucial for this project was pharmacoscopy, and probably a lot of you know what pharmacoscopy is. I will just try to summarize this. This technology was developed by Bernd Snyder at the ETH. It's a drug screen that also allows visualization of cells by multiplex staining, for example, and it's an automated microscope that reads the data, which is then again integrated by multiple different approaches. So you can of course look at cell death, the likelihood of response of a specific drug. The machine can also distinguish healthy cells from tumor cells because it has learned about the morphology, also particularly about acute myeloid leukemia cells. But since it's a microscope, it can also look at the interactions between the tumor cells and the immune environment. And I think this is very useful not only in acute myeloid leukemia, but also, for example, in solid tumors and other diseases. And this is another matrix which shows the ex vivo drug response in the landscape of relapsed refractory acute myeloid leukemia. We used 79 drugs that had been used in the past as a library: chemotherapies, targeted therapies, no immune therapies. And the most significant association we found between the clinical response and the response on the in the ex vivo drug screen was an association with venetoclax sensitivity. So again, the BCL2 inhibitor that I already mentioned. And we could clearly see that patients who had been exposed to venetoclax basically had no response to venetoclax ex vivo, whereas patients who were venetoclax naive, most of them responded. There is some that were also resistant without being exposed to the drug ahead. Now, venetoclax, as I mentioned, is a BCL2 inhibitor. BCL2 is a anti-apoptotic protein. Venetoclax induces apoptosis by displacing Box and Buck in the mitochondrium and leads to the death of AML cells. And in this figure, you see also multiple other cellular targets for precision medicine in acute myeloid leukemia. Some are still being developed, some have been abandoned. Unfortunately, also the anti-CD47 antibody magrolimab, which was also a focus of interest of our lab. So, you know, there will be new drugs, some may work, some may not. So it's always very difficult to predict which ones will make it to a clinical, to the clinical practice. This is the acute myeloid leukemia treatment algorithm, a bit simplified, but it's about what we would do these days in the clinic. So unless we have a clinical trial, you stratify patients into fit patients and unfit patients, and the patients that are unfit or ineligible for an intensive therapy, they will get, in most of the cases, unless they have a mutation in the isocitrate dehydrogenase 1, in most of the case, they will indeed receive venetoclax plus a hypomethylating agent. And the reason for this is that this combination leads to a significant survival advantage over hypomethylating agents as a monotherapy. As you can see here from this pivotal landmark study, the VIALE-A trial. And however, most of these patients at some point will relapse, as you can see from the survival curves. Most of the long-term survivors indeed are patients who have been transplanted with an allogeneic stem cell graft. That's why venetoclax resistant is area of clinical need. And since we had this nice correlation in the tumor profile study I showed you before, the correlation between the clinical resistant and the ex vivo resistant by pharmacoscopy, and since we had all the data from all the other technologies, I'm going back to integration. So we were able to look a little bit more into this venetoclax resistant setting. Again, looking at the patients who were exposed to venetoclax, these are these ones, and you can see that you have barely any response ex vivo. And it's obviously different in the ones who had not received venetoclax. But you see that there is some patients who are resistant to venetoclax, and we would call this an innate resistant because they had not seen the drug before. In the venetoclax exposed patients, we see typically a downregulation of its target of BCL2 that's well known and described in the literature. So that's just a proof that our approach also works. But we were also able to look at other markers in an unbiased way. And we found that, for example, CD36, which is a fatty acid receptor, is upregulated in the setting of venetoclax resistance. And this was the case by single-cell RNA sequencing, but since we also had site-of data, we could also confirm this there. But I agree that it's always a few, it's not many patients, but still we were surprised to see these correlations. When we look at the t-SNE plot and look at patients who were exposed to venetoclax or who were not exposed to venetoclax, I highlighted them here, we could see again that their response ex vivo to venetoclax was poor, but was similarly poor. So there was no difference between the ones who had a treatment-related resistance nor innate resistance. And this was also the case for the CD36 expression. So venetoclax resistance, whether it's innate or treatment-related, goes along with similar molecular features, and one of them could be or is CD36 upregulation. And to round up this story, the roundup will come just afterwards, but we also confirmed that CD36 upregulation also confers poor prognosis. And since there is a lot of in silico data also available, we confirmed that also the lack of response to venetoclax ex vivo correlates with high 36 expression in 2 independent studies. Now, the rounding up came with indeed this immunotherapeutic approach. We then tried with 7 AML samples with various levels of CD36. And so we basically took the mononuclear cells that we had from these patients, since we systematically banked these samples and froze them down. So we had still some leftover samples. And we took these samples and added the CD36 antibody. And we were able, as I said before, to look at cell death, but also at spatial organization. And this image shows you that if you use the isotype, obviously not much happens. But in the case, if you add the 36 antibody in a sample that has a high 36 expression, you see the clumping of immune cells around the AML cells. And we were also able to quantify this again with a limited number of samples and show that the AML cell killing really happens only in the 36 high expressing cells, and this in a dose dependent manner, whereas in the CD36 low cells, low AMLs, the antibody didn't do much. And needless to say that the killing also went along with increased interaction with T cells and monocytes in the presence of the antibody and high 36 expression, and this was not the case with low expression of 36. So to summarize this part, we use this unbiased multi-omics approach in relapse refractory AML samples, and we're able to identify novel and known mechanisms of Ven resistance. Both the innate and the treatment-related resistance to venetoclax follow the same pathogenetic mechanisms, and CD36 may represent a novel therapeutic target in Ven-resistant AML patients, whether you use an antibody or other approaches to target this pathway. I will end with 2 brief slides. One slide is to introduce, for those who don't know, the clinical trial program we have here at the Comprehensive Cancer Center in Zurich, a program which encompasses 10 study units, including the Kinderspital, where we try to bring these units closer together and get an overview of the trial portfolio and harmonize processes within these study units to make them more efficient. And we also work quite a bit on the outreach, and that's what I'm doing here. And you can also see all the trials that are running, interventional trial, diagnostic trials, if you want to use the QR code or the address that I'm showing you here. Now, more relevant to what I've showed you before today is that we use the tumor profiler to set up a clinical trial, and with the support of the personalized health related technologies from ETH, we were funded for this. And I think this is very, very interesting times for AML, and particularly relapse refractory AML. These patients would sign informed consent. They will undergo pharmacoscopy. We will do pharmacoscopy if the method works. So if we have the quality is good, we will then randomize the patients, and one group, the control group, will be treated with a standard salvage therapy, as shown on the top right. And the other group, in the other group, the physicians in charge will receive the pharmacoscopy data. So the control group will not receive the pharmacoscopy data, but the PCY group will receive the pharmacoscopy data. And it doesn't mean that the physicians have then to use the treatment suggested by pharmacoscopy, but they have this as an additional data set to maybe make a decision on the treatment they want to give to the patients. In contrast to the tumor profiler, we were limited here a bit more with the drugs, since Swissmedic had to approve the trial, and they are basically approved only drugs that are indeed AML indicated and reimbursable. And we have short endpoints, complete remission at day 28 and multiple secondary endpoints. And in the meantime, this trial is also running at the INTeRCePT. And we have, I think, included about 10 patients at the moment, but of course, no data from the trial so far. And with this, I would like to thank you, and I'm happy to discuss AML and so on.
Thank you, Alex, for the great presentation. There is a question from Guckenberger.
Thanks, Alex. I'm still rearing from the talk of Valentina earlier, which begs the question right now. You're looking at multiple modalities, for instance, in your second part of your presentation. Which of them are useful and which ain't?
Yeah.
Did you test that? And if you haven't, can you test it?
Well, I mean, if you're asking me, I think single-cell RNA sequencing and drug screens were useful for this study. If I could pick two, just like this, we are not systematically testing this or don't have an unbiased way of looking at this, which one is very useful or not. But I could say that they could be technically useful. They work technically, that's a criteria. I think, again, the single-cell RNA sequencing and the pharmacoscopy data we had from Berend were just very reliable and worked, I think, more than 95% of the patients. If you look at, for example, bulk proteomics, you need a large proportion of blasts in the blood, which often you don't have. And so these bulk approaches, whether it's bulk proteomics or bulk RNA-seq, are not very useful, just because the tumor size varies and sometimes is too small.
Maybe I didn't ask the question well enough. I mean, I understand that technically things work and that even you may get a scientific output, but since we're talking about precision oncology, in the end, patient outcomes need to be at least put into the equation. And I think that's what we heard earlier. So if it hasn't been done yet, I would strongly urge to make that effort and to just verify that you not only, I'm all for getting new academic data. I think that's hypothesis building,
I got your point. So I think from the earlier studies, so in the first part, I think they also derived from the data they had from the drug screens and the RNA sequencing, they derived the 7-gene signature. Based on this, that they then correlated retrospectively and showed that indeed they had like 7 markers that were then predictive of response, also for certain drugs. And I think if you do that prospectively, then you will have the answer to your question.
Okay, that is Thorsten.
So thanks a lot, Alex. So for the RAPID trial, why did you allow different treatments in the 2 arms? Because I understand that Swissmedic asked you in the investigational arm only let the allow choice from things that would be in label, but in the standard arm you have much fewer options.
So I think in the standard arm, I put combinations, and I think in the other one, it's just depicted, we have the single drugs depicted. So I think we have access to the same in both arms. Yeah, we can use combinations in both.
Thank you, Alex, for this nice presentation. This upregulation of CD36 that you observed in the venetoclax-resistant patients, is this specific to venetoclax, or is this a general resistance mechanism?
Yeah. So we have the strongest association with venetoclax, ex vivo response and clinical response. This, we didn't have such a strong association with other drugs. So with this limited number of patients, I wouldn't be able to tell whether 36 high also is, let's say, a marker of resistance for other therapies. Could well be what in the retrospective analysis, in the reanalysis from other data sets, we could see that 36 high levels were indeed associated with poor outcomes. So could be that it also relevant for other drugs. On the other hand, I think it's a fatty acid. It has some role in fatty acid metabolism and also other functions, which may indeed suggest that there is an association, a stronger association with venetoclax.
Thank you.
Staying. Oh, I have also a question, but.
Hi, very nice talk. So I'm curious about this rapid trial and the clinicians, and they can choose if they want to use the functional data or not. What happens if they don't choose it?
If I don't, okay, yeah. So
or have the clinicians bought in? Are they excited about the trial?
Yeah. Of course, I mean, I think, we have situations where indeed a patient has received chemotherapy and we would just suggest another chemotherapy for relapse. And with this drug screening approach, we may suggest a non-chemotherapy approach, which you may not give as a standard of care. And I think we have defined an algorithm, basically, to integrate the pharmacoscopy data in our decisions. So we always take into account what would we do without pharmacoscopy, and then we check what pharmacoscopy tells us. And in the best case, maybe this matches. In some cases, there is discrepancies, but we have a team that tries to do this in a structured way. No.
So good. Thank you, Alex. The last question from my side. You are too quick. Yeah, but it's short. So basically, do you think that pharmacoscopy could be also helpful in generating hypotheses? So you made the example of CD36, but also the example of sorafenib was nice because you said basically all the cells express the FLT3, but the ones that are less mature are basically less responsive. So can you generate hypotheses that could help then a treatment according to precision oncology?
Yeah, I think again, coming back to the integration of data, I think integration of these functional tests with single cell RNA sequencing, for example, and maybe also other technologies, is definitely hypothesis generating. I mean, we had CD36 was just one example, but we also had, for example, papolo-like kinase 1 as one target in the venetoclax resistant setting, and then you can of course look at a lot of biology with the same approach.
Okay, thank you very much, Alex. So as the last speaker, it's an honor to introduce Professor Andreas Moor from ETH in Basel, and he's going to talk about targeting cellular interaction in the tumor microenvironment for therapeutic intervention.
Thank you very much, Chiara, for the kind introduction, and thanks, Thorsten, for the invitation to this beautiful day. So I will talk about solid tumors, so a little bit change of gears. And I want to start with this provocative slide that some of you have seen already, which shows you that if you were unfortunate enough to be diagnosed with a new cancer and go to a clinic in the US in 2020, that there was only about a 13% chance that you were eligible for one of the genome-targeted drugs that are available. So there's a large fraction of patients for whom we simply do not have such inhibitors, and there's a big unmet clinical need for the identification of novel targets. I will focus on metastasis today. So you're all aware that you might have a primary tumor. This will be hopefully detected early and then resected, and then you might get adjuvant chemotherapy or targeted therapy. But many of these disseminated metastatic cells might remain somewhere in the body, and they can then later on grow and form a full-blown metastasis that ultimately could kill the patient. And this process is responsible for about 90% of cancer death, at least in solid cancers. And unfortunately, there's no specific therapy which acts specifically on metastasis as opposed to the primary tumor. Now, if you think of metastasis, here I'm looking at colon cancer, where you see this here in the colon, we have this green tumor that is forming. if the tumor is getting big enough, there might be single cells that are escaping from the tumor that might enter the blood supply. And anatomically, they will then end up likely and be captured in the liver, because that's going to be the next capillary net. And if these single cells arrive in the liver, they can then have multiple options. So it's a very, very inefficient process. The majority of them is not going to be surviving, but some of them might grow and form a micrometastasis. Other ones might be cleared by the immune system, and even further ones might just stay dormant for decades until they're maybe reactivated because there's an inflammatory stimulus in the organism and so on. And what I think is very important is that the decision-making process that decides the fate of the metastatic cell is very much influenced by cell-cell interactions, as we have nicely heard today by Judith. So cell interactions, meaning the host organ, the liver in this case, are crucial in defining where the metastasis is going. Now, targeting cellular interactions has led to huge, very meaningful clinical benefits. And here you see the interaction between a T cell and a cancer cell based on the expression of PD-1 and PD-L1. And if you block this with a checkpoint blocker, with an antibody, you can then reactivate the T cell and kill the cancer cell. And this has led to very long-lasting benefits in previously incurable diseases such as advanced melanoma. This was, of course, rewarded with the Nobel Prize a few years ago, but we still lack a systematic way of identifying cell interactions and of functionally testing them. Because of course, cell interactions might occur, but just be correlated to a phenotype. So my lab hypothesizes that cell interactions are very important for tumorigenesis. And here you see a picture of a colon cancer where you see this very big spatial heterogeneity. You have necrotic areas in the middle, you have areas that almost look like a normal colon epithelium, and you have invasive areas and so on. So this is a very complicated ecosystem with very many different cell types. And all of these cell types can communicate with each other, and we hypothesize that that can fuel tumor progression and metastasis. So what do I mean by interactions? Well, of course, a cell can express a ligand, as you see here in red, that can then interact with a receptor in yellow in a neighboring cell, so paracrine signaling. We're not so much interested in long-range endocrine signaling for the purpose of this study. And thanks to the revolution in single-cell genomics that we have experienced in the last decade, we can now use single-cell transcriptomic data sets to predict which kind of cells are communicating. So you have these databases, CellPhoneDB, we've heard about NicheNet, niches, and so on. And you basically look at your single-cell clusters, and you look in the cluster 1 to see if a given ligand is expressed, and in another cluster if the cognate receptor is expressed. And if both of the components are there, putatively they might interact, but if one of the components is missing, then these two cell types are not interacting. That sounds very nice and straightforward. And during my postdoc in the Itzkovich lab at the Weizmann Institute, we did this for colon cancer liver metastasis. So here you see a single cell data set. Every dot is a cell. And this is a human liver metastasis. So we have normal hepatocytes, tumor cells, and a lot of microenvironment. Now, if you now do this ligand receptor prediction based on a database, you end up with thousands and thousands of putative interactions. So this is not really actionable for functional testing in the lab because you don't really know where to start. There was a very elegant way to address this problem that was proposed by Martin Guillaume, and he argued, well, if an interaction is important, then it's likely conserved by evolution. So you can take 10 suspect interactions and look, are they conserved across, let's say, 5, 6 model systems? And maybe you're lucky and there's only one that is conserved in all of them, and this might then explain your phenotype. Now, this is not really possible in our area of interest, in human colon cancer, so we argue that space might be important. Because if you look at paracrine interactions, this is a drastic simplification, but you can say, most of the paracrine interactions happen in a finite space of roughly 100 to 200 to 300 micrometers. So we say we need methods that give us this diameter of 200 to 300 micrometers in terms of the neighborhood. and single cell resolution. So how do we do this? This is now a human sample, so a liver metastasis of colon cancer. You see on the bottom you have the metastasis, on top you have the adjacent liver tissue. And we are interested now at interactions that occur exactly at this interface. So not in distant regions of the tissue where they might never see each other, but that are actually spatially confined to the region where they might matter. So in this case, we have used an older array-based technology, Visium, not HD, but the classical Visium, to get basically the gene expression in space. And then we run this ligand receptor prediction specifically at the interface. And now we come up with a few dozen of interactions that occur between hepatocytes and adjacent metastases, as opposed to hundreds or thousands before. So we've started with a long list and reduced it to a shorter list. But it's still a descriptive endeavor, right? It's just a correlative list. So now, how can we prove that these are actually important in deciding if the metastasis might successfully grow out? Well, for this purpose, we need to perturb. We need to switch into a model system that we can perturb, which is in our case a mouse model. So we established a mouse model of liver metastasis by using intestinal organoids. So these are, of course, in vitro cultures that we can control, mutate, and so on. So these organoids are from syngeneic mice, and they have APC-P53, KRAS, and SMAD4 mutations, which are mutations that you often find in human colon cancer patients. And if you inject them now into the spleen of a mouse, they go through this freeway of the portal vein and end up in the liver, and they form micromets that are visible within about 2 weeks. So a very rapid model of liver metastasis. And if you look at the histology of these, they look like adenocarcinoma. So you see these glandular structures, very different from normal traditional 2D cell lines. But they're still in vitro models, so we can engineer them. So now we introduced a proximity labeler. This is the so-called SLP cherry from the Ilaria Malanki lab at the Crick Institute. And this is basically a fluorophore that is secreted and cell penetrating. So the tumor that you see here expresses this very strongly, and it goes into the neighboring hepatocytes and labels them. And that means we can basically use flow cytometry to sort out hepatocytes that were close to the met and hepatocytes that were far away from the metastasis. Good, so that's a tumor model. Now we need the perturbation model. And the perturbation model, usually when you want to perturb many things in cancer, you take a cancer cell line and you put in a CRISPR library and you transplant it. But if you do this, you're of course affected by a bottleneck effect, which means the diversity of your library is going to be massively reduced if you transplant it. And this is irrespective of the perturbation, but mostly stochastic. And of course, we are interested in cell extrinsic effects, but here you mostly get cell intrinsic effects. So we said, let's not perturb the cancer, but let's perturb the host instead. And for that, we call this now a mosaic liver. So we basically introduce different perturbations in different areas of the liver. And then we let our metastatic cell come in green. And maybe by chance, it ends up next to a perturbation which is favorable for its outgrowth, and it will then form a metastasis. But if it's unlucky and ends up next to a perturbation which is incompatible with outgrowth, it will be depleted and not growing. So how do we do this in the lab? This is basically a CRISPR-A approach, so a CRISPR overexpression mouse. So we introduce transposon vectors that carry guide RNAs with a sleeping beauty transposase by doing hydrodynamic tailwind injections into the mouse. And they are then stably integrated into the genome of hepatocytes mostly. And we do this in the context of a mouse that has this catalytically dead Cas9, which is fused to a transcriptional activator domain, so CRISPR-A. And CRISPR-A is only activated in hepatocytes because there's also an albumin Cree in this mouse model. And we do this, of course, not with any random gene, but we take exactly these ligand receptors that we predicted to be active at the interface that I showed you a few slides earlier. How does this look like? This is one gene, glutamine synthetase, and you see this is usually expressed in one layer of hepatocytes around the central vein. This is a non-targeting control. Now we put the guide RNA against this gene, glutamine synthetase, and you see after 1 day, 4 days, and 6 days, we have a broad ectopic overexpression all across the liver. So now instead of doing it for one gene, in one mouse, we can do it for 300 genes using 1,000 guide RNAs, because the mouse has 100 million hepatocytes. And if we target 1 to 3% with this approach, this gives you still 1,000-fold coverage, which is good enough to look at 1,000 guide RNAs in one individual animal. So that's the perturbation model. Now we just combine these two parts, cancer model and screen. So the cancer, as I mentioned, you have these METs that label the surrounding hepatocytes, and we can use flow cytometry to identify nearby hepatocytes and far away hepatocytes. The tumor model, as just explained, going into a liver that's mosaic. So the hepatocytes overexpress different receptors and different ligands in different areas. And then we just use a sorter and we sort hepatocytes that were next to the tumor, far away from the tumor, and PCR amplify the guides to then get to the perturbation list. A lot of validation, a lot of in vitro work to show that the screen works, which I'm jumping now, and I'll just go straight away to the results. So the results are as follows. You have here, that's the genes that we frequently find overexpressed in metastasis nearby hepatocytes, so they're enriched next to the metastasis. And these are the genes that we find overexpressed far away from the metastasis. So these are likely incompatible with metastasis. And of course, we have some positive controls, some growth factors that make sense, but we're going to focus now for the second part of the talk mostly on one specific gene, which is this plexin B2 that you see up here, which is an axonal guidance molecule that hasn't been really implicated in the context of liver metastasis so far. So now to validate this, we need to now do a single gene perturbation, and I'll show you straight away the in vivo data. So now we use again a CRISPR-A mouse, but we bring in the gene now with an AAV. So now we can hit about 90% of the hepatocytes. So you see this gene, so this is now basically the GFP of the vector. So we target 90% of hepatocytes. This is the plexin, which is a surface receptor in a control setting. And here you see the overexpression after doing CRISPR-A for this plexin in hepatocytes. And now we come in with the cancer model. And now you see this is how the liver looks like in a control mouse. So 1 or 2 visible macro mets, but this is how the liver looks like when we overexpress the plexin. So the same tumor, we just modify the hepatocytes by overexpressing this plexin B2. And this is working in the context of a colon cancer model, and it's also working in the context of a syngeneic pancreatic cell line, KPC lines. What's happening? Well, if we take sections and look at these tumors, you see the size of the METs is not really changed, but what is changed is just the frequency. And when we look at the H&E images in more detail, you see that these plexin overexpressing livers seem to harbor more epithelial-looking metastases, which brought this whole concept of ENT, which brings a tumor cell from an epithelial to a mesenchymal state, and then putatively the reversal, where a mesenchymal state that arrives at a distant host organ to then form an epithelial tumor again needs to undergo a reversal of this process. And so we then stained in collaboration with the Basel lab, and Hassan is here in the room, different markers. Here you see E-cadherin and smooth muscle actin in red in the control, and you see that this is very different if we look at the plexin overexpressing liver. I'm in the single cell session, so I'll show you some single cell data as well. So then we used, we microdissected these tumors and ran single nuclei profiling with the multi-omic assay from 10x Genomics, which gives you RNA profiling and attack, so accessibility data from the same cells or same nuclei. When we did this, you see here from microdissected tumors, we get a lot of hepatocytes, but also some tumor cells. And we can then specifically focus on the tumor cell cluster. And I show you here in green, the RNA data, so plexin overexpression liver in green and control liver in gray, and now the gene, the attack data, the accessibility data. And when we now look in the accessibility data, what is different? Which transcription factor activity seems to be different? We get a strong signal for the KLF4 transcription factor. And also this now we verified with stainings, and you see here that in these tumors that arise in plexin overexpressing livers, we have a lot more KLF4 signal compared to control tumors. So likely there is this epithelialization process which seems to be necessary for the metastasis to grow out, which can be induced by hepatocyte plexin. Now I've shown you that we can induce more METs, which is of course not so interesting from a clinical point of view. Next question was, can we also do the inverse? And now we just use a conventional knockout. So this is Randy Platt's Cas9 knockout mouse, and we basically induce guide RNAs with AAV. So we create a plexin knockout liver and then come in with the metastasis. This you see now in summary. So this is a control mouse with a lot of plexin, and we titrate the system so that we get a lot of metastases. Now we target again the whole liver with AAV. The plexin is gone, and we don't get any more METs in this context. And we can also quantify this. This is all with an artificial 2-week intrasplenic model. So the reviewers asked us, does this also work if you have an orthotopic tumor that then metastasizes spontaneously to the liver? This is, of course, much more challenging, but thankfully we had experts help from the Konrad Basel lab, again, Thomas Valente and Hassan Fasilati, that established colonoscopy-guided injections of these organoids. So you see here, this is how it looks like. This is an animal colonoscope, and you can inject these cells, and they form these little bubbles after successful injection, and they grow out to form tumors. And we can do this in the context of wild-type livers, and again, plexin knockout livers. And here you see the result. So the primary tumor is there in all of these scenarios, very reproducibly. But if we look at the livers, you see that in the control liver, you get very big macroscopically visible METs, and this is not the case if we knock out plexin in the liver. So what's the ligand? Well, based on the computational database, we suspect that the semaphorenes are the ligands of these plexins. So the semaphorene would now be expressed on the cancer side. And when we looked in our spatial omic data, we saw that there's 4 different semaphorenes that are predicted to interact with plexin B2, which are expressed here. So we just did a quadruple knockdown initially, and later on with Cas12, a quadruple knockout of all 4, did then competition injections. So you basically have green knockdown organoids and gray control organoids that are just carrying the vector, and we inject these with 50-50. And then you see basically the distribution before injection, and in the liver, you basically only find, or you mostly find organoids that actually have the wild type status for these semaphorenes, indicating that it's likely one of these 4 semaphorenes which is mediating the phenotype that we're seeing. So let me summarize this part that is published. We have shown that we can use spatial transcriptomic data to prioritize which ligand receptor interactions we should focus on then with perturbation studies. We have established an in vivo CRISPR screen to study cell interactions in the liver, and we have done this now with hepatocytes, which is probably the lowest hanging fruit, but by using different CREs, we can also switch to other cell types in the tumor microenvironment. We have shown that liver-derived, hepatocyte-derived plexin seems to be a gatekeeper and can induce epithelialization in these METs, and we hope that blocking this interaction might shield the liver from incoming METs. Now, in the second part that is not published, I will talk about how we try to come up with inhibitors in academia that we can then use to specifically test this very last point. So this is now work by Jan, a very talented PhD student in the lab, and he has used a recombinant protein of plexin B2. And you see here, plexin B2 has this very large extracellular part. This is the transmembrane domain, and this is the intracellular part of the protein. And there's a specific domain, which is the interaction domain with the semaphorenes. So he expressed this domain in a recombinant manner. And we went to the core facility of the alpacas at the University of Zurich. So some of you might have seen them next to Strickhof in Irchel. And these alpacas are actually part of a core facility because in their immune system, if you vaccinate them, you can raise single heavy chain antibodies. And these can easily be converted into nanobodies. And nanobodies are small, so they penetrate the tissue quite easily, and they're easily engineerable. You can clone them, you can arrange them in bispecifics and so on. So we collaborated with the Nanobody Service Facility of the University of Zurich. We immunized an alpaca with 5 different recombinant proteins, so human version, mouse version, and some other targets. And then we ran a phage display, 3 rounds of phage display, and then we ended up with the sequences of these putative binders. And now in the lab, I'll just give you an overview of what we got. So this is basically phage display against the human protein. So these should bind the human plexin B2. So we have 7 versions that we tested. And here you see the affinity based on BLI, which are all in the lower nanomolar range. And they're binding the human plexin. And you see, we also measured them towards the murine plexin, and we have a few of them that are cross-react. And nicely, they're also with a roughly similar affinity. And now, when we look at the mouse versions, we have the opposite picture. So we have quite a few different binders that all bind in a lower nanomolar range. Now, this protein has a function in normal physiology. It's mostly important in the neural system. And when we genetically remove it in the liver, we don't see a phenotype after 4 weeks. But to be safe, we thought it might be nice to design an inhibitor that specifically works only in the liver and not in other places. How could we do this? Well, we focused on this receptor, which is the ASGP receptor, so an asialoglycoprotein receptor, which is highly expressed in hepatocytes. So each hepatocyte expresses about 1 million copies of this receptor. So it's highly expressed, it's specific to the liver, and it shuttles very frequently. So when you engage with this receptor, it's then internalized and recycled in about 15 minutes. And this receptor is important in terms of drug delivery to the liver, because there's a company called Alnilam that produces siRNAs that are linked to GalNAcs. And the GalNAc interacts with this receptor, and you can thereby target siRNAs specifically to the liver. And there is about 4 or 5 approved drugs on the market which work exactly with this mechanism. So this is safe and established. So we said, we will use a slightly different strategy, but make use of this receptor. So we took our nanobodies here in red and bound them to an endotag. This is basically a peptide binder that was published by the lab of the Nobel Prize winner David Baker last fall. And this engages as a binder with this ASGPR receptor. So now we have something like a bispecific antibody, partially binds plexin B2 and partially binds ASGPR. And if this binds, the whole thing is internalized and the plexin is then going into the proteasome and being degraded. So it is a degrader. We convert our binding nanobody into degradation. And since ASGPR is specifically expressed on the liver, it should be liver specific. So in the last 2 or 3 slides, I just tried to show you some very early data of how this behaves in our hands. Here you see now a test on a murine hepatocyte cell line. So untreated, that's the amount of plexin B2 protein that we see in the flow cytometer. Now we use the nanobody alone. So this is just a binder. So you see the binder doesn't do anything to the expression level of the protein. This is now the endotag. So this is the one that the bispecific version. And as a control, we have now a different nanobody which came out of our screen but failed to bind. So this is now a non-binder. So you see only in the control where we have the binder with the hepatocyte degradation tag, we get a reduction to roughly 70–80% reduction of the surface protein. The same analogously is true for a human version to a human hepatocyte. And now we look at the dynamics. So you see in vitro how long in hours does this take, and you see after like 5-6 hours, we see already a very strong reduction of the protein. And this is after a single injection of this, a single exposure to this nanobody or bispecific antibody. And last but not least, this is data that is only a few weeks old. This is the first in vivo experiments. So now we have a nanobody that is cross-reactive to human and mouse, which is very nice for testing it in our syngeneic mouse model. Here you see no injection, how much plexin we have on the hepatocytes. And these are 2 expression levels where we can also, 5 hours after the injection, go down about 60 to 70% of the protein level. And now we're going to test this further. So to summarize this part, we have expressed and purified many of these nanobodies, and many of them bind in a 1 to 20 nanomolar affinity. We have some blocking phenotypes in vitro, but now we're pursuing the binding ones mostly. And we have some lead molecules that have this species cross-reactivity. What is ongoing is PK/PD and safety studies in the mouse. And in the next month, we have planned the first real experiment where we then study if we can have a preventative effect on the metastasis model by intrasplantic injection. And in the very medium or long term, we hope to see this in the context of a colon cancer patient. And we hope that we can kind of block this protein in the high seeding stages, so around surgery and adjuvant therapy setting, maybe in patients that have, persistent circulating tumor DNA or other high-risk profiles after surgery. So let me summarize. This is our vision. We start with patient material. We then perform spatial omics, single-cell analysis to predict cell-cell interactions that might be crucial, and then hope to develop such inhibitors in academia to ultimately go back to the patient. With that, I'd like to thank the people that did all the work, thank the funding sources, and all of you for your attention. I'm happy to take questions.
Beautiful stuff. So the bispecific nanobody seems to be working very well, but how about just using the RNA delivery technology to deliver plexin siRNA? That would be kind of more trivial.
That's an excellent question. We didn't have the synthesis capacity and the breadth in the lab to test that yet, but we'd love to test that in the future. And we're looking forward to have collaborators in the chemistry area that make this possible, especially since it's safe for existing targets.
Am I next? Yeah, very cool. So I actually have a question to the very early parts of your project when you do the spatial transcriptomics and you identify these ligand receptor targets. Of course, you find a lot of them, right? And then how do you, in the end, how did you decide to go for this one specifically? Is there some algorithm or was there some luck or some screening involved? How did you end up?
So the spatial analysis shortened it to roughly dozens, and this is small enough to go into in vivo CRISPR screen. And from there, we still have 5 to 10 that give a solid phenotype in both directions. And then, which I didn't show you, we established an in vitro co-culture system where we're perturbing and measuring effects in vitro. It has the benefit that it's a direct cell interaction. Many of these effects are likely mediated by the immune system, so 3 cells are involved. But if you in vitro model it only with a hepatocyte and an organoid, it's only these 2. So this was rather complicated to establish. I think modeling cell-cell interactions in vitro is still quite challenging, but we use this and we're also trying to make this more efficient so that we could also pre-screen in vitro and before going in vivo to decide maybe on a more informed basis which screens to go in vivo with.
And this plexin was specifically upregulated in the patients with the liver metastasis, or you don't care about this?
I mean, we do care about it. I think we think it's an interesting target regardless. Mm-hmm. We see that there is heterogeneity of plexin expression in the normal liver. It's zonated. We don't really know yet if higher plexin predisposes to earlier metastasis because nobody measures liver expression. What we know that the ligands, so the semaphoranes in the primary tumor, they're correlated. At least 3 of the 4 are correlated with earlier time to liver metastasis.
Cool, thanks.
Are there any other questions?
Hi, thank you for the nice talk.
I was wondering why you decided to block plexing and not semaphoring on the tumor cells?
It's an excellent question. So there's several semaphorins being expressed on the tumor side, and I think the tumors are genomically unstable and prone to develop resistance. So we thought it might be attractive to block something on a hepatocyte that is stable in terms of its genome. But of course, maybe one can do both and have a better therapeutic window. This is just very early days at the moment for this strategy.
Okay, thank you very much. If there are no other questions, we can finish this session. Thank you very much to all the speakers. It was fantastic discussion, and also to the audience. And I think there will be another coffee break.
I think we should get started. It's 3:45, so we are approaching the end of this very interesting and very insightful symposium, but not before we are having our last session, and this last session will be focused on my favorite gene and protein, and that is p53. And we have two really excellent speakers in this session, and it's my pleasure now to introduce our first speaker. This is Professor Kasper Karlsson from the Karolinska Institute in Stockholm in Sweden. Professor Karlsson is a systems biologist and is using human genetics and functional genomics, and also what I find very interesting, also organoid assays and experiments to understand tumor evolution and clonal evolution. And I think one of the genes that he's focusing on is TP53. And he will talk about genomic evolution in p53 mutant pre-malignancies. And I think we can all look very much forward to a very exciting talk. Thank you very much. This stage is all yours.
Well, thank you very much for that introduction. Let's see if this works. Oh, there's Mas. Yes. Okay, so first of all, thank you very much for the invitation to come here, and I've had a very nice time. It's been some great talks, and I will leave inspired. So as several of the previous speakers, I was not allowed to set my own title. It was kindly set by Thorsten, and it clearly places me in the p53 session. So p53 was something I studied during my postdoc, and I'm currently not studying it in my new lab, so I want to have a couple of slides in the end about current research. So the title is Deterministic Evolution in p53 mutant cancers. And this study was partly inspired by Stephen Jay Gould, who was an evolutionary biologist and popular science writer. So he once said, wind back the tape of life to the early days of the Burgess Shale, let it play again from an identical starting point, and the chance becomes vanishingly small that anything like human intelligence would grace the replay. The Burgess Shale is an excavation site in Canada where they study early fossil records. So what he's essentially saying that if we would restart life again, there's a very high chance that something like human intelligence would not develop. And that's an interesting theory. The problem with this theory is, though, that it's very hard to experimentally validate because we can't really operate on those time scales. But we were thinking perhaps we can do something similar in cancer. So if we start with the cancer, we start with normal cells, and we can start this evolutionary process here by knocking out p53 and then follow it over time up until hopefully we'll come there to transformation for the fully competent tumor cell that then later would take over in the body. And if we would be able to do this process, we would also be able to do it repeatedly to start from a very similar wild-type cell, knock out p53, and see if this process is deterministic or stochastic. And another motivation was that most of the tumor studies are studied over here when you take biopsies from the tumor where we have a full mature tumor. But no matter if you do single cell sequencing or multi-region sequencing, you will only be able to see something what happened from transformation up until the end point where you got your material. What occurs before that from the normal cell up until the transformation is essentially a black box for many tumor types. And of course, if we could understand this process better, we might be able to find some way toward malignant development. So many people use organoids to study cancer, of course, and it's usually done in a photogenetic model where you, for example, study the biology of one or two genes and how they contribute to cancer formation or cancer function. What we do is slightly different. So we study nascent evolution following this initiating hit. So we start with the primary tissue, the body of the stomach. In this case, we generate organoids. We knock out p53, as a biallelic deletion. And then we keep evolving for quite some time. So what we wanted to study is if we can, to some extent, recapitulate in vivo tumor evolution. And that would be quite interesting because the organelle system we're working with is very simplified. It's just the tumor cells. There's no other cell types in there. And the situation in the body, of course, very different, where you have nutrient flow, you have different oxygenation and so on, immune cells. But we also want to study how repeatable tumor evolution is and if we can say something about the map between the phenotype and the genotype. So we choose to study gastric cancer. It's one of the most common cancer types. And recently, 2014, there was a new classification of gastric cancer into 4 groups, and we chose to study this chromosomally unstable type, SYN, where p53 alterations are relatively common. It's also thought to occur relatively early. And that's the official story. One of the main reasons we study this, that's because I should actually study colon cancer to start with, but the postdoc there was not very helpful in getting me up to run that system. So then the postdoc studying gastric was much easier to work with. So I switched to gastric, and p53 is one of the genes that we were able to knock out relatively easily. So that's kind of the unofficial study, but fortunately those two stories align. Okay, so these were settings. We had 3 different donors. We established 3 different single-cell derived cultures from them that had CRISPR alterations. You can see the allele shifts there. And for 2 of the cultures, we also deleted APC, which is another tumor suppressor. It's relatively common in gastric cancer to see if there was a difference. There was not much of a difference. I'll just focus on P53 now. And when I started this project, I had no idea how like I was a relatively young, naive postdoc. I had no idea how long time it would take because the tumors in the body can take decades to manifest. And now I don't have decades as a postdoc if I can continue to do something else in my life. So I was very happy when I got this data and I saw in shallow whole genome sequencing that you see here that already after 55 days, we start to see something. The first alteration here in 4q. You can see over here, and then as the days in culture progress, we see increasingly complex genotype. And if we look at all the different cultures across the 3 different donors, what we could see, yes, looking at this, we could see that there's a big difference. For example, donor 1, culture 1 and 2, they look very different from each other. So from that perspective, it doesn't seem like you have the same copy number alterations that are selected for in a deterministic manner. But we could see that there are some alterations that occurred repeatedly, like 3p deletions, for example. 9p deletions occurred in 7 out of 9 cultures. We have 18q deletions and 20q amplifications that were also relatively common. So we were curious, of course, are we doing something that has some kind of resemblance to cancer, or is it just totally esoteric that we're growing in our cultures? In our in vitro culture, so what we choose to do is to look at TCGA data. So that you see on the top there are the organoids, and then we have stomach adenocarcinoma, which is what we try to model, then esophageal cancer, colorectal, rectal, which is also part of the gastrointestinal system, and then you have breast cancer and glioblastoma and pancreatic adenocarcinoma as references that should not be very similar to what we're doing. And you can see that there's some alterations that are relatively common. For example, here, 18q alterations are common, 9p relatively common, and so on. Let's keep in mind that our organoids, we had 3 donors and 3 culture per donors. It's total 9, and TCGA, there are hundreds of samples, so it cannot really be totally reflective of that. But we wanted to see some statistics around us, and the way we chose to do that, we looked at our cultures, and we look at the chromosome arms that were altered in 1 or 2 or more of our organoids, and the chromosome arms that were altered in 1 or 0 to see if there's, because if you only altered in 1, there will perhaps just a stochastic effect, whereas if you're altered in 2 or more, there's likely to be something that's in there. And then we looked at the frequency in stomach adenocarcinoma and TCGA, and we could see that there's a statistical difference. So they are also increased in frequency. The was even more pronounced in esophageal cancer, which is kind of similar to stomach adenocarcinoma. It's also p53 driven. So there's, at least on the genomic level, there's some evidence that what we're culturing is not totally stochastic, which was nice. We also observed this interesting phenomenon that some of the copy number alterations were selected for early on. For example, if you look at 3P there down to the left, it was altered in 4 out of 9 cultures, and it was altered within 200 days of culturing, and it didn't happen after that. So if the alteration didn't occur early, it was not selected for, whereas, for example, 11Q and 11P were selected for the much later stage, and they didn't contribute to this early development of the p53 alteration. Then we, of course, did single-cell sequencing on this. So we looked at an early time point, a mid time point, and a late time point. Here I show only donor 3, which was one of the well-behaving donors. So donor 1 and 3 behaved well. Donor 2 was a little bit more complicated. So what we saw here in donor 3 was that the mucin-related genes were downregulated over time, which happens in some cases in gastric cancer. And if we look at the gastric cancer-related genes that are upregulated in gastric cancer, we saw that some of them got upregulated. That included Claudin 3, Claudin 4, and Claudin 7, for example. And I must say that this is, we tried to implant these cultures after more than 2 years in culture into mice to see if they had transformed and become tumor competent, and they did not. We could not alter them. They did not grow in mice. Whereas if we had a collaborator who also knocked out ARID1A, and together with p53 and ARID1A, they started to grow. So perhaps you need something more that just doesn't happen, at least within 2 years of culture. Perhaps you have to have those 10 years to get there. If we look at the pathway alterations, they're a little bit more clear. So here we compare a late time point or a mid time point, so 300 days or 700 days, compared with an early time point, which was around 100 days in culture. And we could see upper regulation of specific pathways like TNF-alpha signaling via NF-kappa beta, hypoxia, apoptosis, and quite interestingly also p53 signaling. So you knock out p53 and then you select for clones that upregulate downstream p53 signaling, which kind of makes sense. So you have p53, it's such a chaotic thing that you do to the cells when you knock it out, and cells that somehow resembles or can restore the rewiring after that knockout effect is, according to this data, selected for. So then we also wanted to look at this in a subclone level. So I started to develop this expressed cell barcoding system where we have a random stretch of DNA that we insert to the genome of cells, and then we can track them over time. I thought that was a very clever thing to do, to express it. You can capture it in single-cell RNA sequencing, but I was not alone. It took me a year to develop this, and then when it was developed, there were 4 publications on it already. But this is what we wanted to use it for. So we made cells unique, we expanded them, we split them up into different replicates, and we followed them over time. And we wanted to see, do we see a deterministic advantage? So will the same subclone be selected for across all the different replicates? That would mean that this subclone was kind of magic. It must have been something that made it more fit already in the parent population. Whereas if you had different subclone that grows out, something happened along the way, most likely. And this is the lineage, so we can go from lineage to single cell to, and then also infer copy number status. We used INTeRCePT for that. So this is how it looks like when you split into different replicates. So we put in the barcodes at day 140 or even earlier, but then we had to expand it before we split into replicates. But we put it in as an early time point as we could, since we had a single cell to start with. It took some time to expand mass. Then we split into these different replicates, and what you see here is replicate 1 and replicate 3. They develop copy number alterations that were not present in the parent that were the same across those 3 replicates or 2 replicates. Then replicate 2 developed another one with this 8Q amplification that was different. And if we look at the barcode data, it looks like this. So you have to orient you. So you had the parent up here, and then we have the barcode, the data, the parent is down here to the left. And then it just goes along the time. So what we can see is the replicate 1 and replicate 3 is the same subclone that grows out over time, whereas there's a different subclone that takes over in replicate 2, likely because of this 8Q amplification. If you look at the other donors and cultures, we see something similar. It's mostly the red subclone here that wins out over time. So it's shows that there's more of a deterministic process. So most of this is driven that you have a very rare subclone at the parent population that has an initial fitness advantage that makes it takes over the different replicate cultures. So we want to focus in on one of these subclones. We chose donor 2, culture 2, replicate 2, and time point 2. And we did single cell RNA sequencing. The first thing we did was to look at INTeRCePT to see, can we capture the copy number alterations? Will they line up with the different subclones? And they did quite nicely. So most of them had the same copy number alterations that you can see on the bulk genome, like 3P, 3Q, 9P, 9Q alterations. And then there's one subclone here, that, for example, that doesn't have 3Q. And we also saw many subclones that you had kind of a subclone where you had a different copy number status within the barcoded subclone. So for example, this red one, the winning one, we could see that this consisted of one subclone that also had 4Q deletion and 20Q amplification. And that's the subclone that later on took over the culture and won out in this competition, and we named that 0A. And when we looked at this top upregulated genes in gastric cancer, they were indeed upregulated in 0A, but not in the other subclones. And just looking at the gastric cancer gene signature, we also saw that this 0A clone had an upregulation of gastric cancer-related genes. And it was also having an upregulation of the same pathways, like this TNF-alpha hydroxyapoptosis and P53 that we saw before when we compared mid, late, to early. And we saw the same across 3 different replicates when we grow them out to a later time point. So this was nice to see, because then that clearly shows that these pathways is something that's selected for because they are already there in that subclone. Otherwise, you could think that there's a long-term experiments, 2 years. Things can happen. We use conditioned media, for example. You could think there's a change in the conditioned media that would lead to different transcriptional phenotype, but since it's already in that subclone, it's likely that it's selected for. We could see that across a similar result across multiple different cultures that we looked at. So to sum up this part of the talk, so we have this platform for experimental evolution that we try to look at both genotype and phenotype at single cell resolution. Genomic and transcriptomic evidence shows that pre-malignant evolution, at least to some extent, can be recapitulated in vitro. And there are deterministic elements of pre-malignant evolution with the subclones that get selected for. P53 has widespread structure and copy number alterations. Didn't have time to go into that too much about the structure, but we saw that also, and with defined temporal order. And the phenotypic convergence is more constrained at the transcriptomic level compared to the genomic. So if you remember this first plot that I showed you in the beginning, the genome between these different cultures were very different, whereas the same pathways gets selected for over time. So that seems to be more constrained, and you can come to the same transcriptional phenotype having a different genetic phenotype. So for this, I would like to thank the people in Christina Curtis' lab and in Calvin Koh lab. So they were my 2 mentors at Stanford. And there's a lot of people who helped out in this study. I especially want to shout out for Maurice Presibilla. He came in as a master student and ended up a co-first author in this paper, and he did an amazing job helping me sort out all this data. So I decided to have a second title for my second part of the talk. So modeling and targeting intra-patient heterogeneity. So this is what I'm focusing on now. And it's a challenge of heterogeneity, so I thought it would be perhaps relevant also for this Precision Oncology Symposium. So we know that heterogeneity is important in cancer, and we know it's one of the drivers of relapse. And the way we as a community, I think about treatment of heterogeneity is that we need multiple different drugs to target different subclones, so drug combinations in order to overcome this heterogeneity. But then if you think about how we evaluate our drug combinations today, it's mainly by drug synergy. So we put them together, 2 drugs together, to see if they have an increased effect compared to the drugs alone. And I think one of the reasons we do that is because it's relatively easy to do, because you just put them together and you see the result. You see if you kill more cells or not. Whereas to look at heterogeneity is much more challenging, although that's one of the concepts we have in our mind on when we use drug combinations. So we want to target to have another way of defining drug combinations that look at heterogeneity. And now we have these barcoding strategies, so we can start to look at tumor subclones, and we can see which drug are targeting what subclone. And I work with neuroblastomas, a pediatric tumor type. We have neuroblastoma organoids in the labs, very simple organoids, just tumor cells again. There's no co-cultures in any ways. And we transfuse them with the same barcode, so we make them unique, we expand, we split into multiple different replicates, and instead of just having replicates over time, we treat them with different drugs. And what we want to find is something that doesn't have cross-resistance. The drug A and B here, when you treat with them, you see the same red subclone emerging, so likely they would also survive when you treat with the combination, whereas with drug B and C, there's no cross-resistance, and it's likely better drug combinations. And I must say that this concept is not developed by me to start with. I'm very inspired by a paper by Peter Sorger and Adam Palmer, who has been using this concept and showed that it kind of works in leukemic cell line, I think. And another thing in this concept also that the drugs should be individually effective, otherwise it's likely doesn't work. So you should have individually effective drugs, and then they should target orthogonal cell populations. But we quickly run into a problem because we know that patients' tumors are very heterogeneous, and what we generally use in the lab to model them are cell lines, which are known to be very homogeneous. Organoids is likely slightly better, but they're also established from usually a small part of the tumor, and then they're cultured in vitro for a long time and passaged, and likely you see a selection effect there also, so it will not be very reflective of the heterogeneity of the tumor. And if you want to study heterogeneity, you have to have a heterogeneous starting population, otherwise it does not make much sense. We want to know like how heterogeneous are our organoids, how are they more heterogeneous than cell lines, and can we find some way to induce heterogeneity, for example, by treating with a mutagenic compound like cisplatin or ENU. And how far away are we from patients? And then we come into almost the philosophical questions like what is meaningful tumor heterogeneity? What heterogeneity should we look for? Is it something like this or is it something like this? Both of them are heterogeneous. The main way we study heterogeneity is for looking at tumor subclones, which is usually defined by mutations or copy number alterations. It's relatively low resolution. Most of the time, even if you have single cell DNA sequencing, there's usually like 7 main clones or something like that. But we wanted to have a heterogeneity measurements that's based on single cell RNA sequencing, partly because single cell, the heterogeneity that we want to induce, we would induce this type of heterogeneity, right? All of the cells would be slightly different, and then over time you would reach this stage where you have some subclones that runs out. But we wanted to see if we induce our heterogeneity, how well are we doing? So then we would need a single cell based heterogeneity measurements. And several measurements have been used before to study heterogeneity in single cells, but they're usually single study choose one of these metrics and look at it. And we wanted to have a more systematic comparison which of these metrics would actually work to show or measure meaningful heterogeneity. And they show they have slightly different things that they are measuring. But how do we know what is more heterogeneous than something else? So we had to create some kind of ground truth data. And the way we did that was to look at, okay, if you have 1,000 cells from a single patient, that is likely more homogeneous than if you have 1,000 cells or 500 cells from 2 different patients. That should be more heterogeneous. And the more patients you add in, the more heterogeneous environment you should have. And this is what we use to evaluate these metrics. And this is just an example metric. So when you increase the number of samples, you increase the heterogeneity score. And then we choose to use Spearman correlation. And if you have a Spearman correlation of 1, you see this monotonic increase, which means that the more samples you include, the more, the higher the metric. So this is how it looked like when we measured heterogeneity. So coefficient of variation looked very strange. We compared 1,000, 2,000, 3,000 variable genes, and then with all genes, CV looked a little bit strange. The other ones looked kind of okay, whereas Simpson index did not perform so well. So we chose Gini coefficient because it kind of looked good. Let's see how that looks in another setting where we know that we should have something that's very clear. And then we choose to look at single cell data from cell lines and single cell data from patients. And if patients are as heterogeneous, or cell lines are as heterogeneous as patients, that then we have a problem, right? Because it cannot be, the patients must be more heterogeneous than cell lines. And what we see is that patients on an aggregate for Gini coefficients are more heterogeneous than cell lines, but some cell lines are more heterogeneous than patients. And this is clearly not good enough. So then we looked at all the different metrics to see, is there any one of them that separates patients and cell lines? And none of them do. So we had a moment of frustration there until my bright student, Ernest, a PhD student in lab, came up with a way to weigh these metrics together. So since they measure a slightly different type of heterogeneity, if we have a weighted combined metric, it's much better at capturing the heterogeneity in single cell data. And now we could see there's a clear distinction between cell lines and patients. But then I wanted to test it in another situation where we also knew of a more homogeneous setting and then a more heterogeneous setting. And this is in the paper that I just walked you through. So we have this barcode data, and we know at an early time point, we have a much more complex population here. At the later time point is more homogeneous because we have seen this clonal sweep or partial clonal sweep. So at a later time point, it should be less heterogeneous. So we're curious to see, especially since we knew that the early time point were heterogeneous because we had different copy number alterations of the different subclones. So we used our metric for single cell data at an early and late time point, and we could see that indeed the early time points were more heterogeneous than the late time points using this weighted metrics. And it's even the most heterogeneous metrics of the late time point was this setting here where we had least complete clonal sweep. So more subclones survive and you see a more heterogeneous populations. So the question then is, we have cell lines, we have patients, where are the organoids? Are they more heterogeneous than cell lines or not? And we found that they indeed were, and now we have a bit more data, so we can also see that they are more heterogeneous than cell lines, but we are not where the patients are yet. Some of this we also induced heterogeneity to try to make them more complex with INU or cisplatin, and it seems that you can induce, but we need much more data to say it conclusively, and especially to know what concentration should you have. If you treat with too much drug, then you will decrease the heterogeneity. So we're still playing around with those numbers. So and with that, I would like to thank you. This is my lab, and collaborators, and funding, and thank you for listening.
So yeah, thank you very much for this fascinating talk. Are there any questions from the audience? Maybe I can start, and it will be on the first part of your presentation. So the model was gastric cancer, and I cannot recall from the top of my head. So are TP53 mutations an initiating event in gastric cancer evolution in patients? I know from esophageal cancer this has been done. Patients have been sequenced, and actually TP53 mutation can be early mutations. But I'm not so well aware of the data from gastric cancer. I mean, it's in close proximity anatomically it will be, but is this known?
Yes, well, at least when I did a study 5 years ago, we found some papers indicating that it was a relatively early event. It might not be initiating, but it should be relatively early.
Okay, good. And I was also wondering if I can ask you. So you basically studied in your organoid cultures. I naively call this like the natural evolution of these organoid cultures. Now, in patients, they are exposed to environmental stress factors, to mutagens. Gastric cancer is associated with, smoking, obesity, and environmental cues. Have you tested this in your system, or are you further evaluating these environmental effects? Because I think this could be also very interesting to study, to see how that changes the evolutionary trajectory of these subclones under certain selective pressures.
Definitely. So yeah, that's something that's super exciting to try out. So as a postdoc, it took me around 13 hours to passage these organoids. It was my Tuesday every second week, so it's a lot of work. But as a parting gift from my PI, I helped set up a robotic system for her. So I think she's continuing these studies, and hopefully she can try out multiple more perturbations, because that might need it. Like if you induce inflammation, for example, will that be the thing that together with p53 triggers something into a real tumor?
And I'm not an organized person, so I don't know all the intricate details of how hard it is to handle those. I learned something that is indeed the case, but on the flip side, it might actually speed up the whole process because it took still quite long to observe like the full full complement of the genomic heterogeneity that you observe. So you could speed up things maybe with some addition of mutagens or sort of in vitro mimics of environmental cues that patients are exposed to. So it might be interesting.
Sure. It would have been nice. We could have saved me a year of postdoc. I think I'm extra mutant.
Okay, thanks. So I'm not going to bother you much more. So any questions from please up there.
So I also have a follow-up question regarding the first part of your talk. It was very interesting to see the deterministic evolution after the knockout of P53. I was just wondering if you have another set of patients that already have the TP53 mutation first, and then did you observe similar deterministic evolution in those patients?
Looking at patient data?
Yes.
No, we did not. There were not many great data sets, I think, at that time point to look at like which mutation occurred early and what did they lead to. So we didn't do any of those correlations with real patients' data, unfortunately.
Okay. Yeah, thank you.
But perhaps with your models it could be possible.
Yeah, I was wondering the same because if we could reconstruct the evolutionary history of different patients, then we could see for those patients with early TP53 mutations whether they have like more deterministic evolution as seen in as compared to your experimental results.
So one thing we didn't see was recurrent mutations. So no recurrent mutations. We saw recurrent copy number alterations.
We also have tools to reconstruct evolutionary history from copy number data. So it would be nice to have a follow-up talk. Yeah.
Sounds good.
Kasper, very, very nice. So you established that there's the model is not stochastic but deterministic. But so what is the determining feature? So why is it 3P or like, so do you have any speculation on what drives this sequential order of acquiring the same mutations as that you found?
Yes. So yeah, what is deterministic and what is stochastic? So in terms of mutations, not much determinism. In terms of copy number alterations, there were some recurrent, but I'm not sure if I could call that deterministic. What we saw was that if you have different subclones in that, then it was deterministic which subclone would take over the culture after a couple of hundred of days in culture. So that seems to be a deterministic feature. And in terms of pathway alterations on transcriptomic level, there also seems to be some determinism there in which pathways there are selected for. But in terms of mutations and copy number alterations, not so much.
Maybe I can follow up on the question. So to me, genomic instability and copy number alteration, I think, is a uniform feature of TP53 mutant cancers across many cancer types. Obviously, between the different cancer types, the copy number alterations are different. So the ones that you showed us in gastric cancer are completely different from the ones that we observe in leukemia. So that sort of implies that there's a functional reason for acquiring these mutations. It's not just random noise as a surrogate marker epiphenomenon of the genomic instability. The counter argument would be that in different cancer types, chromatin accessibility and just the likelihood of certain copy number alterations to occur are a consequence of the chromatin state, and it would still just be noise that's just different noise in different cancer contexts. So would your model allow to functionally interrogate and understand whether these copy number alterations actually drive the disease and the progression from this pre-malignant state to full-blown malignancy?
Perhaps if you come up with a clever experimental setup to do that. What we saw was that they were not that selected for in our model. Well, 9p deletion was common, for example, but in terms of transcriptional phenotype was much more stably selected for, which means that likely it has less effect than the copy numbers. But on the other hand, you saw some, if the 3p deletion occurred within 200 days of culture and it didn't occur afterwards, which says that it has some kind of function. It's selected for at a specific time point.
Yeah,
but yeah, don't have a good answer for that.
Any further pressing questions? Then I will hand over to Thorsten. Thank you very much for this fascinating talk. Kasper.
Thank you.
Thanks.
Yes, so it's a great pleasure for me to introduce the last speaker. I made a suggestion for a title. I forgot what I suggested, but Moshe suggested a different title, so that's perfectly fine. So Moshe comes to us from the Weizmann Institute, and I mentioned earlier that the cancer challenges are so big that it's not going to be solved by a superhero, but still you're a superhero, right? So you are one of the leaders in P53 research. So I'm very grateful that you come to us and educate us on P53 in cancer. Thanks for joining us, Moshe.
Okay, so I think the title that you suggested, Thorsten, was P53, surprisingly.
Could be.
Okay, so this is going to be mostly an overview lecture. So I apologize for those who have prior P53 knowledge. Maybe you will not find much news there. Okay, hopefully some others will learn something new. So I would like to start with a paradox. Okay, so here's the paradox. About 7 years ago, something like that, Nature had an editorial on the 10 most studied genes of all times, the 10 most popular genes. And P53, not surprisingly, perhaps, was up there, kind of lonely at the top. And this is not surprisingly, if you now do a PubMed search for P53, right, you hit 125,000 papers almost. So we know a lot about P53. I often say we know too much about P53 because part of it probably is not adding, but in fact confusing. But on the other hand, with all this knowledge, if you now send a tumor sample for a cancer gene panel, right, and very often it comes back with a P53 mutation, it falls into this category, right, of genomic findings with no reportable therapeutic options. So why is that? Why, despite all this knowledge, we still don't have any P53-based therapy? And maybe before that, I want to ask, why is P53 so popular in cancer? Not in cancer research, but in cancer itself. So I'll give you a quick overview of about P53, trying to focus only on the things that are relevant eventually for cancer therapy. And so what we know about P53? P53 is a tumor suppressor, and more than 50% of all human tumors carry TP53 mutations, some of them more, some of them less. Functionally, P53 is mainly a sequence-specific transcription factor, which means it can induce the expression of hundreds of target genes, many of them only in a restricted cell type-dependent manner, and likewise repress potentially up to several hundred other genes. And so, okay, so why is P53 so frequently mutated in cancer, inactivated, more than any other cancer-associated gene? And we think this is because P53 has this central role in receiving pretty much almost any signal that somehow could drive cancer or could characterize a cancer cell and translating it to changes in cell fate. And this is the kind of one of the typical P53 diagrams in response to a whole list. Some of that is here. Actually, one of my favorite experiments is a Japanese study in which they sent a rat into outer space, and P53, of course, was activated. But I don't think the main role of P53 is to protect us from flight outer space. Okay, anyway, so what happens is we have p53 activation. And because p53 receives signals from almost anything that is connected to cancer and responds in a way that will restrain, inhibit, or eliminate cancer cells, I'm not going to go into all these different cell types really for the sake of saving time. If you inactivate p53, many things can happen in the cell that would require an appropriate response, and they don't happen because the p53 node is taken out of the game. And therefore, the cell can proceed, go into genomic instability, whatever you want, and eventually develop into cancer. So how is p53 activated? Okay, here we have to introduce another protein, MDM2. MDM2 qualifies as an oncogene, oncoprotein. MDM2 is an E3 ubiquitin ligase, and that can bind to p53, promote p53 polyubiquitination, and p53 proteasomal degradation. The important thing is that MDM2, as biology tells us, is the main negative regulator of p53 in normal cells. There are other proteins that can shuttle p53 for degradation. There are other proteins that regulate p53, but the fact is that if you mess up MDM2, very often p53 gets out of control. So there's another interesting aspect to that. So MDM2 sends p53 proteasomal degradation, and a nice part of the crosstalk between p53 and MDM2 is that p53 as a transcription factor will transactivate the MDM2 gene, which will now make MDM2 protein, which will inhibit and send p53 for degradation. And this has the kind of, this maintains a normal equilibrium in which you have just the right amount of p53 and just the amount of MDM2 that makes sure that you have just the right amount of p53. This is when everything is fine and the cell is normal. When oncogenic stress comes in, this loop is disrupted. I think we go to the next slide. And MDM2 is taken out of the game by one way or another, by modifying p53, modifying MDM2, and by other mechanisms, so that now the balance is shifted. p53 is accumulating, but MDM2 cannot prevent its accumulation, and therefore we have p53 activation and cancer tumor suppression by one or another mechanism. As some of you know, there's still an ongoing debate of what in p53 downstream effects is really critical for tumor suppression. My bias is that there's no single downstream arm. It's probably a combination of all of them, or depending on the context and what is driving p53 activation. But in any case, p53 is activated and it does the job as a tumor suppressor. Of course, this thing can be spoiled. Let's say if you have a cell in which MDM2 is overexpressed, as happens in quite a number of tumors, let's say glioblastomas, neuroblastomas, mostly sarcomas actually is most often. You have an excess of MDM2, so you shift the balance, and even though you have signals coming in, there's so much MDM2 that p53 is kept inactive, and this could drive tumor progression without the need to touch p53 itself, without the need to mutate p53. So I think the bottom line is that in 50% of human cancers, roughly, we have p53 mutations which inactivate the tumor suppressor function of p53. And there's most probably, in the vast majority of the other 50%, p53 is wild type, but is not doing its tumor suppressor job because of one mechanism or another. MDM2 amplification, but many other mechanisms could come up in cancer cells and prevent p53 from doing its proper job. Okay, so a few more words about p53 mutations in cancer. So 50% roughly on average, but it depends on the cancer type. Some tumor types have very high p53 frequency, TP53 mutation rates. High-grade serous ovarian cancer, I think this would be highlighted here. High-grade serous ovarian cancer is essentially close to 100%. Some cancer types, including some pediatric tumors, have very low rates of p53 mutation. So mechanisms are different. Okay, Fine. Okay. So what's unique, perhaps, p53 is a tumor suppressor. What's unique about the mutations of p53 in cancer is that unlike what one sees in other tumor suppressors, so mostly what happens is that the protein is gone or truncated and practically taken out of the game. p53 mutations that you find in cancer are very often missense mutations, which means that the cells continue to produce full-length but mutant p53, and very often these mutations, the mutant protein is accumulated to high levels in the cancer cells. And okay, maybe I rush to an example and we come back to this. Okay, now we're going back. Okay, so I'll get back to that in a minute. Okay, anyway, where's it? We'll get me there. Okay, next one. Why? Okay. Okay, fine. So just to illustrate this, so this is an example that pathologists know very well. If you go into tumors that have missense p53 mutations, very often you find very strong staining of p53 in the nuclei of the cancer cells, which is indicative of massive accumulation of the mutant p53 protein, which you don't see, of course, in the surrounding normal tissue. Okay, now I want to go back to where we started. Okay. So where are the mutations occurring? Most often they occur within the DNA binding domain of p53. p53 is a transcription factor. It binds specifically to DNA sequences within the genome that have p53 binding sites. And most of the mutations that one sees in cancer occur within this DNA binding domain and eliminate the ability of p53 to bind to its canonical targets and activate their expression. Now, there are several mutations that are more common than others, and we tend to refer to them as p53 hotspot mutations. And I want to point your attention to 3 of them, mostly. The most common hotspot mutations are the position arginine 175, arginine 248, and arginine 273. And another mutation, which I mentioned briefly at the end, which I kind of call a warm spot mutation. It's not as common as these, but it has a substantial frequency nevertheless, is this tyrosine to cysteine mutation, Y220C. Okay. So what happens when the TP53 gene becomes mutated? We know the main consequence, the primary outcome of these mutations is the loss of the tumor suppressor activity of wild-type p53, probably mainly because it does not act anymore as a sequence-specific transcription factor. However, there's evidence which some people take more seriously, some people still question, but I'm a strong believer in that, that some cancer-associated p53 mutations can endow the mutant p53 protein with new oncogenic activities. This is referred to oncogenic gain of function of mutant p53, and this is definitely context dependent. The same mutation can have a gain of function in a particular context, particular cell background or particular biological context, and no gain of function mutations in another background. And I'm not going to talk about dominant negative. There was a beautiful poster outside, but I want to proceed to what is needed for the therapy part. Okay. Okay, so this you saw already. So one question that comes up, I'm going to touch very briefly on a couple of studies from our lab, just to bring in some data before we go back again to the general picture. So one question that one would ask is, are all p53 mutations equal, or are some more equal than others? And Ori Hassin, and a student in the lab, sometime PhD student in the lab, compared specifically 2 of the hotspot mutations, this arginine 175 hotspot to histidine and the 273 mutation. And what he found, just to get to the bottom line, is first of all looking at human data of colorectal cancer. Okay, I should qualify. This was done in colorectal cancer. If you look at survival data, right, this is just looking at compiled TCGA and CPTAC data. survival probability, disease-specific survival of colorectal cancer patients that have the R273 mutation and the R175 mutation, you can see right away that, I mean, having colorectal cancer is not such a good thing to begin with. But if you have a colorectal cancer with R175 mutation, you do much better than if you have an R273 mutation. And for Stefan, the R248 mutation, which is the third hotspot, behaves very similar to R273. So both of them have bad prognosis. R175 is much better prognosis. So not all P53 mutations are equal, even though all of them take out the tumor suppressor activity of P53. This is also reflected experimentally. So this was human survival data. And what Ori could show, a variety of tissue culture experiment that I will skip, in which each of those mutants conferred a different transcriptional signature on colorectal cancer cells that had been depleted of their endogenous P53 by CRISPR, reconstituted with either this mutant or that mutant. And one could show, Ori could show, that cells that have this 273H mutant are more aggressive in culture, more migratory, more invasive than cells that have this mutation. And this is also carried on in vivo. So these are two types of experiment. This is the kind of, I would say, quick and dirty experimental metastasis, experimental colonization assay, in fact, that you tailor injection of the cells and then going and counting liver mets. And these cells have GFP, so you can see them and when you excite the lungs. So here we're looking at lung metastasis. And you can see that with R273H, you get significantly more metastasis than with R175H. And this is significant. And I think the experiment that is more relevant is autotopic injection of these cells, intrathecal injection, and then waiting until the mice are waiting for 7 weeks, and then harvesting the organs of the mouse. And in this case, you get both liver metastasis and lung metastasis only with R273, but not with R175, which would be at least in line with the fact that people who have this mutation fare much worse than people who have this mutation. Okay, so just telling us that knowing which mutation is there plays a role. Okay, so all of these activities that I described until now are cell autonomous activities, right? Senescence apoptosis, I actually didn't talk much about that, but senescence, apoptosis, growth arrest, the canonical functions of P53 that most people read about in the textbook are all cell autonomous activities. But there's an increasing awareness now that P53 also has non-cell autonomous activities, actually substantial ones. And so there's evidence that P53 mutations in the cancer cell also modify the tumor microenvironment. And one manifestation of that is that P53 mutations in the cancer cells modify the tumor immune microenvironment. And again, I just want to run quickly. So what happens when P53 is lost? In this case, we're talking about loss of wild-type P53 in cancer. So a seminal study, really beautiful study that showed the way there is a study that was published quite a few years ago by Karin de Visser from NKI Amsterdam. And what they found is that in breast cancer, mouse breast cancer models, loss of P53 triggers systemic inflammation. And mechanistically, loss of P53 in cancer cells induce secretion of Wnt ligands that stimulate tumor-associated macrophages to produce IL-1 beta, driving systemic inflammation. So losing P53 in the tumor cells drives systemic inflammation. And they implicated, in this case, macrophages as mediators. We took the system and looked a little bit further. And in fact, we were interested in adipocytes because these are cells that I was happy to see that in one of the lectures today that adipocytes are taken care of and not totally disregarded, but not many people think about adipocytes as part of the tumor microenvironment. And actually in breast tissue, adipocytes are a major component of the tissue, much more than fibroblasts actually. So what Ori asked is, does loss of P53 in breast cancer cells have an influence on adjacent adipocytes? And most of the work was done in vitro setting. And what we did is he took, this is mouse pre-adipocytes, differentiated them in vitro, and did that in the presence of conditioned medium for breast cancer cells that either express wild-type P53 or been knocked out for P53. And as expected and as known from the literature, exposure to conditioned medium from cancer cells induces an inflammatory response in the adipocytes. So these adipocytes are turned into inflammatory adipocytes. That's one of the hallmarks of cancer-associated adipocytes. And this is exemplified, let's say, induction of IL-3, serum amyloid A3, which is acute response protein, which is in this system is a very strong responder. So you see the induction with wild-type P53 conditioned medium. But if you do the same experiment with conditioned medium that has been conditioned by P53 depleted breast cancer cells, you get a several-fold stronger induction of this inflammatory signature. So losing P53 in the breast cancer cells increases the capability of the cancer cells to modify adjacent adipocytes into inflammatory adipocytes, which are known to support cancer progression. Now, to show that this also has in vivo relevance, what was done by Ori and collaborators in the lab is to do a somewhat complicated experiment. I'll try to simplify it. And this is taking P53 wild type or P53 knockout breast cancer cells and injected them into mammary fat pad of syngeneic mice, either alone or in combination with adipocytes that have been conditioned in culture with conditioned medium of P53 knockout cells. So these would be kind of converted adipocytes and asking whether these P53 knockout educated adipocytes can have any effect on tumor growth. And what was found is that, in fact, when you inject the wild-type P53 or the wild-type P53 tumors, wild-type P53 tumors go a little bit less than P53 knockout tumors. But if you inject them with these preconditioned adipocytes, you enhance tumor growth, not to a massive extent, quite mildly. But what was most significant is when, actually this was Adi and the team, another student, that performed cytoanalysis of the immune compartment of these tumors. And what she found is that, first of all, if you just look at the tumors induced by wild-type cancer cells, or P53 knockout cancer cells, you see that these markers, which are indicative of monocytic myeloid-derived suppressor cells or granulocytic myeloid-derived suppressor cells. I know that not everybody likes this term, but in general, they're myeloid immunosuppressive cells, which are known to promote, to repress the immune system and promote cancer progression. So you see, to begin with, that the knockout cells by themselves induce more of those and more of those in the tumor microenvironment than compared to wild type. Just compare this to this. But if you now take those wild type cancer cells and co-inject them together with ex vivo preconditioned adipocytes, now essentially you lose the difference between the wild type and knockout. They enhance the recruitment of these myeloid-derived suppressor cells. And we believe that this is then, so we have now a three-way game, loss of wild type P53 in the cancer cells, reprograms adjacent adipocytes, which become inflammatory adipocytes, and render the tumor immune microenvironment more suppressed and therefore enable tumor growth more efficiently. Okay, I think I made, okay, so this we can skip. Okay, and very briefly, So this is just loss of wild type p53. What about mutant p53 gain of function? And again, this is a study that was done, published quite a while ago by Martino Maddalena in the lab, who is actually now a postdoc in Bellinzona. What Martino did first, he analyzed human TCGA data using a CD8 T cell signature. This is just part, he analyzed all the immune cell compartment, but what he noticed is that human pancreatic ductal adenocarcinoma tumors that have either wild type p53 or p53 nonsense or truncating mutations, which we define as not because they don't really make any mutant p53 protein, have a higher proportion of CD8 T cells than PDAC tumors that have missense mutations, suggesting that having a missense mutation has a gain of function in reducing CD8 T cell infiltration into the tumor. And going to a mouse model, you could show that this is experimentally demonstratable. And this is KPC cell lines, which was already also mentioned briefly today. So this is a cell line derived by David Tuveson from a model which has KRAS mutation and p53 mutation, giving rise to pancreatic tumors in the mouse. Cell lines established from that, injected orthotopically back into the pancreas of mice, of syngeneic mice. And what you can see, if you take the control cells and you look at the tumor that develops, there's very few CD8 T cells in that tumor. So essentially it's pretty much cold. But if you now deplete, if you knock down p53, mutant p53, right, in these cells, now you see a substantial entry of CD8 T cells into the tumor, and this is quantified here. So telling us that mutant p53, through what would appear by this definition as a gain of function, is causing the tumor microenvironment to be more cold, to be less anti-tumoral. Okay, and this has been also subsequently demonstrated also in other contexts like this paper, which shows that mutant p53 drives an immune cold tumor immune microenvironment in oral squamous cell carcinoma and several other papers along that line. Okay, this is just parenthetically. This is a paper that is still in bioRxiv, but I brought it in because it is co-authored by two very famous scientists, which have not been involved in the p53 field until now, Rakesh Jain and Phil Sharp, Nobel Prize winner. And they also report that mutant p53 has a gain of function activity through transcriptional regulation and causes an immunosuppressive environment and can impair the response to immune checkpoint inhibitors in pancreatic cancer. So this is coming up again and again. Okay, so finally, so we know all these things and we know much more. How is this knowledge being employed towards developing p53-based cancer therapies? And the short answer is it's being employed. Not successfully enough, but there's definitely lots of efforts. And this is just a kind of summary of clinical trials with p53 targeted drugs or p53 targeted agents, not only drugs. So, and as you see over the years, there have been close to 100 clinical trials. This is ending in 2022. There's a few more since then. So quite a number of p53 targeted drugs have hit the clinical trial arena. Unfortunately, none of them has still made it beyond that. So just very quickly, what kind of basic strategies are being explored for cancer therapy on a p53 basis? So we can divide the tumors roughly, right? 50% have wild type p53, but we assume that it is not doing the job because it's being kept somehow in an ineffective state. So the tumors that retain wild type p53, we would want to increase the activity of the endogenous wild type p53 that they have, but apparently is not doing a good enough job. Tumors that have lost wild type p53, so that's the other 50%, and they have, which have mutant p53 genes. One approach is simply to reintroduce wild type p53, right? They lost it, we give it back. And for tumors that overexpress missense mutant p53 proteins, the most logical approach is to restore wild-type p53 activity to the mutant p53 protein. Now, this is different than this because this takes advantage of the endogenous mutant p53, which, as you already saw, is expressed and is accumulating in high levels in the cancer cells. So if we can convert this mutant p53 functionally, not genetically, but functionally, to wild-type light and restore its wild-type activity, the cell will be confronted with a massive level of p53 activity, which will not be seen in normal tissues because normal tissues don't have these amounts of p53, and therefore one would expect that there will be massive tumor elimination. And an approach that has been explored based on the idea of gain of function. I must say that this is probably not where the field is going, but in vitro it has been shown to work using degraders. We just heard about degraders, using degraders to eliminate mutant p53 protein to get rid of this gain of function activity. So I don't know how much time I still have, or nothing, no time? Very little, okay. So, okay, maybe I'll even just run quickly without getting into showing data. So just talk briefly about each of those. So I started with reintroducing wild-type p53. Actually, this was the first approach that was taken back already in the 1990s, using gene therapy, adenovirus-based gene therapy to reintroduce wild-type p53. It went all the way to phase 3 clinical trials, and the company that was established next to MD Anderson. It went all the way to phase 3 clinical trials, but eventually in 2008, based on insufficient evidence, it was not granted FDA approval. The company appealed it. 2010, again, they didn't get FDA approval, and that was the end of this effort in Texas. However, in China, exactly the same vectors, the same strategy were exported to China, exported is a gentle term, and there it was approved. Adenovirus p53 gene therapy was approved for cancer treatment, mainly for squamous cell carcinoma, head and neck squamous cell carcinoma by intratumoral injection. And that it's been approved in around 2005. And the data is, according to what has been described there, the data is promising, but still it's unclear to what extent this is really going to be successful. I think we would like to know more about it. So some people in some parts of the world are getting p53 viral gene therapy as we speak. For restoration of wild-type p53 activity to mutant p53 proteins, there have been many efforts. The main, I think the most advanced work was done at the Karolinska or next to the Karolinska by a company called Aprea, which was based on the work of Klaus Wiemann and Galina Selovanova, developing small molecules that reactivate mutant p53. Again, very promising result, a lot of excitement. Unfortunately, a big phase 3 trial in myelodysplastic syndrome, for which people had multi-center, had big hopes, failed to reach clinical significant effect, even though there was some effect. And that pretty much collapsed the effort. All the trials with this compound, which is known as APR 246 or INTeRCePT, were discontinued. And in fact, the company is now focusing on WEE1 inhibitors and drop the p53 reactivating. And the last minute, getting back to what Thorsten mentioned, the main effort that actually has been sort of successful is increasing the activity of the endogenous wild-type p53 by targeting MDM2. And there are quite a number of MDM2 inhibitors that have been tested. Again, none of them has gone at this point beyond phase 3 trials. Many of them have not gone beyond phase 2 trials. There are many issues with that, but that seems to be the easier part. Reactivating wild-type p53 in tumors that have it is probably viewed, at least viewed, as the lowest hanging fruit. These ones are probably more challenging. So I think I'll stop here.
Thank you very much, Moshe. Questions for Moshe?
Everybody's immunosuppressed.
Well, the nut then, I mean, the strategy of inducing MDM2 and killing the cells, I mean, this works experimentally, but also clinically.
Yeah.
But you were saying that the wild type, for wild type p53, you would assume that somehow it's not doing a job, but then if you induce MDM2 and degrade p53 or induce p53, cells don't like it and die. So how do you put that into?
It's doing its job. The problem is, at least with the clinical trials that have not gone further, is that there were limiting toxicities, especially the initial observation of mostly neutropenia and almost complete thrombopenia. So apparently megakaryocytes are extremely... What happens is when you inhibit MDM2 in a non-targeted way, you will activate p53 in all cells of the body. Now, most normal cells don't really care that much about it. But apparently there are some progenitor cells, especially in bone marrow, which are very sensitive to activation of p53. And I think this is not random. I mean, this is not so exceptional that you find it in the hematopoietic system, because usually when you activate p53 in the hematopoietic cells, they will undergo cell death. If you activate p53 in, let's say, epithelial cells or fibroblasts, they will undergo growth arrest, usually senescence if you do it. So I think hematopoietic precursors are programmed to respond to p53 activation by committing suicide. And if you do it with nothing like molecule, they just feel that now they have to commit suicide, and you get toxicities that are life-threatening. So this might still be solved, but I think this is now a major challenge.
Yes, there's a question in the back.
Yeah, thank you for the nice overview. I have a question regarding the role of TP53 in the tumor microenvironment. So you've shown that in some tumors, at least if there's TP53 mutation, there's a loss of T cells. And there's been a lot of work showing that in AML, you might have the opposite, and actually people suspect that TP53 mutated group would be more responsive to T-cell engager therapies. I'm wondering if you have any opinion of this, if it's different mutations, if this makes sense at all?
I would suspect that it's not different. I don't know the formal answer. I would suspect that it's not different mutation, but it's really different tissue context that makes the difference here. And yeah, I mean, everything else will be speculation. I don't want to over-speculate. Stefan.
Stefan. I would like to get your opinion on what do you think about these immunotherapeutic approaches? So people have do HLA ligandomics and actually were able to identify the missense variant being presented in the context of MHC class 1, and then they generate, I think, bispecific antibodies. And this, I think this could be something interesting. So what's your opinion on that rather new approach?
Yeah, so I mean, this is spearheaded by Steven Rosenberg, and I think there are now other people following the same approach. Some of the mutant, I mean, essentially, a mutation in p53 is potentially generating new antigens. So on paper, that should be very, very easy. I think what Steve Rosenberg has done, in fact, is synthesized a long protein which contains all the common p53 hotspot peptides. And they show that for some of them, you can get specific T cell response. They can be presented and can be recognized as new antigens by T cells. So there's potential there. Whether it's going to work or not, it's hard to tell. I think the main concern that we have, if you target a specific TP53, we know that in at least both in hematopoietic tissue and in many epithelial tissues, you have on the ground level, you have a substantial presence of P53 mutations in apparently totally normal tissue. And you have a spectrum of mutations. It's not that you have one dominant mutation. In the tumor, you can get clonal expansion, but they're essentially, for whatever reason, the P53 gene can get very easily mutated. There's been some paper that suggested that there's something in the structure of the gene, but anyway. So it's quite likely that when you treat with any treatment which is targeted to a specific P53 mutation, there's a very good chance that you'll already very soon have taking over by pre-existing cells that have other mutations. But you know, this is something that you can predict, but it should not prevent from trying.
Okay, thank you, Moshe. We will do a very short wrap-up. Thank you all. It was a very long day, a lot of energy that leads to some tiredness, I'm sure. Thank you all for coming. I want to thank the speakers, particularly the speakers from far away, for coming. I hope you enjoyed the day as much as we enjoyed having you here. I want to thank the Comprehensive Cancer Center for hosting us. And especially Silke, who's pulling these things off, and I think it's Ariane, if I read the Nastassja. Oh, sorry. So thanks for making this work, even though everyone seemed to have a different computer. Thanks a lot. Yes, I hope you enjoyed the day as much as I did. We put together a diverse topics. So we included some physicians who kind of formulate where's the need. We have the importance of data science, a crucial bottleneck. We had a session on the technology, which helps us leverage things, but we also have the basic research with a particular focus. So the mechanism is really critical. So to me, these are the ingredients to a nice meeting, but they're also the ingredients to how we can work together to advance the field. So thanks a lot for coming, and have a nice evening.